daily living. So there's multiple clinical trial in IRD currently. I took the list from Foundation Fighting Blindness USA, which has been updated in April with several gene replacement therapy, small molecules, cell-based therapy, or pharmacological studies. I will only concentrate on gene therapy for this talk because that's where the field has progressed more. So just to remind you that what is gene therapy is the introduction of genetic material in nucleic acid in target cells in order either to cure, slow down, or prevent a disease. And there's definitely three main approaches. The most common approach that have been studied is when there's a loss of function due to a mutation, then compensate the loss of function of the protein and bringing the normal coding sequence in order to restore the normal functioning protein through either gene replacement, gene augmentation. But also when there's a mutation that is toxic, silent, or correct a genetic alteration, either through antisense oligonucleotide or CRISPR-Cas, or on the other side, to express the therapeutic protein. I already mentioned neuroprotection, promote cell survival, or gain of function, such as with the optogenetics. So we are lucky because the eye is very specific. Because you may inject something with a limited risk of dissemination. There's a so-called immune privilege, although it has its limitation, and we see when we inject foreign particles with a virus that there's still some inflammation. Retinal cells are non-dividing cells after birth, so there's no risk of diluting the transgene, and there's a limited risk of integration, so limited risk of leading to cancer. And as ophthalmologists, we are very lucky because, unlike our colleagues cardiologic or working on the liver, we have tests that are non-invasive in order to precisely document visual function and visual retinal structure. And there's, as a retina specialist, I'm still amazed of how precisely now we can go to document in vivo the structure of the retina through spectral domain CT or other things. So it really led, and Jean already mentioned that, to interesting protocols. So the work of Jean Bennett with the RP65 gene replacement therapy. And I think we need to remember these dates that I always put when I teach medical students, you know, the 19th of December 2017 for the US with approval by the FDA of the first gene therapy product for retinal dystrophies, and for us in Europe it was the 22nd of November 2018, and we were really lucky, thanks to the energy of José Sahel, to be the first center outside of the US to treat the first patient on the 5th of December 2018. Gene replacement therapy in RP65 in this very specific disease where patients have lost their visual function, but the retinal cells are still there, so there's room to restore function. Mainly on the dim-like condition, and a sustainable visual function as was documented in a paper in 2019. So a lot of hope, but also some need to follow when these drugs are approved. There was a sort of pave in the pond, as we say in French, when the group of Nagel Aaron reported some cases of chorioretinal atrophy in a patient treated with Luxturna with time. We still don't understand the full picture of these secondary changes, which may be related to the procedure, which may be related to the virus, or which may just be the natural history of the disease that continues. So there's really a need for these post-market studies to document the side effect, and we're still very happy because these changes, chorioretinal atrophy, don't usually impair the visual recovery, especially for dim light conditions. So obviously these first product led to numerous clinical trials, and Jean already mentioned some of them. We were nicely impressed by some recent results presented by Atsina Therapeutics on Gucci 2D. I forgot to pinpoint to the LCA5, but Jean already mentioned that from Optus Genetics. Johnson & Johnson Janssen is conducting a phase 3 trial for RPGR, a very serious retinal dystrophy linked to the X chromosome. So hopefully there will be some results soon, and it may be the next gene therapy product. Beacon Therapeutics also is working on the same gene, and they're launching their phase 3. So there may be some other project in the future. Jean already mentioned that, in fact, there's up and down, so numerous trials, but some of them with not good outcome. The choroideremia trial from the group, the Nightstar Biogen, which actually failed to reach the primary outcome, and a lot of discussion around that. The primary outcome was a recovery of more of at least 15 letters. And this is leading to a lot of discussion of what is the best endpoint for inherited dystrophies to show efficacy. When there's a photoreceptor disease and photoreceptors are degenerating, how come we can expect when there's no longer photoreceptor to have vision recovery? And what is actually meaningful for the patient besides having 3 lines of letter recovered or having a stabilization of their disease? And this is ongoing discussions around that. So there's other approach. Gene replacement therapy may not work if the gene is too large, or if the protein, the mutation actually lead to a dominant toxic effect. And so there's research, and Jean already presented that, and I think Mark Penesi will have a talk on that for CRISPR-Cas, with the first CRISPR-Cas in vivo trial in humans ever on a retinal dystrophy targeting this intrinsic CEP290 mutation. The phase 1/2 study results were recently published, and we are happily expecting that phase 3 will follow, but this is highly uncertain. There's also ways to use small RNA molecules, anti-antisense oligonucleotide, either to silence or promote the skipping of a part of the protein carrying mutations. So these were studies that were carried on by a Dutch company, ProQR, either to silence the deep intronic CEP290 mutation leading to severe Leber congenital amaurosis. The phase 1/2 was very promising. Then they launched a phase 3, which didn't give much of the results. And the other product is to skip exon 13 on H2A, which is a site of very frequent mutation, which was studied with a through a phase 1/2. But unfortunately, because the phase 3 for the CEP290 product was not convincing, EME asked for another study, and then ProQR decided to change gear and then concentrate on other things, looking for another partner. And after long discussion, the French company Thea decided to go on board, and now there's a very active and very motivated team with Sepal Bio continuing this work, and hopefully. So there's other approach, like I said, not gene therapy for gene replacement, but to gain a function. So for example, optogenetics will, with the principle of when photoreceptors are gone, the retina is no longer photosensitive. So by bringing another a photosensitive a gene coding for a photosensitive protein using the inner retina as a new layer photosensitive. That could be an option. This is the work led by GeneSight, for example, with some first results that were published in 2021, and there's a paper that is under preparation for the future results. Other companies are working on optogenetics, but their publications are awaiting. And then I was telling you about the fact that instead of correcting the disease, we can promote cell survivals. And this is the work that had been carried on by José Sahel and Thierry Leveillard, who passed away a year ago today, and which led to the identification of the NXNL1 genes that has 2 splice isoform RDCVF that promote glucose metabolism in cones and promote cone survival, and also RDCVFL, a longer form which is buffering the oxidative stress. And Dan Chong will mention this later, but we have the ongoing gene therapy study. So as you see, this is a very exciting time for translational research, and I hope, like the same graph as beginning with rhodopsin, and now we have 300 genes. I hope that beginning with Luxturna, at some point we'll have the same graph with so many approved drugs, but we're not still there. I think, and it was mentioned, these new trials were also made possible because of improvement on genetic testing, progress of genomic, has being better to structure our RD cohorts, to identify patients that may be potential candidates for research, and then approved treatment, and also for natural history study. We had a very nice meeting on Wednesday discussing where are still the hurdles and where the energy should be put. There's definitely more dialogue with the regulatories, but regulatories don't usually agree with each other, from FDA, EMA, the UK regulatory agency, the Japanese regulatory agency. What we want also is to better define, based on the failure of the choroideremia trial, what is actually really meaningful for patients, and probably bring patients more in the discussion with the regulatory agencies. Another thing is that we are dealing with rare disease, with subgroup of ultra rare disease, and we definitely need to build a model where when we have a treatment with very expensive clinical trial, then we need to have a model to make these treatments available for as many patients as possible. The need to improve the technology and the vector, and also the need to fund post-approval drugs in order to better document the side effects and improve the management of the patient. And I thank you for your attention.
Isabel, thank you so much, Isabel, for a lovely overview of clinical trials and all the technologies and the clinical trials ongoing right now, and many of which you are participating in yourself and are central to and doing an amazing job. So thank you so much for that overview. Thank you. Our next speaker is Mark Panisi from Oregon. I'm going to invite Mark to the stage. Mark has been leading a clinical trial on CEP290 and CRISPR-Cas editing, some of the technologies that Isabel beautifully elaborated on gene editing, and so bringing that forward. What I love about gene editing is actually that technology was found for the first time to work in mammalian cells in 2012, nothing to do with retinal degenerations. And we're already at the point where we have clinical trials. So we're seeing technologies move more quickly from academic research into clinical trial, and that's really nice to see. So we really look forward to Mark's presentation on CRISPR-Cas editing.
Thank you. It's really a pleasure to be here. So my talk will be a little technical, but before I get into it, I really want to talk about why is gene editing so important for patients. And I think it's important to think about genetic diseases a little. And if we think about a gene, it's a little bit like a recipe book to make your favorite meal. Mine is baked ziti. And we inherit these recipe books from our family, 25,000 different recipes. We get one set from our mother, one set from our father. And these make all the different proteins in the body. And mutations essentially would be a mistake in that recipe book. You could imagine that you have something added to it, like put in a pound of salt. Well, that's not going to end up with a very good recipe. Similarly, you're not going to make a very good protein. In gene replacement, the idea is that, well, you're missing 2 copies of a gene. We're going to put in a new copy. We're essentially giving you a new recipe book. And that works very well for some genes, that are very small. But some genes are quite large and complex, maybe like the Book of Kell. And so instead, we really need a different method, and that is CRISPR or gene editing. And what that allows us to do is, rather than have to put in a whole new recipe book, it allows us to go in and actually edit the recipe, almost like a word processor, where we can delete out a part, or add a part, or change a letter. And that is an extremely powerful technique that is going to expand gene therapy for many additional genes, and this is going to be very impactful for a lot of patients. So I'm going to talk about one particular disease, and we've heard of Leber congenital amaurosis, which is a very severe form of inherited retinal degeneration caused by upwards of 25 different genes. And CEP290 is one of the more common genes that can cause LCA. This is a picture of a retina from one of my patients that has CEP290. And this is a protein that's very important for photoreceptor function in the cilia. But what's really fascinating about this disease is patients are born with very severe vision loss, sometimes just light perception vision. But when we look at the structure of the retina, you can see that there are still photoreceptors there. There are still cone photoreceptors there, but they're not working. So there's actually the potential to restore vision. This is the way to do it. So we have a gene editing vector, and this is something that was developed by a company called Editas, and it uses many of the same components that we use for gene replacement therapy. It's injected underneath the retina, but it does work a little different. And we take advantage of the fact that many patients with CEP290 disease have a common mutation. And it's a mutation that occurs in an intron, which is in between coding sequences. And essentially, it creates a stop sign such that the full protein doesn't get made. And the way that Edit 101 works is this is a vector that codes for 2 different parts. It codes for 2 different guide RNAs, which are going to be a little bit like GPS targets, and a Cas protein, which is a bacterial protein that has the ability to cut DNA. So these 2 guide RNAs bind and team up with the Cas protein, and then these are then targeted to where that particular stop sign mutation is, and they basically cut it out of the DNA. And when you cut that stop sign out of the DNA, you can now make the full-length protein. So we've conducted a phase 1/2 trial. Here are all the different sites that were involved in this. And we treated the first patient back in 2020, and we've now published some of the results, and I'm going to talk about those today. So this was the original design of the trial. The original plan was to treat 18 patients. This trial was conducted entirely during COVID, which was really challenging, and ultimately we ended up treating 14 patients. These were the patients, and we started off first with patients who had very severe vision loss with a very low dose of the virus. So many of these patients were right around hand motion vision. We then went up to a little bit higher dosage, and some of the patients also had poor vision, but some of them actually had better vision as well. And in the final group of adults, we went up to the highest dose. And then finally, at the end, we treated 2 pediatric patients with the middle dose. One thing I want to point out is 2 of the patients were homozygous, meaning they had the same mutation, but many of them were heterozygous, and both groups showed benefits. So this is a breakdown of how long we have followed these patients for. This is what we've published. We've actually followed them for longer, and I can tell you that the results do continue. And we used a variety of different ways to assess success. You heard earlier about the FST, which is a test where you look into the dome, you flash a light. If the patient sees it, they hit the button. You go dimmer and dimmer and test what is the minimum amount of light they can detect. And so if we look at the patients treated in this trial, 6 out of 14 of the patients showed what was an improvement outside of just the repeatability of the test. And we actually had pretty good data as to how variable these patients were because we did a natural history study. So more than half of the patients showed an improvement in this sensitivity. What about visual acuity? When we looked at visual acuity, we saw that 4 of the patients actually had a significant improvement, which means 3 lines of vision. But some of them actually showed a dramatic improvement of vision, 1.0 logmar. That's almost 10 lines on the eye chart. So that's going from hand motion to almost being able to see the big E on the chart, which is quite phenomenal. We also did a mobility test, and this is a test that's scored from 0 to 21, very similar to what Jean Bennett described earlier, a little bit different version of it. But once again, we saw that 4 out of 14 patients had a 3-level improvement on this test. And when we look at all of the data, in red, I've shown the tests where the patients had improvement that was outside of the realm of just the variability of the test. And if we kind of take a little bit more strict criteria and say, well, how many patients had improvement in 2 or more factors, including whether or not they felt there was improvement using something called a PRO, or patient-reported outcome. 6 out of 14 patients met that even stricter criteria. So this is really exciting for such a severe disease. Now, what about adverse events? Well, we did see some inflammation in a few patients, but this was treatable. And we did see a few patients who showed some retinal deposits, which we think might be inflammation, and you can see those here on the right, also here shown on the OCT. However, these didn't really seem to impact the functional improvement in the patients, and they did seem to resolve over time or with steroid treatment. So this is still an early trial. We need to continue and test more patients. However, in conclusion, we can say that we did see functional improvement in these patients, and this does give us proof of concept that gene editing can work in vivo. And if we just think about one metric of improvement, 11 out of 14 patients had at least improvement in one of those metrics. With a little bit more strict criteria, 6 out of 14, or almost half, had some kind of an improvement. And you know, we don't expect 100% of patients to improve, especially in a phase 1/2 trial where you're starting with very advanced patients. So I think this is very encouraging for an early trial. The treatment was generally tolerated, although there was some mild inflammation. And finally, we saw effects in both homozygous and heterozygous patients. So I think this is just the very beginning of using CRISPR for a whole variety of inherited retinal degenerations, and this is really a new era where this technology is going to be adapted for many other patients. Thank you very much.
That was a lovely presentation. Thank you so much, Mark, for your work on CRISPR-Cas editing. if there are any questions in the audience on editing, on Mark's presentation. There's a roving mic somewhere, actually, I think. We have a roving mic. Would you like to stand up and come? I think we have a mic here. There's a mic just behind you. Yeah.
Thank you so much for that hopeful presentation. Is CRISPR-Cas used only to cut out parts, or can you also do stuff to replace the letter or even add a letter?
Yes. So there are many different iterations of CRISPR. So I would call this, the most basic method is just to cut something out. But then there are more advanced techniques there's something called base editing, where you can change a letter. And then there's an even more advanced technique, something called prime editing, where you can use a template such that you can actually introduce a sequence of your choice. So those are still have not been tested directly in humans, but we know in the lab that is something that is possible.
Thanks.
Thank you. Do we have any other questions? Oh yeah, the back there's.
Hi Mark, that was a lovely presentation. I'm with CRISPR Medicine News. I was wondering, since the Edit 101 obviously shows some improvements which will give a lot of hope to the people who receive the treatment, but also others, if you could comment on what will happen with Edit 101 now that the Brilliance trial has been suspended?
Mm-hmm. Well, I can't really speak for the company. They made a business decision not to continue the program, although I think now that we have published these results, I hope that they will partner with someone else, maybe to continue this.
Do we have any other questions? If not, I think we'll. Thank you so much, Mark. Thank you again for a fantastic presentation. The take-home message is, I think, that we have new tools. Gene editing is a new tool that will absolutely be applied to lots of different problems. And it's great to see those first results with a gene editing tool in patients. So we'd like to ask Leah Byrne now to come up and give her presentation. She's going to continue the theme of gene editing. Leah's a professor in Pittsburgh, and we look forward to your presentation now.
Thank you so much. I'm going to be talking about CRISPR as well, but in my talk, I'm going to be using it a little bit differently. So rather than using it to treat disease, I'm actually going to tell you about how we've been using genome editing to create new animal models that we hope will allow us to test additional gene therapies for new diseases. Before I start, I'd like to go ahead and acknowledge the people who really led all the work I'm going to tell you about here today. So Fan Huanqi came from Shanyang University in China, was a medical student in my lab, and did this amazing work. And Hamza Ueda led the work on the primate model I'm going to show you some preliminary data on today. I'm going to tell you again about how we're using CRISPR to develop new animal models, specifically of a disease called PRPF31, which is also called RP11. So for those of you who aren't familiar with this, it's a disease. It's relatively common, actually, one of the more common forms of dominant retinitis pigmentosa. It's caused by mutations in PRPF31, which is a splicing factor. So in this diagram here, you can see a red box, and that's PRPF31. It's very important for processing of mRNA, so it's a very basic step in processing mRNA that is then used to make protein. Without this protein, that mRNA processing doesn't happen, and it leads to retinal degeneration, so loss of photoreceptors and RPE cells. Like I said, it's relatively prevalent, so PRPF31 mutations account for somewhere between 2.5% to 10% of autosomal dominant RP. And what's very interesting about this disease, and I think very important in terms of developing it for clinical translation, is it's caused through haploinsufficiency. And I'll tell you a little bit more about that in the next slide, but it is sort of a naturally occurring model of why gene augmentation could be really promising for PRPF31. Yes, RP11 is caused by haploinsufficiency, and what that means basically is that if you inherit one allele, one copy of the gene that has mistakes in it, whether or not you have the disease is determined by if the other allele that you've inherited has high or low expression of that protein. So in people, the expression of PRPF31 varies by around 5-fold along a continuous distribution. So you could have high levels or you could have low levels of it. And the severity of the disease is correlated to the expression level of PRPF31. So as I said before, if you carry one copy of the mutation, whether or not you have the disease is determined by whether or not your wild-type copy is a high expressor or a low expressor. But this is really interesting, actually, because it shows you just with the information we have about the disease, that gene augmentation or providing additional PRPF31 is really likely to be able to treat this disorder. We think that it's a really promising indication. One of the obstacles in the field, however, has been that we've lacked animal models in which to study the disease and in which to show that gene augmentation actually will work. People have done a lot of amazing work in PRPF31 in a lot of different models, including in yeast and fruit flies and zebrafish. But those models are very difficult to use if you want to look at how gene augmentation will work. The best models for gene augmentation for looking at gene therapy are models in which the AAV vectors that we're using are efficient. So those are mouse models or dog models or hopefully primate models, and we haven't really had a good mouse model of PRPF31. So one problem with mice is if you knock out both, or if you mutate both copies of PRPF31, then it's lethal, so the mice do not survive. However, if you only give the mouse one mutated copy of the gene, then they have very mild forms of the disease that aren't exactly like what people have. And so what we have done is to actually use CRISPR to create new models, new mouse models that more accurately represent or model the human disease. So we did this using genome editing, using CRISPR, and so Mark already introduced kind of how CRISPR works, but it's basically a way for us to rewrite the genome. And in this case, what we're using it to do is to rewrite the genome to introduce disease into the animal model. So what we've done is to create a construct that drives expression of this bacterial protein that can cut DNA, and we also put into that construct a guide RNA that targets this pair of molecular scissors to a specific location in the genome. So in our case, we're targeting it to PRPF31, and we've designed a bunch of different guide RNAs and CRISPR constructs, in fact, to target different locations in the PRPF31 gene, and we did some testing to figure out which of those was the most efficient and identified specific guide RNAs and specific CRISPR constructs that really efficiently knocked out the PRPF31 gene. And then we used AAVs to deliver these molecular scissors into mice. And we used different methods of delivery in order to target different cell types in the eye. We used subretinal injections, where the vector was delivered under the retina. We used intravitreal, where the vector was delivered into the center of the eye. And then we used systemic injections to deliver it to the whole body of the animal. When we injected this CRISPR construct under the retina, it targets most efficiently to photoreceptors, to the cells at the back of the retina. So what we found when we injected this CRISPR construct subretinally was that we got really good editing. So we were able to get between 40 and 80% of cells with their genomes edited. And we were able to chop out kind of a big chunk of DNA that really led to very strong retinal degeneration. When we looked at the function of the retina following administration of this CRISPR construct, what we saw is that the function of the retina was really impacted. So when we looked at ERG traces, so that's the electroretinogram, it's this response of the retina to a flash of light conditions. And if you look at the retina itself, what you see is that the retina starts to look a lot like retinitis pigmentosa. So as I mentioned earlier, injected it into the center of the eye, into the intravitreal space. When you do that, the vector encounters ganglion cells on the surface of the retina first. When we injected that, it's a little bit more dispersed when you inject intravitreally, so we got lower rates of editing. Around 10% of cells were edited with this route of injection. But we did see that the retina started to get thinner, but specifically the inner part of the retina, which is the part that the vector encountered first, that starts to get thinner more so than other layers. And if you look at the retina with immunohistochemistry, if you look at a cross-section of the retina, what you see is, again, that it's those inner layers of the retina that are lost first. So we're seeing a loss of retinal ganglion cells and other cells in the inner retina. And again, if you look at the function of the retina through ERG, you see that the function of the retina is impacted, but specifically the part of the ERG trace that correlates to the inner part of the retina. So this is kind of a second model of PRPF31. It doesn't exactly mimic what we see in patients quite as well as the subretinal injections because it's targeting mostly the inner retina rather than the outer retina. And then a third approach we used was systemic injections. So PRPF31 is a protein that's expressed throughout the whole body, and so we wanted to see what would happen when we edited PRPF31 systemically. So in this case, we injected this genome editing construct through the vasculature to deliver it to the whole body. And what we found is that in following these injections, the mice were very low in weight and they had a poor survival. So they were very fragile and usually did not survive very long, which makes sense because this is a ubiquitously expressed protein. Your whole body needs it. And so this does show that, yes, the PRPF31 is required during development of the animals. We also saw that there was that there was significant editing of the genome throughout the body. I'm showing here that the genome was edited specifically in liver cells. And there was a reduction in the PRPF31 protein itself throughout the body. So again, this is another model of PRPF31 that we've created through genome editing. But this is, again, different than what we see in people because, interestingly, in people, this is a disease that's expressed specifically in the eye and does not seem to affect the other parts of the body. So it's an interesting part of PRPF31 and something that we hope we can look into further. So out of those three models, I think it's pretty clear that the subretinal injection was the one that mimicked or modeled PRPF31 RP the most closely. So then we used that to further investigate how well gene augmentation could rescue or could treat PRPF31 RP. So what we did was we injected both our knockout construct, so the construct that would edit the genome, as well as a healthy copy of the PRPF31 gene, but the second copy of PRPF31 was altered such that the CRISPR couldn't edit it anymore, so it was sort of protected from cutting. And in both eyes that we just had the knockout vector and in eyes that had the knockout plus the treatment, editing was very efficient. What we saw was that if you look at the picture of the back of the eye, in the location where the genome editing occurred, the retina starts to look thin, it starts to look pale, whereas with the rescue construct, it's protected against that degeneration. And if you look at cross-sections through the retina, 5 weeks after these two different combinations of vectors were injected, the knockout or PRPF31 diseased eyes start to get a little bit thinner, and the rescue eyes are protected against thinning. 10 weeks after injection, the difference is much more apparent. The PRPF31 eyes become quite thin, whereas the eyes treated with gene augmentation are protected from degeneration. We quantified that and showed that, that thinning was really significant. And we also looked at heat maps just to quantify that again. And in these heat maps, you can see that the gene augmentation really protects against degeneration, especially in the area right under the injection. It's also very interesting that you can see that there's both expression of the Cas9, of the genome editing tool, in cells that are still surviving. So this really shows that the gene augmentation does protect. And just, I'm going to move forward really quickly to show you that one thing we're moving on now, this is some preliminary data, is we're moving now to translate this approach into primates. So large animal models are really important for clinical translation because large animals have eyes that are very similar to humans. So I think we've all heard stories in the news about treatments not translating well from small animals like rodents into the clinic. So we feel that it's very important to create large animal models with eyes that are very similar to humans to show that these therapies are really going to work in people. And what's great about this CRISPR approach that we've developed is that it's something that can be translated directly into primates really easily. It can be very difficult to find or make large animal models because it takes a lot of time and it takes a lot of investment. But this CRISPR technique can be used in adult animals to really rapidly create large animal models of PRPF31 or potentially any other disease that might be of interest. So I think it's a really promising approach. CRISPR is a really promising approach for treating disease, but also for laying the foundations for studying a lot of different diseases. So really quickly, we use this approach to edit primate retinas in culture and show that we could edit the genome of both primate and human, actually, retinas in culture. And then we've also started to translate this into in vivo models. So here we've injected subretinally these CRISPR constructs, and the eyes got through that process very easily. And what we saw was that we were able to produce a reduction in the retinal response to light in primates as well. So in summary, we have used CRISPR to create mouse models of PRPF31 that model the main features of RP11, and gene augmentation was used in those models, and I think it shows a lot of promise for treating PRPF31. And I hope that we can continue to use the same approach in primates, and it remains to be seen if we can also rescue in the primate models, but we're working on that right now. And I just wanted to point out one limitation of this study, which was that I talked a lot about haploinsufficiency. With CRISPR, we're actually editing both copies of the gene, and so we're also moving forward with trying to create models that are more accurately model the haploinsufficiency as well. And so I think I'm a little bit over. Thank you very much. And I'm not sure if there's time for questions, but happy to answer any now or later.
Leah, thank you so much for a beautiful presentation on really focusing our attention not just to CRISPR for therapies, but CRISPR for making animal models and making animal models quickly. If there are any questions for Leah, there's a question over here. Does anybody have a mic? There's a microphone just coming. Yeah, no, so these are really exciting tools that we now have in our toolbox in genetics, which is great just to make animal models quickly.
Yes.
So the question's over there.
Thank you. Thank you for a wonderful presentation. My name is Martin Smestad. I'm from the Norwegian RP Association. We have an ongoing RP11 project in Norway where they are using patient-derived induced pluripotent stem cells to grow retinal organoids. Have you considered this as a potentially more accurate model to test your gene augmentation therapy on instead of using primate models?
Yes, we love retinal organoids. It's a really great approach. So we have actually tested gene augmentation in iPSC-derived retinal organoids in collaboration with the Institut de la Vision in Paris. And it worked. It worked quite well to rescue the phenotype in those organoids. The difference between organoids and in vivo models is that in those organoids, the photoreceptors are on the outside, so they're kind of easily accessible because they're displayed on the exterior of the organoid. And in patients and in animal models, photoreceptors are actually the most difficult to access. They're all the way at the back of the retina. And of course, there's other structural and immunological barriers in vivo that don't exist in organoids. So there's pluses and minuses to all these models. And actually, I think what we need is a combination of different models in order to thoroughly understand the disease and to thoroughly research potential treatments. And so organoids are a very important part of that. And yeah, in general, we love them. And I hope that they can be something that can accelerate translation of therapies into the clinic and something that can be used to file INDs and things like that. So yes, thank you for that question.
Do we have any other questions?
Yeah, Jane.
Yeah.
Yeah. Thank you very much. I'm a clinician, and if I understood it correctly, the expression on the wild type depends the severity, is that right, of the disease?
Yes.
So this might explain a lot in some pedigrees we've been looking at. So the question is that easily measured? I mean, can that be done commercially, or is that a hard thing to do?
Yes, so I should say that it's the most, that is our best understanding to date is, yes, it's the expression levels of the wild type allele that determine whether or not disease is actually expressed. In terms of measuring those levels in patients, I actually don't know, and I would defer to any of the clinicians in the room if there's some sort of commercially available test for that. I'm sure it could be done. I think you could always maybe get iPSC cells or skin cells or something and check for levels. I'm not really sure, okay. But yeah, I think I don't know if there's any clinician in the room who could answer that question.
I mean, it would be really, for embryonic testing, if you were able to test the embryo and know that the wild type was expressing well.
Yeah.
That would be clinically important.
Yeah, I mean, just from my basic science perspective, one of the difficulties with creating the animal model was, yes, we'd have to find some sort of breeder that was had a low expressing allele in order to pair that with one mutated copy of the gene in order to create the animal model, and that was quite difficult. So. Yes, I think that would be a really interesting approach. And again, would love to talk to any clinicians in the room that have perspective on that.
Thank you very much.
Are there any other questions for Leah? If not, thank you once again, Leah, for an absolutely beautiful presentation. Fantastic. So we've heard from Jean and Isabel and Mark and Leah a lot about different methods of gene therapy, gene replacement therapy, clinical trials, and technologies, new technologies like CRISPR-Cas editing, both for treatment and for animal models. We're going to move now to Professor David Keegan, who's homegrown as such in the Mater Hospital in Dublin. He's going to talk to us a little bit more about the patient care pathway and getting patients ready also for clinical trial.
Great. Good morning. Thank you very much, Jane, for the invitation and the organising committee for letting me come on the panel and speaking on behalf of the Target 5000 team. Also to Retina International and Fighting Blindness for putting the whole session together in a couple of days. It's a great program, and to pull the great and the good of World Retina into one room over a couple of days is an incredible feat, and I think it's testament to Jane standing in this field across the world that we can attract so many people to come here. It is a great privilege to present on behalf of Target 5000 team, which is Jane, Paul Kenna, Emma Dignan, Julie Silvestri in Belfast, we're the sort of the leads of it, and then the many others who work in it. I got to know Jackie Turner, genetic counsellor, Jim O'Byrne, clinical geneticist, and many, many others in Jane's lab and in our clinical groups across the country. We've heard a lot about the new therapies and the advances that Jean and her group have made in bringing these treatments. And my team make fun of me for making this statement on so many occasions, that what Jean and Jane and Robin and many others across the world have done is shift from an existential problem, how do we replace a malfunctioning gene, to a practical problem. How do we get it into the right patient? How do we identify the right patients? How do we get it into the right patients? How do we get people to approve it? How do we prove it's safe? How do we get it paid for? These are all practical, logistical issues. So many years ago, I abandoned my hopes of being a lead researcher in ocular stem cell or retinal cell replacement treatment and shifted gears with our group to try and lay the foundation, the platform, along with many others, that we prepare our patients for these exciting treatments that were coming down the line. And this is what we hope to do, and we sort of poached the idea from Jane and Paul, who were looking to sort of characterise about 600 patients with IRDs in Ireland, and Avril Daly, who was then CEO of Fighting Blindness, was all over that as well and sort of dragged me along. So that was the genesis of Target 5000. These are some personal financial disclosures, but really we've got to thank Novartis for some very generous support around the genetic testing that we're going to present today. So inherited generations, to this group, you know this, but it's that diagnostic odyssey. I got the privilege to do a radio program show slot with Paul Boskind yesterday, who suffers from Stargardt disease. And again, reiterated the story of the average time it takes to get a diagnosis, the number of clinicians you need to see before you get that diagnosis, and the many misdiagnoses you get along the way. And there's many patients with a vision impairment and blindness due to a genetic condition in this room, and that story is all too familiar. This is just an example of a patient with dominant retinitis pigmentosa, and the genetic heterogeneity, which you've heard about from others, so I'll move on. But the other aspect that Paul talks about in many of the patients is not just that physical limitation of vision impairment and blindness, but it's also that impact on their mental well-being, anxiety, depression, the feeling of social isolation in those affected with these conditions, but also those who look after them. So as Avril points out, and quite rightly so, it's a public health issue, because rare diseases aren't rare. They're individually rare, but they're not rare. They're quite common in a society. So bringing it back to a patient focus, and when Kirk Stevenson was a fellow with us in the Target 5000 program, we really sort of looked, we were struggling how we were going to structure this. And his paper out of Graham Black's lab really shone the way. What do patients want out of a service? Well, they want a diagnosis, prognosis, inheritance pattern so they can do family planning properly, and that's our paediatric program has been really important in this regard. They want to know about upcoming research. But they want practical supports. We talked, I think Isabel pointed that out in Paris and Oxford and Moorfields and Pittsburgh. I've been really at the cutting edge, not only of gene therapy, but also vision rehabilitation and new technologies. But we've got to help our patients with that too. And then psychological support. But really then a lot of it drills down to access to clinical trials and approved treatments. So this is essentially it. This is our pathway in a nutshell after a lot of iterations and development. And we created the low-cost pathway with Jane in the lab, initially doing research-grade testing and then variant confirmation analyses to upgrade those to a clinically accredited test that we could pass on to patients. So this is essentially it. We sucked the referrals in. We were on an electronic referral system, a database, which we're upgrading just soon. And we brought them through a clinical assessment, research grade testing and genetic testing. If they were gene negative, they now go out to a program in Jane's lab and partner with Franz Kremer's lab about looking at those intronic mutations and trying to resolve those to whole genome and whole exome sequencing. But once you get a clinically accredited test, we go through a multidisciplinary team meeting, and that's led out by Jackie Turner and Jim O'Byrne, and then the clinicians and scientists feed into that. But then we can create this multifaceted pathway for our patients about managing morbidities, cataract, macular edema. You can actually shift a patient's vision as effectively with treatments for their macular edema as you can with some of the early gene therapy treatments. It's not that we don't do them, but we've got to optimize the treatments with what we have available for now. But then obviously this is the route then for bringing patients into clinically approved treatments and therapies such as Luxturna. So we've published that work. And we've resolved now about 82% of our cases. So initially, I think in that first paper, Jane, we were looking at about 68, so 69% resolution, but just with the improved techniques and the iterations and improved phenotyping, we're getting better. Julie Sue, when she was fellow with us, sorry about all this, she developed the pediatric pathway during COVID when we couldn't bring our patients in. And we developed a WebEx system that Jackie and Julia worked on there, and we resolved a lot of cases, and we published that work. Now, this is an enhanced cohort from the first lot that we did, so this was an 85% resolution, but it was an enriched cohort. So we've now tested over 200 children. It's a slightly lower resolution rate, but again, diagnoses such as X-linked retinitis pigmentosa, achromatopsia, which treatments are on the horizon. And we look at the overall success of Target 5000 and compared to other registers across the world. We now estimate from our studies we've done in Stargardt disease and Ushers, we have a capture of about 85 to 90% of all the patients in Ireland with inherited retinal degeneration are on the books now. The Ioneer Hospital, the Mater Hospital, or Belfast with Julie. So that's a significant impact we've had over these last few years. At the Mater Hospital, I'll just speak about, they've all had accredited testing. With an 80% resolution rate, we have genetic counselling. But we also bring in the eye care team and the eye clinic liaison officer. That's our gateway to rehabilitation for these patients and access to clinical trials. And we're developing the syndromic multidisciplinary care access and pathways. We were enhancing our phenotyping, and this is from work, this slide was done up by Kirk Stevenson during his time, but just sort of looking and delving deeper into all the aspects of these patients so we can intervene in a meaningful way in their condition. The Clinical Genetics Service, and now we're members of the European Reference Network, so this service is now available, initially funded by Fighting Blindness, established in 2018. So now we have a significant pathway for resolving diagnosis, upgrading variants, and giving back into the community and contributing in that way as well. So there's just some slides from just the Clinical Genetics Services, mainly the work of Jacqueline Turner in our group. So what's the impact of that? So this just the other day, this is a really interesting case. And this highlights why we need systems like this to pull patients into our pathways and into our service. This patient was lost to our clinical care for 12 years and just recontacted Fighting Blindness and got into into our system. And this is just got inherited retinal degeneration, diagnosed as something else in his youth, classic lack of autofluorescence on autofluorescence imaging, and some residual retina left there, and he's biallelic RP65 disease. We've also published on our Ushers population across the group, and really we can sort of show, I'll move along quite quickly now, and I just want to make sure I don't get bitten on the leg. And but we're now able to identify that, again, feeding Irish information into that global knowledge base around these conditions. The same for Stargardt disease, and this really was led out of Julie's lab, and Laura Whelan drove this work about this sort of deeply enriched, this intronic ABCA4 variant, and that it is a particular variant in the Irish population. And again, showing it it's classically associated with milder phenotype, but not always. And we're also looking at some of the other challenges, that when you take out the ABCA4s and Best disease from a maculopathy, we've got a very low resolution rate. So if you look at these two pictures here, you've got you've got examples of a macular dystrophy. Well, one happens to be C or B, one of the other one's gene negative. So we have a non-resolution rate of 41%. Compared to 18% in the overall population, showing the further challenges we have around tackling some of the subgroups, and particularly macular dystrophies. We're now linked into the National Rare Disease Office, so we've got retinitis pigmentosa on the National Care Pathway. So that is the route, not yet, but that is the route to getting these care pathways properly funded, and properly funded genetic testing across the way. So we're now working on the Usher's syndrome pathway and a Stickler syndrome pathway. We develop care plans out of the group, so we're giving a patient, empowering patients with all their information. We've just recruited 2 data managers to work with the ERNI on the registry, the Rare Eye Disease Registry, and to develop a national updated electronic register with OpenApp. We're developing specific pathways around the syndromes and other named conditions, and we're all set to deliver gene therapy for the first time in this country. We only approved Luxturna in Ireland in November last year. Only approved it last year. So all that work, but we're better late than never. And okay, and this is a breakdown of the clinical trials. Other people have talked about this, but this sort of gives you an idea of having got your pathways, your register, you now have patient cohorts that have access to clinical trials. And this is highlighted through the registry and the Johnson & Johnson Global IRD registry. So the registry project, the Rare Eye Disease Project, which we've been asked, Elaine Dolphus, the lead of ERNI, has asked us to lead this out of Dublin in collaboration with Bart Leroy from Ghent on the registry, the Rare Retinal Disease Registry. And we're just looking to input data from patients across Europe and collate all that information across Europe. So that data set is looking to, we've got a common data set for all rare disease patients across Europe here on the left, and then on the right are the added information that we will be putting into this data set. This will be a rich data set. We'll have thousands of patients up on it over the next couple of years, all interoperable. And that's based on the FAIR principles here, findable, accessible, interoperable, and reusable. And that data will be held in a central repository in Europe for most countries. And these are the pilot sites that we'll start off with, 9 pilot sites across Europe, with a goal of putting 25% of all the rare eye disease patients in Europe on the registry by the end of 2026. And we're delighted to partner with Johnson & Johnson on their disease-specific registry, which gives us an opportunity to delve further deeper, and they've introduced patient-reported outcome measures onto this registry, which will become important in our clinical trial outcomes, and that's on X-linked RP and achromatopsia. But when they come and meet us, we can say already that we've got 28 patients with X-linked retinitis pigmentosa, and we've got 16 patients with achromatopsia, and we'll start uploading that data as soon as possible. So this is how it all going to fit together. So the Rare Eye Disease Registry is going to be global across not just retinal, but all disorders. And then it's going to feed into Bart's registry here, which is rare retinal disease across Europe. And that just is they're a holding house, and we get good epidemiological information. But it's not till you dive down into disease-specific registries, such like the cystic fibrosis registry, that you can start to do that meaningful work, particularly around patient-reported outcome measures and other outcome measures for clinical trials. And this is the important bit for your negotiations with the FDA and the European Medicines Agency. So going right back to the first slide, what's meaningful to patients? Can we measure it? What's the best tool to measure it? Can we agree on it? And then we lobby together as a community that that's accepted by the regulatory agencies as the right tool and the right metric to collect. We'll do our work through ERNI in the Low Vision Working Group around the development of some of these, and we're bringing out some guidelines on vision testing later this year. I get the opportunity to stand up and present all this work. It's a lot of people are involved. We lean on the shoulders of giants such as you in this room across the world, and Jane and Pete and Marion, who've led the way in Ireland. And we're delighted to make our contribution this way. Please keep doing your work, finding those genes. We'll find the patients that are appropriate for you to put those genes and cell therapies into. Thank you very, very much.
So thank you, David, for a great talk and, focusing our attention back on, I think, diagnosing disease, because, there's still lots to do in the IRD space. So do we have some questions for David? As the roving mic is just coming.
Thank you so much for that hopeful talk. Of course, the registering of patients and gene testing is very important. But my question is, do you also train patients to give feedback in a certain way to have better to get better measurements and equal measurements during a clinical trial? Do you train patients in giving feedback?
I'm trying. So we're not set up quite like that. That'd be a better question to those who are actually running the clinical trials. But if we do use a disease-specific registry, such as the X-linked RP or achromatopsia, patient-reported outcome measures are built into those. And then you've got to iterate that with the constant feedback back from patients and caregivers. That's an important piece within that, and take that feedback, but also the involvement of public and patient groups in the development of clinical trials and the development of any research. And I think, actually, in fairness to Retina International and Fighting Blindness and Fighting Blindness US, the involvement of patients in driving research and driving registries is really important. And in our own working group in ERNI, the patient representatives sit on that particular working group 5 that I chair. So it's constantly been iterated. But it's a very good point that it could be done probably a bit better and more comprehensively.
Thanks.
There's a question. Do you have a microphone? Yeah.
Sorry, thanks a lot for the really interesting presentation. Tried to keep it brief. Presumably, the earlier that somebody with one of these diseases gets the treatment, the better the outcome. So I was wondering how newborn screening ties into all of this, and how many of these diseases are or can be included in those screening programs.
It's a really good question. So the other hat I wear is diabetic retinopathy screening. So when you bring screening into the conversation, you've got to be careful that we've got sensitivity and a specificity, so how accurate that test is going to be. And then if you're going to bring it into the genetic sphere, you've got to be conscious of, is it an actionable condition or an inactionable condition? So they're really the considerations, how accurate your test is. So when you actually challenge people who are doing this routinely, you realize their big concern are false positives, so over-reporting genes. So they shift the parameters down so they limit their false positives, but the net effect of that are some false negatives. So patients who may have actually a genetic diagnosis will not be picked up if you've shifted the parameter. So screening is a bit of a minefield. It's a subgroup in this. I can understand the concept, hypothesis, and wish to get something from a screening program, but there are balances that we will have to do with that. And actually, we're about to publish through my diabetic work the ethics of screening later this year by Louise Campbell, and the important consideration for genetic testing at a screening level.
Thank you.
Okay, so I think if there are more questions for David, we can. Sorry.
Yeah. Good
Oh, there
morning.
is. Sorry. Yeah, Mary.
Yes,
Yeah, Mary.
Good morning. Yes. Good morning. A question, and thank you for all the wonderful talks this morning. My question is, and I'm fully aware how important all the registries are for all the, hopefully, upcoming treatments. However, I do have a safety concern about the information that is on these registries. In the world we live in today, it's so important that only the people who really need the information can access it. So I suppose my question is it safe from cyber attacks?
It's a very good question, Mary. So we've sat, Elaine, Davos, myself and Bart, I've sat over the last 5 hours of meetings this week, specifically looked at legal and GDPR aspects of the Rare Eye Disease Registry, and this is on the back of many hours over the last couple of months. When we I left those couple of slides out. When you're setting up a registry, there are issues of usability, the legal compliance, and GDPR compliance is the data safety, and they are paramount. So they are set at the highest levels at the credit card encryption level, but you can never give a 100% guarantee, Mary. But it is for health-related data moving in an electronic format, it is the highest level of encryption that we can apply, will be applied. And the company that runs it has that track record. Secondarily, the information will be coded and pseudo-anonymized. So this is a tool that's been developed by the Joint Research Commission in the European Commission, a tool called SPIDER, that will automatically remove identifiable information and apply a pseudocode as soon as the information goes onto the central server. So without getting too technical, we agree with your concern. We're addressing your concern, but in this day and age, you can't give a 100% guarantee, but it'll be as safe as we can make it.
Okay, thank you.
Thank you, David. I just to finish off one little thing in this area, just When I started my work, we thought there were 3 genes. There are now 300 genes. And when we look at global populations, about 1 in 3 people carry an autosomal recessive variant in an IRD gene. 1 in 3 of us globally, carry a variant in an IRD gene. And that is something that we now know. So actually, I think as a community, we should, stop talking about rare all the time. These are rare diseases individually, but as a group, there are really unmet needs, large, high unmet needs with lots of patients. And so we should pivot our discussions a little towards that. I'm now going to call on my own colleague from Trinity College Dublin, that's Sophia Millington-Ward, who has been leading a lot of the work in our lab, and she is going to talk both about some of the Target 5000 projects and also gene therapy projects in the lab.
Thank you, Jane. No. No. Oh, there we are. Okay, well, I'll start talking a little bit about the Target 5000 project, which, of course, David has spoken about extensively, and, of course, we are a part of as well, so I won't introduce the project so much, but I just want to highlight that this is an all-Ireland initiative, and it is our goal to genotype all of the patients in Ireland. So you can see there's a huge list of people who involved in this project, and here are the PIs. And I want to highlight that there are a lot of posters and sessions dedicated to it as well. So I'll talk about some of the Target 5000 studies that are happening in our lab at the moment. This study was done by Laura Finnegan in our lab, a very talented postdoc who's leading Target 5000 within Jane's lab. And this is on the landscaping of the rhodopsin variants in the Irish population. And Laura has looked at, I can't even read it, 933 RP patients and found that 88 patients with rhodopsin variants, and 22 of these are novel. And you can, this shows all the mutations she has found along the rhodopsin gene, mostly in the exons, as you can see, all in the exons, and how their phenotypes are slightly different depending on the mutations that they have. This is also a little study done by Laura Finnegan again, and she has a poster number 8, if you're really interested in finding out more. And she was looking at the landscape of variants in pre-mRNA processing factors in the Irish cohort. So this is the spliceosome, which is very important in retinal degenerations. Mutations in any of these subunits can cause mostly dominant forms of IRDs, and of 1,620 IRD patients that Laura's looked at, she's found 33 variants in 5 different spliceosome genes, and 15 of these mutations are completely novel. This is a study done by Anna Ridgway, also in our lab, who is a PhD student, also very talented. And she's been looking at the genetic architecture of Bardet-Biedl syndrome in the Irish cohort. She had 40 BBS patients, and she has identified also 28 non-syndromic RP patients that have mutations in Bardet-Biedl genes, which is usually syndromic. And the heat map at the bottom shows the variability of Bardet-Biedl syndrome, very, very different depending on the gene involved, but also the mutation involved. And she has identified 24 variants in 8 different genes and 3 completely novel mutations. And that's in poster 9. And then Ella, also a final year PhD student in our lab, also very talented, has been looking at the genetic architecture of inherited macular degenerations. And she's been doing that with Franz Kramer's lab and Rebecca Hitty-Mallon's working on it, that too. And they made an inherited macular degeneration panel. They designed that, and it includes 105 genes. And Ella has looked at 223 inherited macular degeneration patients and solved 83 of these and found 11 novel variants. So you can see a little bit more on that in poster 10 and ask Ella anything you want to know. So the next part of my talk is on gene therapies that are developing in our lab. These are gene therapies for dry age-related macular degeneration and for glaucoma. And these are very, very, very common conditions. They have a lot in common, but they're also quite different. They're multifactorial. So they have risk factors, which are age, genetics, environmental factors, loads of different underlying causes that we don't really totally understand yet. But generally, dry AMD affects the retinal pigment epithelial layer of the retina, and this malfunctions, and then eventually photoreceptors also are affected. Glaucoma mainly affects the retinal ganglion cell layer, so that's the other side of the retina, and the optic nerve, and the lamina cribosa and trabecular meshwork cells are also affected. Now, there are therapies available for these conditions, but they're not always effective, or not not always effective, or not very effective. So we're still very much looking for therapies for these conditions. So one of the hallmarks of both of these conditions is mitochondrial dysfunction, and oxidative stress, chronic inflammation, elevated levels of reactive oxygen species. And these are the hallmark features that we've decided to use and to try and address. So our strategy to deal with both of these conditions is an overarching therapeutic approach that seeks to improve mitochondrial function. And the gene we have been focusing on is a yeast gene called NDI1. NDI1 is a complex 1 equivalent, so complex 1 of the electron transport chain. So it's involved in making energy for your cells, which is really important. It's encoded in the nucleus in yeast, and it's a single subunit gene, so it's quite a simple, smallish gene, whereas complex 1 in mammalian systems is really complicated and has 45 separate subunits. So this is much simpler. It's a single gene and a single subunit protein. We've optimized expression from NDI1, and we call that off NDI1 optimized. So here is a little figure of the electron transport chain, which is on the inner membrane of the mitochondria, of course. And you can see complexes 1 through 5. And here you can see how off NDI1 fits into that electron transport chain. It doesn't replace anything. It just slots in and gives the cell a little bit of a boost and a little bit of extra ATP. So I was listening to Leah Burns' talk with great interest, where she's looking to make models, and of course for age-related macular degeneration, which I'm going to talk about first, there are not very many, there aren't really very good models. So we have decided to deal with that by using lots and lots of models. And hopefully, if we can show that our therapy works in lots and lots of models, we can have confidence in our therapy overall. So the first model that we use is a CFH knockout mouse model. So CFH, complement factor H, variants are found in 50% of patients with dry AMD in this gene. So CFH is involved in inhibiting the alternative pathway of the complement system, so it kind of dampens down your immunity. And inflammation is obviously a key feature in age-related macular degeneration. So in RPE, retinal pigment epithelial cells that are knocked out for CFH, people have shown mitochondrial deficiency. So that makes it maybe a good model for us, hopefully. And we know that this mouse has ERG amplitudes that are reduced. Oops. We have shown that the ERGs are reduced from pretty around 6 months, but really you need to look from about 9 months on to see this model showing its degeneration. So it's a slow model, which makes it difficult, actually. And we also showed in electron micrographs that this model has abnormal cone photoreceptor cells, and that the mitochondria in the cones are also very abnormal. So here we show our therapy delivered with an adeno-associated virus, AAV, subretinally to this mouse, and we've been able to show that the ERG responses are very much improved in treated eyes. We've also been able to show that cellular ROS, reactive oxygen species, are lowered in the retinas of these mice that are treated. We've been able to show that the NADH oxidation rate is increased, so that's more energy. And we've been able to show an increase in cone numbers in this mouse. We used a second mouse model because we want to use lots and lots of models to have confidence. The second mouse model that we used was a sodium iodate-induced mouse model. Sodium iodate is an oxidizing agent, so it's very toxic. You can administer it to a mouse systemically, and it specifically impacts the retinal pigment epithelial layer, and it's actually very detrimental and very rapid and very severe. But even in this mouse, we were able to show improved retinal function and histological benefit. And also, we used not just ERGs, but we also used OKRs, optokinetic responses, which is a lovely assay. You put the mouse in a drum, a stripy drum, and the drum rotates around the mouse, and the mouse tracks the stripes as they turn, and it's kind of an indication of how well the mouse can see. So I just want to explain an assay that we use quite extensively. It's called the Seahorse Mitochondrial Stress Test. This is a test that measures oxygen consumption rates in real time in live cells, and oxygen consumption rates are an indication of mitochondrial function, because if mitochondria are very active, you have more oxygen consumption. And you can add chemicals to these cells in succession, which target the electron transport chain. So we can add oligomycin, which is a complex 5 inhibitor, and all of the oxygen consumption kind of stops. Then we add a decoupler, FCCP, you get a maximal rate of oxygen consumption. And then we add rotenone, which is a complex 1 inhibitor, and everything switches off again, except when we add our therapy, because our therapy is not complex 1, it's a complex 1 equivalent, and it's actually resistant to rotenone. So cells that receive our therapy actually continue a little bit of mitochondrial function after rotenone, and you can see how the therapy is working really nicely in the cells like that. And then we add antimycin A, a complex 3 inhibitor, and everything stops. So that's just the assay. And we use this assay in iPSC-derived RPE cells from patients with the CFH mutation Y402H, which is a very common mutation associated with dry AMD. So the pink cells are the patient cells, and they have very much a reduced electron transport, that's quick, reduced mitochondrial function compared to wild-type cells, which are blue. But when they're treated, the green line, you can see that mitochondrial function is very much restored. We were also able to show in a different model, a cell model, which is a sodium iodate-induced primary pig RPE cell model. We were able to show improved cell health, decreased ROS, improved bioenergetics. Similarly, we saw the same in an A to E blue light-induced pig RPE cell model, which is a good model of dry AMD. We showed it also in a sodium iodate-induced ARPE19 cell model. And we were also able to show it with a specific promoter. So the previous data, we used a general promoter, CAG or CMV. This is with a VMD2 promoter, which is specific to RPE cells. And we were able to show rescue in all the same models, the CFH mouse, the sodium iodate mouse, the A2E blue light-induced pig RPE, and the sodium iodate-induced pig RPE. So in conclusion, we rescued the CFH mouse, the sodium iodate-induced mouse, patient iPSC-derived RPE, sodium iodate-induced pig RPE, A2E blue light, pig RPE, and sodium iodate-induced ARPE19s. So hopefully that gives us confidence that our therapy is working. And I'll just go through this really quickly. We also tried it in glaucoma. So glaucoma, we're targeting the other side of the retina. So this is an intravitreal injection that we would use in mice. But this is in cells. This is an H2O2 insulted primary porcine LC cells. And we were able to rescue bioenergetics, reduce ROS, improved mitochondrial morphology. We were also able to rescue cells from patients, glaucomatous laminar cribosa cells. And thank you, Colin O'Brien, for the cells. We have a couple of other models that we also had, but I have no time to go through. And we were also able to rescue the rotenone-induced mouse model of glaucoma. So rotenone inhibits complex 1, but we were able to rescue this mouse and improve histology. So RGCs, the number of retinal ganglia cells, is increased in treated retinas. We were able to improve the optokinetic response, and the ERG, the PHNR response, was also rescued. The second mouse model is the DBA2J mouse, which we used, which is the most common mouse used in glaucoma research. It's not a great model. It's a very slow model. It has problems. It's variable. It has poor pupil dilation. It's tricky. But nonetheless, we were able to rescue the PHNR and also improve retinal ganglion cell numbers and improve NADH oxidation rates in treated retinas from these mice. So in conclusion, we were able to rescue the rotenone-induced mouse, the DBA2J mouse, H2O2 insulted primary porcine LC cells, patient glaucomatous LC cells, rotenone-induced HEC293 cells, I didn't show that, and LHON patient-derived fibroblasts, which I also didn't show, but it is published. So I'd like to thank our whole FARAR team, and particularly Jane, of course, and also previous members of our team who are also very important, but are no longer with us. And thank you very much.
Thank you, Sophia, for a lovely talk. Do we have some questions for Sophia? I think there's a question over to the right. Just the mic's coming. Thank you. So while the mic's going over, I just want to say it's been a true pleasure to be a colleague of Sophia's for many years. And all of the team in the lab, it's been wonderful. Okay.
Well,
I think
Jane's a fantastic boss. Might I just say that as well?
No. So I think the questions.
Thank you very much for that interesting presentation. I just have one question in relation to the target 5,000 population. What percentage of the patients involved have an unknown genetic cause of the condition?
Of the patients we looked at?
What percentage have an unknown? Yeah.
Yeah, so I think we solve generally about 70% of cases. But of course, the cases that are not solved immediately, we'll continue studying them. They'll go in for whole genome sequencing, and we'll keep going with them.
So it does depend a little bit on the patient, on the disease itself. It's Stargardt's disease. There's a much higher rate of solving mutations because ABCA4 is the primary driver of disease. So it'll depend with inherited macular degenerations, we have a lower rate. For unsolved cases, then they will move and are moving into whole genome sequencing. But just to put it in context, whole genome sequencing is challenging, not the sequencing part, but the analysis of the sequence, because the coding sequence that we're looking at, when we're looking at just the exons, it's 1% of the genome. Now, when we move to whole genome, it's the whole genome. It's 100%, and so it's much more demanding from bioinformatics analysis. So the data that we have is a much larger data set, and so it'll take a lot more time, I think, to really decipher the underlying causes in whole genome. But I would just argue that at this stage, as we move forward in the IRD space, we have to consider whole genome sequencing our patients, because ultimately it will allow us also not only find the primary driver of disease, but also find modifiers, secondary modifier loci that change the severity of the disease. And if we have whole genome sequence, we can actually move in that direction, which is an important direction to move forward. So sorry for elaborating on that. My apologies. So are there any more questions for Sophia?
Yeah. Hi. I was just curious. I have two questions on the AMD and the glaucoma. So in the AMD, you injected your AAV at what age in your CFH mouse? Because you said it works
8 weeks.
At 8 weeks. Did you look at an older age? Because obviously you said the phenotype kind of develops in those mice later. So I was wondering, would it work at a later age? Because obviously that's when patients would more likely develop the condition.
Yeah, well, our mouse, I think, starts to develop a phenotype, as far as I can tell, from around 3 months. So we haven't tried injecting after 3 months, no.
Okay, thanks. And for the second question, it's on the glaucoma side. So glaucoma is linked to increased intraocular pressure. So I was wondering, did you look at that in your glaucoma, however you say it, mouse, the glaucoma mouse? The
DBA2J mouse.
Yeah.
So the DBA2J mouse it's kind of hard to measure the intraocular pressure because the corneas tend to calcify, and it gives very, very variable results as because of that. So it's not a terribly reliable or easy outcome for us.
Did you look at it then did you do it in the rotenone mouse then?
The rotenone mouse probably doesn't have increased intraocular pressure because it's a very rapid phenotype.
Okay, thank you.
Okay, well, if there are no more questions for Sophia, I think we'll move to our last speaker before coffee because we're going to all need our caffeine, I think. So we'd like to ask Oliver Black, again homegrown, from UCD, University College Dublin, to speak to us today about animal models for ciliopathy genes.
Great. Thanks, Jane. So everyone's desperate for coffee, I'm sure, so I'll try to keep this pretty quick. One advantage of going last is quite a lot of my stuff has been kind of introduced, so that's helpful. So it'll cut me on time, which is great. So people have mentioned ciliopathies here, so I actually work on cilia and ciliopathies. And so I thought I'd give a small bit of background about that, and then I'll tell you about how we're using animal models. I'll tell you what the animal is a little bit later. How we can make lots of patient models quite quickly, actually, and do some phenotyping. Okay, so cilia, these are these great structures that you've heard about in terms of the outer segment of the photoreceptor. They're typically hair-like. We have lots of them on most of our cells. They beat, at least the motile ones do, but most of our cilia don't beat. So that's actually what we have in the retina. In the photoreceptors, we've got non-beating cilia, which actually sense our environment. So this here are the photoreceptors, which you all know lots about, and the outer segment is a modified cilium. So it has these major membrane elaborations. It has the ciliary backbone of the microtubule. So these are sensory cilia, and obviously they're used to sense light. This is how cilia might look, actually. This is a beautiful EM image showing the microtubule backbone of a cilium. And you can see the microtubules emanate from a basal body. So the basal body centriole is basically what establishes the microtubule architecture. And the microtubules extend all the way down to the tip of the cilia. So these cilia can be anywhere from 1 micron to millimeters long, actually. So in the photoreceptor, the outer segment, I'm not sure what the exact length of that is, but it's 5 or 6. I could be corrected on that, microns long. Someone could correct me on that later. But nonetheless, they can be very, very long. I will talk a little bit about, in my talk, what's happening at the base of the cilium. This is called the transition zone. It's the same as the connecting cilium in the photoreceptor. This is the gateway into the cilium, and is a very, very important part of the cilium and the photoreceptor for regulating what can get into the outer segments. So cilia have, obviously, they have motility functions, sensory functions. I've mentioned those already. They don't just sense light, of course. They sense smell in our nose, taste in our taste buds. But cilia are very, very important during development as well. They sense lots of internal chemicals and ligands, which are important for cells to communicate with each other. So some of the audience will know about these very famous signaling molecules, hedgehog, Wnt, Notch, and PTGF. So cilia are these sensory developmental signaling organelles, very, very important for controlling cell behavior. And so therefore, if you've got defective cilia, not only do you tend to have sensory defects or motility defects in cells, you can have developmental abnormalities. And indeed, there are lots of developmental abnormalities associated with cilia dysfunctions, the ciliopathies, and you've heard about those today. They affect most of our tissues and organs. They're typically syndromic because of the many different types of cilia we have and the many different functions. You will see defects in many tissue types. Now, the retina and the eye tends to be highly prevalent in terms of a phenotype in ciliopathies. Many, many ciliopathy patients, and of course we have more than 20 ciliopathy disease, probably 250 plus ciliopathy genes, and in that very large cohort of diseases, we very, very frequently see defects in the eye. And in some cases, we see non-syndromic ciliopathies where only the eye is affected. So here are some of the retinal ciliopathies listed here. You've heard about those already. And it turns out for many of the ciliopathies that the genes associated, they seem to function in building and maintaining cilia, or building and maintaining the outer segment of the photoreceptor for that matter. And what we know about how cilia and outer segments are built, we know that there's some very, very important pathways there. So for example, we know at the base of the photoreceptor outer segment is the connecting cilium. And this transition zone of a cilium is very, very important for not allowing things to randomly move in and out of cilia. Cilia have to have a very defined composition. They're open to the rest of the cell. So we need mechanisms, barriers, mechanisms to make sure only the right molecules get into and out of cilia. So how do we get molecules into cilia at all, actually, if there are barriers there? Well, we have these transport pathways. One that's famously studied is called intraflagellar transport, or IFT. These are molecular trains that move from outside of cilia into cilia and back out again, and they bring with them the components that the cilia needs to be built and maintained. And you tend to find for the retinal ciliopathies, actually, mutations in many of the genes that are part of these assemblies are causing vision problems. So my lab has studied these different pathways in the context of ciliopathies and more recently the context of retinal ciliopathies. So the animal model we use is C. elegans. It's a nematode. Okay, it's only 1,000 cells. It's a very, very famous animal model like Drosophila, the fruit fly, and yeast, of course, in terms of the lower invertebrate models. It has strong conservation of of many of the cilia genes, many of the ciliopathy genes. It has, of course, sensory cilia. It doesn't have eyes, but it has sensory neurons. And so we actually model retinal ciliopathies looking at the olfactory sensory neurons of C. elegans. So these are the smell sensing neurons of the nematode. We have some great readouts of smell and sensory function in C. elegans, so we can pretty much figure out very quickly and easily if the cilia are functionally normal or structurally normal through imaging. And we have really strong genetic tools, which allows us to very easily manipulate genes and make animal models of genes of interest. So several years ago, I became interested, instead of just looking at disease mechanisms, employing knockouts, so the sledgehammer approach for a model of organism geneticists like me is knock out the gene and look at the function of the gene. So we started doing more sophisticated analysis. So we said, let's try and model the actual patient mutations. Instead of just using a straight knockout, can we introduce a missense mutation that a patient has in a gene of interest in the worm's version of the patient gene, and can we study the disease from that perspective? So really trying to understand the correlations between genotype and phenotype. And we thought then if we can do that, maybe we can do things like interpreting these very difficult alleles, the variants of uncertain significance. So we all know that in ClinVar, 3/4 of all of the mutations deposited in there, maybe 70%, we actually don't know if those mutations are actually causing the disease or not. So can we model those mutations in C. elegans, see if those mutations affect our gene in C. elegans? If they do, that might be evidence that that mutation is actually deleterious. And we've also started some drug screens. So I'm going to, in probably 5 or 6 minutes, just give you a flavor of a few different genes we've been focusing on, show you what we can actually do in C. elegans, how we can interpret human genetic variation, and how this can actually help us to understand disease mechanisms. So the game changer for my lab was CRISPR-Cas9 gene editing. So in C. elegans, the whole field was screaming out for this technique because we didn't have good methods to actually make mutations that we wanted. So Karen, in my lab, basically set up over the last few years the methods for using gene editing in C. elegans. What I can tell you is within 2 to 3 weeks, we can basically make an animal model of pretty much any missense mutation that we would want. So, we buy in the reagents, we inject them into the C. elegans. So we buy in all the Cas9 reagents, so we don't have to make anything. We inject them into C. elegans that gets inherited by the next generation in these hermaphroditic worms. The edits happen in the F1 generation. And because worms are hermaphrodites, which is fantastic, it means if we go one more generation from the F1, we will get recessive mutations. Because usually with gene editing, you'll only edit one copy of the gene. We edit the copy in the F1 worm then produces the Mendelian ratios for us in the F2. So this allows us within 2 or 3 weeks to basically get these mutations that we would want. So I'll tell you about a few of the genes we've been focusing on. So this one is B9D2. It's one of these transition zone or connecting cilium-associated genes. It's associated with Guebert syndrome and Meckel-Gruber syndrome, but many of the patients have retinal dystrophy. This is just to show you how similar the worm and the human proteins are. In fact, for this particular gene, the proteins are the same length. They have 50% orthology. So these are very, very similar genes conserved between worms and humans. So we decided to focus in the first project on this mutation. It's a biallelic mutation in a patient where the patient has a proline to serine at the 74 position or a glycine to serine at the 155, highlighted here. And we said, let's model those two, recapitulate the patient model in C. elegans, and see if it's deleterious. Can we establish the worm as a good model, for this type of mutation? So this is the typical approach that we take. First, we knock in a tag in frame with our gene of interest. So B9D2 is called MKSR2 in C. elegans. So now we can actually tag a fluorescence molecule so we can see the protein in this localization. So it's very, very useful. The protein localizes beautifully to the base of all the cilia. There's 4 cilia here. You can see the 4 dots, very, very specific localization. Then we take this strain and we start editing the patient mutations that we want. So in this case, Karen went along and edited in the P74S and the G155S, and we end up with our recessive worms. So we have loads of different assays, and I'm not going to go through these in detail to assess cilium structure and function in these worms, but we have a very, very quick assay to tell us if cilia structure are normal. We can take worms, dip them in a fluorescence dye. If the cilia are normal and environmentally exposed, the dye gets into those ciliated neurons and they stain up beautifully. If the cilia aren't there or they're short or truncated, the dye doesn't get taken up by the worm. So really fast assay to tell us if the cilia there or not. We can measure cilium length directly. And of course, we can do those behavioral assays. Worms roam around in the place. They sense well. If they don't sense well, they stay stuck in one clump. If they should normally avoid high osmolarity environments. If they don't sense the environment, they'll cross. So all these great readouts to tell us about the function of the effect of the mutations. We can also look at the effect of the mutations in protein localization because we got a tag. So here's the wild-type protein, and here are these two patient variants. So you can see this variant doesn't affect the localization, this one does. Now, these are endogenously tagged markers, so we have a lot of faith in this type of readout. And so we can basically say, you know, for these mutations, they're actually highly deleterious in C. elegans. Very severe defects in diuptake, roaming, barrier assays, and localization assays. These are missense mutations put into a worm, and they're still deleterious. So that's really fantastic, and it shows us in this case that indeed we can model the human variation back in C. elegans. Of course, the patient was biallelic for these two mutations. It wasn't recessive for both. So we recapitulated the biallelic genotype by simply crossing our two worms together, each with the individual missense mutation, and we recapitulated the biallelic mutation. And indeed, the biallelic mutation looks to be as severe as the worms that were recessive for either of the individual mutations. So this was really a breakthrough moment for my lab because it established that we can model missense variation in genes back in C. elegans. And we have really good tools for making the mutations and for classifying the mutations phenotypically. So very quickly, then we decide, well, can we interpret VU mutations, variants of uncertain significance? And so we focus on a different gene, this time TMEM67, MKS3, also a ciliopathy gene. Also patients have blindness in many cases. And we modeled all of these different variants of uncertain significance. You can see these missense variants, along with some pathogenic alleles. So then we ran our different assays on these different patient models. And we found, for example, the benign alleles were benign in our phenotypes. No phenotypes. They smelled correctly. Everything was correct. For our controls that were pathogenic, known pathogenic mutations, indeed, in our modeled worms, they were also pathogenic. So that was great. What about the VUs? Well, interesting, VU2, 3, and 7 in this gene were all normal in C. elegans. So we didn't see any effects on cilia, whereas the other 5 VUs that we looked at all looked pathogenic. So this indicated to us that we could now go back and say, well, we would argue that VUs 2, 3, and 7 in this gene are likely benign, whereas the other 5 VUs are likely pathogenic. Now, if you take the standard way of assessing whether a mutation is pathogenic or benign for a patient, it's mostly sequence-based. And in fact, for all of these different VUs, the in silico analysis suggests that they're either VUs or they're all pathogenic. But when we do our in vivo interpretation of these assays, we know in C. elegans that these 3 are definitely benign. So one of the challenges we have now is because we can roll out these kinds of experiments very quickly, we can make lots of different patient alleles, and we can try and interpret variation, which of course is a very, very important thing that we need in the field. We need to try to convince now those that work in the clinicians that there is value actually to interpreting an allele in a worm model. Of course, we work on very conserved pathways and very conserved genes in C. elegans, and we have a lot of good data over many years to show that the worm is a great model actually for the ciliopathies. So we hope that we can make that convincing argument. I'm going to skip that last one, but I just want to say at the end, we have now moved, thanks to Fighting Blindness and HRB, so we're really trying to model some variants specifically linked to retinal dystrophies. We're focusing on the BBS2. People have talked about Bardet-Biedl syndrome. So there's a whole slew of mutations in BBS that cause non-syndromic BBS, where just the eye is affected. And there are mutations where in the same gene where the patients get full-blown Bardet-Biedl syndrome, where they get the symptoms in all of the other tissues and organs. And so we're going to model those different types of variants in C. elegans to see if we can understand why does mutation X cause a very subtle phenotype in patients versus mutation Y in the same gene causing a much more deleterious phenotype. And finally, we're doing some drug screening. So worms, you can put them in 96 well or even 384 well plates. They won't be super happy, but they'll swim around in there. You can apply drugs, a compound. So we're going to do a compound screen, and we're going to try rescue the cilia defect for this dye filling assay. So here's a patient variant where the cilia are abnormal. It doesn't take up the dye. Can we add compounds quickly and easily on a high throughput imaging system and find compounds that can restore back cilia to this phenotype? And that, therefore, we think could be a good lead compound to go to higher systems. So we've got collaboration with Brendan Kennedy and zebrafish. Will the compound restore, for example, the variant in a fish? And then that could lead on further. I think that's it. Happy to take questions. We've got a great team here in UCD. Most of the work was actually done by Karen, and now the RD SIL project with Fighting Blindness is Niels and Anna. So thanks for your time.
Thanks, Oliver, for a wonderful presentation on ciliopathies. Do we have some questions? There's a roving mic. Yeah, we have a question over here.
Thank you very much. Thank you very much. So my question would be, have you tried a known pathogenic variant in your model organism, and which then showed no pathogenic effects in the C. elegans?
No, we haven't found those yet. So we've tested, I think, 15 pathogenic. We only focus on missense variants. The 15 known missense pathogenic variants, when we model those in C. elegans, are all deleterious. So far, it's nearly been 100% success, but we actually need to keep modeling the known pathogenics and the known benigns. And I didn't have time to show you, we have actually some CEP290 work. I can show someone afterwards. So we're trying to build up a full library of alleles to make a very convincing case that the nematode really can interpret human variation, at least in some select ciliopathy genes. Yeah.
Are there any more questions for... Oh, yes, sorry, Isabel.
Yeah, wonderful work, really very, very useful. Besides cilia, is there any other pathways that are conserved that can be also a good model for the, like splicing or some other cellular functions?
Yeah, so there are several groups across the world now using C. elegans to model different types of diseases and different pathways in terms of human genetic variation. So one study actually made, in an epilepsy-associated gene, they made 55 variants in the C. elegans model. They actually humanized the worm. So that's something my lab is trying to do, put in the human version of the gene, and then we can model any type of variation. Because worm and human proteins are only sort of 20 to 50% identical, so that would allow us to cover everything. It's still in its infancy, but I would say if you've got good conserved biology between the worm and, let's say, mammalian systems, and you're convinced of that conservation, then I think, yeah, you should be able to model it. But at a blueprint biology level.
Are there any more questions for Oliver? We just want to thank you once again. Thanks, Oliver. To conclude this session, I just want to thank every speaker for their wonderful presentations. And we are standing or sitting on the shoulders of giants, as Isaac Newton said. And we have had lots of those giants in this room. At times, I think I didn't give enough time for questions, so my apologies to both the presenters and also people who wanted to ask questions. But I'm sure you can grab the people a coffee and lunch as well. And so just one more round of applause and a big, big thank you to all our speakers.
Delegates, thank you very much. We're now taking coffee. We're going to take the full 30 minutes. So we will be returning at 12:06 Irish time. So if you'll be back for then, that would be great. I would like to invite all of our speakers. It's really important that all of our speakers go to the ground floor for the photo call. If they could make their way there, that would be brilliant. And I'd kindly ask all delegates to visit the upper ground for the meet you all. So thank you all very much. See you all at 12:06.
Okay, we're going to get started. So if people could take their seats, thank you very much. So we're going to continue the same theme from before the break, so still on therapeutic development. And the first speaker in this session is Professor Robin Ali. So very exciting to have him here from the Institute of Ophthalmology in UCL. And he's going to tell us some interesting and very exciting data related to therapeutics. So Robin, if you could come up, we'll get you started. Thanks for coming.
Thanks very much, Oliver, and thank you for the introduction. I just have to, I've got my first slide up, just have to point out that I'm now
Hopefully I get it correct this time. So could I welcome Dr. Daniel Chung to the stage, Chief Medical Officer of Sharing Vision. Sparing Vision. This is not going well today.
No problem. Thank you so much. And if I could, I would love to share my vision. So, as always, it's a great privilege to be here. Thank you so much to the organizers for the kind invitation. I'm always very humbled to be included in these types of conferences, which is always one of my favorites. So today we're going to talk a little bit about some gene-agnostic approaches, which is a little different than your gene-specific approaches, which has really been the mainstay of gene therapy. But the reality is that we're not going to be able to cover all these. But before I get into that, let me get some housekeeping done. By way of disclosure, I am an executive full-time employee at Sparing Vision. I consult for Spark Therapeutics and for Asthernus Therapeutics and sit on several clinical advisory boards. All right, got that out of the way. So when we think about inherited retinal diseases, we all know that there are about 300 or more genes that are responsible for inherited retinal diseases. And as much as we'd like to take each one of these into individual development, it is just not feasible. We just do not have the resources and time, personnel, and above all, funding. And so the idea is, can there be gene-independent approaches where we can take classes of diseases and find some common pathway or something that would work without necessarily going after the causative gene? And this is by no ways to be a substitute for gene replacement, but we like to think of this as coming alongside gene replacement as another alternative, especially for some of the diseases that may be too rare for ever to have clinical development for. So when we think of gene-independent approaches, that is part of the thing is we think about what is a common pathway that can be effective in a class of diseases, such as RP. The other things are things like optogenetics, which I believe Professor Flannery will be talking about after this, which is also very interesting, and a lot of folks are looking at that. There are gene modifiers that certain companies are looking at, that they can use different genes, not the gene that is causing the visual issues, but another gene that may be able to help alter the effect of that. And then for us, we're looking at neuroprotection or metabolic alterations of things. And so specifically, we call this SPVN06, and this is really the basis of RDCVF, rod-derived cone viability factor. And this is something that was really coined by Jose Sahel and Thierry Livellard. As Professor Odo mentioned, this is the one-year anniversary of his death, so we hope to continue his legacy in the trials here for RDCVF. But what is RDCVF? But before we even get to that, let's talk about the disease that we're actually targeting, and that is retinitis pigmentosa. And I'm sure I don't need to get into a lot of detail here about that. But on the screen here, there is a schematic, has 5 different columns. Each one of those columns is basically a progression of the disease. We start off when the retina has a relatively good complement of both the light-sensing cells that you all know as photoreceptors. We have the rods in the periphery, we have cones in the middle. Well, as the disease progresses, what happens is the rods start to degenerate and go away in the periphery, and then eventually the cone cells get affected, so central vision gets affected as well. And then there'll come a time, too, when those cone cells will lose their function, but they'll still hang around a little bit. And then ultimately, those cells, too, will be lost. So that is the principle of RDCVF, and any disease technically that has that type of pattern could be applicable for the mechanism of RDCVF. And so what is RDCVF and what does it do? It actually comes from the gene called nucleorhodopsin-like 1 gene. And as I mentioned, this was the brainchild of Professor Sahel and Professor Livellard. And basically they asked the question, why is it that in RP, or rod cone dystrophies, we know why the rod cell dies because of the direct link to the genetic etiology. But there is no real direct link from the genetic etiology to the cone cells. And so this is what they determined. They identified this RDCVF. And this RDCVF is actually 2 different components because the NXNL1 has 2 different exons. And through alternate splicing, there's a short form, which is actually secreted by the rods. And this binds to a receptor on the cone photoreceptor, and this starts this metabolic pathway that increases glucose metabolism, carbohydrate formation, and therefore it stabilizes the membrane and function of the cone cell itself. And this is something that happens in every one of our eyes right now. So with that, it's exploiting that whole idea that if the rod cells are dying in rod cone dystrophies, and therefore there's less RDCVF to maintain cones, can we use gene therapy to put that back in and hopefully slow down or stop the progression of central vision? It's not going to do a whole lot for rod function, but at least can we maintain central vision for a good chunk of the rest of folks' lives with RP? So here we are talking about slowing or stopping. We're not talking about restoration. And so the other part of that is the long form acts as a powerful antioxidant. And we're all eating our blueberries and raspberries and whatever else has high antioxidants because that really helps with inflammation and things of that nature. So put these two together, and that's the protective effect of RDCVF. And so in this schematic, there's on one side, there are 3 boxes, and this shows the dose escalation part. So the clinical trial is ongoing. We are looking at genotypes of rhodopsin, phosphodiesterase beta, and alpha. And we are enrolling these patients in the first 3 cohorts. What we are doing is that we are bringing in folks that have visual acuity of 2200 or worse, but not off chart. So basically they're legally blind and they have visual fields of 20 degrees. And they receive a single injection in the worst seeing eye subretinally. And so this is where we find what is the best dose. And once we do that, you'll see the second box here is that we go into a dose escalation. And then there are 3 additional boxes there that says we're going to divide patients into a control group and then 2 dosing groups. And this is really one of those designs that FDA really wanted us to do. And that's a whole different conversation. So if you want to talk about that, let me know, because I'll go off on that for a while. But anyhow, for now, that's the way this is done. We're going to look at these patients for a year. We'll look at structural and functional endpoints, as well as patient-reported outcomes. This trial is known as Prodigy. So if you hear that, that's what we've called this. Many of you have heard me say this, but I asked my team to name the trials. They picked rock groups. I've never heard of Prodigy. So I told them the next one, you got to pick a group I know. They picked Nirvana. That one, at least I know. So anyhow, so that's the way Prodigy is going. We're in the 3rd dosing cohort right now, so we are active. And we hope to, if everything works out great, we hope to have this fully enrolled by the end of the year. So just going through the status of this, we did cohorts 1 and 2. We've done 6 patients in our site in Paris with Professor Isabel Odo as our PI there. We will be expanding in the United States. Right now, University of Pittsburgh Medical Center is one of our sites, along with Miami, Portland, Dallas, and Mass Ioneer. So in cohort 1, we didn't have any safety issues of major concern, no SAEs, that's a serious adverse event, no discontinuation, no dose-limiting toxicities, and we have our data safety monitoring board meet after every cohort. We had a few adverse events, but these were what we see usually in subretinal injections, so there was nothing significant there. When we went on to cohort 2, same thing, no serious adverse events, no discontinuation, no dose-limiting toxicities. We had a few more adverse events. We had red eyes in pretty much every patient because it is a subretinal injection. We had 1 patient with a macular hole that resolved on its own. And recently had 1 or 2 patients that had a decrease in visual acuity on their 6th month visit. 1 patient, it was really more about, and this shows the visual acuity is very variable. They came right back to where they were when we brought them in for a second visit, so the visual acuity really did not change. And the second patient had a mild decrease, but nothing significant as well. So inflammation was very well controlled. We have very minimal inflammation in all our patients. And then this brings us to the second program, SPVN-20, known as Nirvana. This is not in the clinic. We hope to be in the clinic by the end of the year, but sounds like it's going to be a little more toward mid-next year. And this is using something called GIRC, G-protein inwardly rectifying potassium channel gene. And this is for cells that we call dormant cone cells. So remember those central cells that we use for daytime and good acuity, is that the photoreceptors, they have this thing called the outer segment. That's where something called phototransduction happens. That's where light comes in, it's translated into an electrical signal, that gets sent to the brain, and the brain says you see. The reality is the eye doesn't see a thing. It's the brain that sees, but the eye is this highly complex relay system. And without phototransduction, that signal doesn't get sent. So with GERK, once you put that in preclinical studies, that phototransduction is restored. And so we're hoping to be able to restore function. This is restorative, which is a little different than RDCVF, which is more about slowing or stopping. And we're going to probably start with very end-stage patients first, maybe patients that don't see any light at all, and see if we can at least bring light sensation back. So this trial is going to be a little different. It's going to be an AAV vector, but this is going to be an intravitreal injection, so it won't be done in the operating room. It will be either done in a procedure room or maybe even the doctor's office. We haven't worked that totally out yet. And so those are the two programs. And remember, both these programs, it doesn't matter what your gene type is. In the first program, it's going to be rod cone dystrophies. We'll look at RP. In the second program, it's going to be inherited retinal diseases that are patients that are at end stage but still have dormant cone cells left. So take-home message when it comes to gene independent is, number one, is we can really probably reach a broader range of patients. We can treat those that have very rare genotypes that may not ever get any kind of clinical development done. The other thing, these are some of the practical things that when you work on my end, I was part of the clinical development with Luxturna, so I was part of the clinical team that did the things with the patients, and then I moved to the sponsor side when I went to Spark Therapeutics. So I kind of get an idea of both sides here. And unfortunately, there is a side of resourcing. And the thing is, pricing as well. So we believe that gene independent also will be a lower price point. And the other thing is that it may be a little more attractive to get resources because it's a larger group of patients. And again, the way I look at this, it's not competitive with gene specific, it's more or less coming alongside, and the idea of giving folks another option. You know, right now we're at a place where we have one gene therapy product, and it'd be great if we get to a place where folks can have a choice of what is best for them as they talk with their physicians as to what would be the best gene therapy product for them. So it's really, like I like to say, I'm a big hockey fan, it's all about shots on goal. Yes, I'm also a European football fan, and it's all about shots on goal. And the more shots on goal, the better chance we have something that will be effective. So I appreciate your time today, and happy to take any questions if you have it.
Wonderful talk. Thank you very much. So questions? Here and here. Maybe on the left first.
Thanks for that very inspiring talk. Thanks for giving hope. Over what kind of periods are you going to follow the patients?
So our phase 1 is going to be really about safety. Obviously, that is our main goal, but as everybody asks me every 5 minutes, what about efficacy? So we will be looking at that as well, but it's a short period of time in a relatively slow-moving group. So we are looking at different endpoints. We did a natural history study, a prospective natural history study, and there is a subset of patients that after 1 year, they're something called the ellipsoid zone, which is a marker on OCTs. We have found a subset of patients that are basically progressing at twice the rate of test-retest value. And so we are going to look at those patients in our extension phase and see if we can change the slope even in 1 year to see if there's a difference there. Otherwise, what will go on after this is we hope to get into a phase 3 study where we'll hopefully add 7 to 10 more genotypes because we'd like to get a gene agnostic label at the end. And that study, unfortunately or fortunately, will run 2 to 3 years if we can get the right people involved in that and the funding for that.
Okay, thanks.
Hi, thank you for that very hopeful information. I'm just wondering when the Nirvana trial opens, who is it open to, and will it be multi-site?
So at this point, we are talking to several centers in Europe. We're talking to a center right here in Ireland, one in Ghent, and one in Paris. So we are still in the preclinical stages in the sense that we're getting or CTA ready, which means we're going to hopefully submit our application to the EMA by the end of the year. And if everything goes well, we will be in the clinic hopefully second quarter of 2025. We are probably going to start with either folks that have only light perception or no light perception. So we're going to start with a very advanced group, and obviously if it's effective there, then we have many other ideas of what we think we can do with that. So stay tuned on that.
Thank you. Just very quickly, whilst it's gene independent, would it be necessary for patients to actually have a gene ID?
Yeah, for that, I'm a true advocate of genetic testing, even for gene independent, because the reality is that really helps in a lot of different ways. It helps you and your physician to understand what your diagnosis is, what the prognosis might be, what the disease course might be, and what other gene therapies or other products may be available. With that, for RDCVF, it still has to be a rod cone dystrophy, or a type of RP. And for the GERK program, we are probably definitely going to still look at rod cone dystrophies for that. And so we'd like to narrow it down just a little bit. So regardless, I always encourage genetic testing.
Okay, thank you.
Okay, great. With that, we move on. Thanks again, Daniel. Okay, so next up, and I won't get this affiliation wrong because it's a very good colleague of mine, Professor Brendan Kennedy from UCD, and he's going to tell us about his zebrafish work in therapeutics. Brendan.
Okay, great. Thank you all for attending my talk, and thanks for the invitation to speak here. It's hard to follow a rock star, even though he might know who Prodigy is. I mean, how do I follow an amazing talk like that? So I'll try, and I'm also very conscious that there are people in the room that are not able to see my slides, and I rely on them a little bit, but I'm going to try and describe what's on the slides rather than the detail that's in them. Okay, so there's three things that we're interested in. We're interested in basic biology of the retina. So what happens? And for me, this is, if you don't understand the biology, it's very hard to understand the disease, and it's very hard to develop proper therapies to be able to evaluate them. The second part is about the diagnostics, the variants of unknown significance. We have a little bit of that, not as advanced as Ollie is, but in a model that we can assess vision in. And then the last one is a little bit about drug discovery, which is different to the gene therapies that you've heard about mostly today. So this is on the slides here is a diagram. It's just pulling out the photoreceptors, which are the rock stars. They are like the lead singers in the band. They are the sexy part of the retina everybody is interested in. And next to that is the RPE cells, the retinal pigment epithelium cells, which are kind of being thought of as maybe more like the backing singers, less important, needed to get everything going, but less important. Okay, and I'm going to tell you, though, the two processes that we're interested in require these two cells to work together, these two cell types. So the first process we're interested in is the visual cycle. This is the retinoid cycle. This is recycle me, recycle the light-sensitive form of vitamin A. So this is like your green recycling bin, but to restore carrots every day, every hour, every minute. So it's the recycling part. And the second part is something called outer segment phagocytosis. Mary loves to, when I tell her that, she loves to remind me that this is the thing that we ended up with the language, eat me. This is the eat me process. So this is an amazing process that happens in mornings and evenings, and it chops off about 10% of the photoreceptor daily. And it's part of the way of removing the waste so that you have a healthy photoreceptor. So these two processes, though, require cells functioning together in order to be able to control these processes. So it's an integrated physiological process. You can't study these in monoculture. You can't study these in organoids. You need an in vivo system to do that. In the background, which I didn't explain, is the 280 different genes that are associated with inherited retinal degeneration. So the text size gets smaller and smaller as more genes are added. We can't work on them all, so we work on, well, I'll talk about 3 of them today. We work on about 5 IRD genes, and the 3 I'll talk about today are all involved in either the recycling of vitamin A or removing the waste products. So on the slide, the next slide is to tell you that that was actually very simple because I kind of told you there was just two players. There was just two teams. There was a photoreceptor team and the RPE team. So it turns out it's even more complicated. There's also a Mueller glial cell team. So that gets complicated. You might think of this as a football match with three teams playing on the pitch at the same time. And it's very difficult to know who has the ball or who's fighting against who and who's winning. But Mueller glia are part of the visual cycle as well. And so we're not going to go into the complexities of it, but there's parts of the process that occurs in photoreceptors and RPE, and other times this recycling happens in Mueller cells and RPE, and sometimes it happens needed in light, and other times it's needed in dark. Okay, and this is the gospel according to zebrafish, but I'm going to try and keep it quite small. We work on zebrafish as our main experimental model. So they're a vertebrate model, they have a vertebrate eye, they have a very cone-enriched retina, so that allows that they have colour vision, visual acuity. And for us, again, a bit like what Robin just talked about, we can look at behaviour. So we can look at vision, we can look at their ability to track stripes, to follow moving objects. And for us, again, that's the most important readout in retinal research is it affecting vision? So we look to create things that alter vision, and then we look to be able to restore vision back to those animal models. So I'll be talking about some of the things that we do in our research like that. So on the slides, there's a couple of these slides that there's a lot of detail in them, but I'm going to try and explain them quite briefly. So the first gene I'm going to talk about is something called cellular retinaldehyde binding protein, or RLBP1. So this is a gene that's involved in the visual cycle. That vitamin A, that carrot recycling, if we want to make it very simple. And that gene is associated with inherited retinal degeneration. RLBP1 would also be kind of thought of as like the backing singer. It's not necessarily that important, it's a carrier protein. But there's quite a few mutations in RLBP that cause IRD. And Dave Keegan's list earlier on, I saw that one of the patients, of course that's what I'd be interested in Ireland, has an RLBP1 mutation. In zebrafish, in humans, we've one gene that's expressed, one RLBP gene that's expressed in the Mueller and RPE. In fish, they duplicated their genome to make it even more complicated, put more football teams on the pitch. But they've one version of the gene that's only expressed in RPE, and another version of the gene that's only expressed in Mueller. But that allows us to figure out what does the RPE version of this gene do. And there's a lot of controversy in the field about what the RPE versus Mueller one does, but I won't go into that today. So we have these two forms. And the next part of the slide shows how we took the CRISPR approach, this molecular scissors, and we deleted out this RPE-expressed RLBP gene. Now, that probably took that person about a year of their research career to do, and I don't even say anything other than that we created that with a little scissors cutting out the gene. And then John, who's a new PhD student, well, not that new, he's coming into his final year, we created these knockout fish, and then we wanted to know, what's their vision like? So we have a behavioral assay, it was talked about earlier on from Sophia, where you have a rotating stripe, and the fish will move, follow their eyes, follow with the stripes. And so John, here's this model, we've knocked out this gene that's important in the visual cycle, and when he looked at their vision was perfectly normal. Okay, so that says that it's not just, but that says, okay, what's going on here? So John went to the literature, and he saw that the patients who had RLBP1 mutations have a defect in dim light vision. So we had to adjust the way we do our assays to the fish, and so he did the optokinetic response where the light levels that the fish were during the assay were much reduced. And when he does that, then these fish that have lost their RLBP1 don't see the moving stripes as well. So in this case, it's in low levels of light that these fish are affected, and they don't have the recycling of the vitamin A sufficient to support that. The second one is a very similar one. This is a gene called RGR. So we tend to always seem to work with genes starting with Rs, but RGR, it doesn't matter. This would be regarded more like the lead singer again in the rock band. RGR is meant to be the enzyme that enables us to continue to make vitamin A, the light-sensitive form of vitamin A, in bright light conditions. So there's a system to do it in the dark which involves RP65 Luxturna. That's meant to be a slower process, but it's not sufficient to do it in light. And so RGR is meant to be that rock star that can do that in the light. But it's also a bit odd because there's very few, there are some, but there are very few inherited retinal diseases linked with RGR. So I don't know if that was 10 minutes because they switched to 20. Will I keep going? Yeah? Okay. Okay, thanks. And in this case, we proved again, we make a knockout of the fish, which becomes very easy at this stage. There's 2 RGR genes. In this case, both RGR genes are in this retinal pigment epithelium. So what we had to do was knock out both genes and create a double knockout. We proved that that's the case. When we do our standard OKR, the fish, they're perfectly fine. They survive, they grow to adulthood, they mate. They have no problems in a standard OKR assay. But in this case, when we put them in the assay where we then expose them to bright levels of light, so Tess put them in bright levels of light for a day and a half, or Grace put them into greater than 50,000 lux for 8 hours, when they're put into that level of light, they can no longer, they have a deficit or a significant loss of function in their visual behavior assay. So this one seems to be more important in having vision during bright light conditions. In both models, we now have phenotypes that we can assess whether we can rescue them. Okay, the next one is RAB28. This is a gene that we worked on with Ollie. We had a fantastic shared student called Ailish. And outer segment phagocytosis, this is chopping off the tip of the outer segments. So does it somehow, not fully understood, when the lights come on in the morning, chop off 10% of your outer segment. So not kill the whole cell, prune away 10%. And in the evening when the lights go off, prune away another 10%. So the idea is to remove the waste material. It's not fully understood how this process occurs. It occurs between RPE and photoreceptors, again, those same 2 cell types. RAB28 is also another IRD gene. So what I'm showing on the slide here is a lot of work that Stephen started with Ollie and I, and then Ailish followed up on, and now John is following up again afterwards. But when RAB28, when you knock out this gene, you have these peaks and troughs normally of outer segment phagocytosis. So in the morning, then it goes off, and then it goes back up in the evening. And in RAB28 knockouts, they're flatline. They don't have peaks or troughs of outer segment phagocytosis. So this RAB28 is really important for this waste removal machinery. From a basic research point of view, one of the things that Ailish was able to do was take the fish at peaks and troughs of outer segment phagocytosis and identify which proteins that are normally upregulated at a peak are now no longer upregulated when RAB28 is missing. So these are most likely the key players in controlling outer segment phagocytosis. So on the screen here is a wonderful list of proteins, but what I've been able to do is identify ones that are lost at both peak, the morning peak or the evening peak, are lost at all time points, or normalized at all time points. And so these are a candidate list of proteins that we're really interested to hopefully follow up in the future to see how they control OSP. Okay, so we're going to switch now to the more diagnostic and therapeutics. Again, we're nowhere near what Daniel just talked about. But before I do, I just wanted to mention one thing about PPI. So at the table over there with me is Jerry and Mary. They're part of our PPI panel that we have. We've had that panel for about 5 years at this stage. And it's a really important thing in terms of the quality of our research. So Ailish wrote this blog that was published on Arvo, and I think it's a really nice, honest account of PPI. And I already told you that Mary would say to me, oh, we're talking about the eat me thing again. And so I often now talk about eat me rather than outer segment phagocytosis, so don't get offended if I say eat me to you at some stage today. Okay. The other part is, Jerry, I use this line a lot. When I asked Jerry why he felt it was important to be involved in this, he said it humanized it humanized research for me. And I think he meant that at so many different levels, but that actually just shows the motivation and that everybody actually benefited from this PPI. So I'm encouraging PPI, and I know you're going to have a session on this tomorrow, which unfortunately I can't attend. So one of the things that we did was, we said if we put back in this RAB28, now this is firstly the fish, we put back in the RAB28 gene to the knockout, just into photoreceptors, because it's expressed everywhere, put it back into photoreceptors, and the nice thing was it restored these peaks and troughs of outer segment phagocytosis. So whatever RAB28 is doing, it's doing it in the photoreceptor, not the RPE. So that was good for proof of principle. But the second thing that allowed us to do was, can we now test patient mutations in RAB28? So John created the human RAB28 gene, and then he's picked 2 patient variants that are well known to be pathogenic, and he's created those, and we have humanized the fish. So it's the fish no longer having the fish RAV28 and now expressing human RAV28 in their photoreceptors. And on the first slide, you can see that, to make it easy for our researcher, for us, he's tagged them all with green fluorescent proteins, so we can see where this protein goes. The nice thing is the human RAV28 goes to the photoreceptor outer segment where you'd expect it to be. The first patient variant looks like it goes there, but not to the same level. And the second patient variant gets stuck. It doesn't get to the outer segment at all. So this means we now have a platform or a pipeline in which we could test the pathogenicity of patient variants. The last part of this is just to say that the human gene also rescues the outer segment phagocytosis phenotype. Okay, I have to be careful of that because this is preliminary data with an n equals 1, but I think we're fairly confident from the other the rest of the analysis that that'll be true. Okay, so last thing, I'll try and skip through this more quickly. One of the things we do in the lab that I really like to be able to do is screen for drugs that restore vision. We have different ways of doing it, like taking some old drugs and repurposing them, or doing an unbiased drug discovery. So this is a bit old. This is actually from 4 years ago. And again, this is with a HDAC inhibitor. It's a selective one. It's a HDAC6 inhibitor. But what I'm showing you on the screen is a genetic model in zebrafish where they have very impaired vision. That's the one shown in the green dots. And then we treat these fish with the HDAC6 inhibitor, their vision improves. So very significantly improves with this HDAC6 inhibitor. People hate HDAC6 inhibitors or HDAC inhibitors in general in one sense. It's not the thing we really wanted to follow up on, but of all the drugs we have tested, these HDAC6 inhibitors give robust, reproducible rescue of vision, and we'd like to understand why. I'll skip the videos because they take too long to play. This was on halt. People have talked about funding a lot. It's hard to get funding. Projects get stalled. So we had nobody working this for the last 4 years. And then Patricia, with some funding from IRC and Fighting Blindness, is a postdoc in the lab that has rectified this. And we have a collaboration with a group in Italy that have a HDAC6 inhibitor called Repistat. And on the slide, I am just showing you that, again, in the genetic model of blindness in fish, this Repistat HDAC6 inhibitor significantly improves vision. And the other one, the other model we use is an environmental model. So on the slide, it looks like the fish are smoking cigarettes. So that's not exactly what they're doing. We take cigarette smoke extract, we buy it, we add it to their water, and that causes oxidative stress most likely on the fish, and they have a visual impairment. And when we treated with the HDAC6 inhibitors again, significantly restored their visual function. So we have a genetic and an environmental model where these drugs are able to restore vision. So it's not just one model, there's several models, and Patricia is interested in testing some more. The last thing I'll say is we have a very complicated way of screening large numbers of drugs. So large is not like high throughput, but medium throughput. So we have a way of drug pooling drugs and then assessing the effects of those drugs restoring vision. We had a hint. We only got to screen 720 drugs in the library initially before Justine, who was doing this, left. We got a couple of potential hits, and I'm delighted because I love doing this sort of stuff that Patricia and Jody are now starting this back up again, and we hope to screen a library of 5,000 drugs. So these are random chemicals. This is completely unbiased, and you look to find a drug that restores vision, and then we'll look to push it through other model systems. And with that, I know we're all looking to catch back in time. I think I've told you the conclusions. We're still very interested in understanding these two processes, outer segment phagocytosis and the visual cycle. We think zebrafish is a good model to looking at the pathological resolution of some variants, especially when your readout has to be something like vision or visual cycle or outer segment phagocytosis. And I'd love that we could complete a large-scale unbiased drug discovery and find hits that preserve visual function. And with that, I just thank everybody who's involved, all the funders, and thanks for listening to me today.
Fantastic, Brendan. You covered a huge amount of ground there. It was wonderful. So we have time, for, I think, for one question, if there's a question.
So I'll keep it brief. As I understand, Stargardt's is roughly within this mechanism. Is this part of the research? Is that a future for potential Stargardt's or ABCA for therapies? So that's not gene therapy specific, but working on the mechanism that is being impacted by the dysfunctioning proteins.
So ABC, I always say it the wrong way, ABCA4 is involved in, a loss of that causes an accumulation. So in that case, we'd be trying to block the visual cycle, like some of the drugs you've heard about, mixostat and fenretinoid. In this case, these are proactively making the vitamins, so they're there to make the light-sensitive form of vitamin A. So in these cases, we're actually trying to rescue the visual cycle, where in the Stargardt, you're trying to probably dampen it down. So you have two different ways of coming at the visual cycle. So in one case, you want to block it, and in one case, you want to mimic it, give drugs that mimic it. Thank you.
Okay, thank you again, Brandon. Okay, to finish off this session, we've got Professor Sarah Doyle of the Trinity Institute of Neuroscience, also local here. Welcome.
Thank you very much. Now, I'm very aware that it's me standing between all of us, my stomach and lunch, so hopefully I'll keep to time. So today I'm going to talk about the immune system. So we've heard a lot about kind of targeting very specific genes that are involved in inherited retinopathies. And in my lab, we look at the immune system's role in retinal degeneration across a number of different diseases. We're pretty much disease agnostic because our immune system is involved in all responses to tissue damage. And so it's particularly our innate immune system that we're interested in, and that's because our innate immune system can respond to what are called damage-associated molecular patterns, or DAMPs. And these DAMPs are generated in a completely gene-agnostic way through injury to tissue. And so that can be injury due to trauma, it can be injury due to genetic mutations, or it could be acquired from cigarette smoke or anything else that might give you, that might damage the cells. So, for example, in diabetes, excess glucose, things like that. So we're kind of looking at this overarching gene-agnostic approach to understanding how retinal degenerations might progress in a gene-agnostic way. So there are lots of different ways or means that can provide DAMPs within the eye. Simply break down products of extracellular matrix, which is the kind of matrix milieu that our cells sit in. If they are broken down due to tissue damage, then that can actually drive various different inflammatory processes. And similarly, if you have cells that are dying kind of through trauma, then those cells will release DAMPs that are supposed to be kind of held inside a cell and are very safe when they're inside the cell, but when they're released to the extracellular space, they signal to our immune system and to the surrounding cells that there's damage here that we need to clear. And ultimately, that's what our immune system is trying to do, is trying to repair or resolve any tissue damage. And it does this through the activation of pattern recognition receptors, or PRRs. I don't know if does my laser work if I do it? Oop, jeez. This here. So these are pattern recognition receptors, and these are the innate immune receptors that respond to these DAMPs. And once they respond, they induce a very strong genetic signature that enhances the inflammatory process, pulls in and attracts various different other immune cells, and then ultimately should lead to tissue repair and resolution. However, what happens when you have a situation where you have the persistence of these damage-associated molecular patterns, or DAMPs, which occurs in chronic disease, then you ultimately end up with chronic inflammation and tissue damage that is occurring because of the bystander damage through activation of immune cells. And so instead of getting repair and resolution, you get this persistent inflammatory process occurring. So when it comes to identifying targets then, what we like to look at is these pattern recognition receptors of the innate immune system to see if we can see if inhibiting them or reducing their ability to signal might actually reduce the chronic inflammation that occurs in the retina. And so there are literally thousands of proteins and genes involved in the inflammatory system. So how do we start to kind of focus down? Well, we can learn a lot from looking at non-ocular disease and chronic inflammatory diseases that have nothing to do with the eye, but that are also driven by these pattern recognition receptors. And so one of the families of innate immune receptors that we look at is this toll IL-1 receptor domain superfamily. And there's 3 families within this superfamily: the IL-1 receptor family, the toll-like receptor family, and then the TEER adapter family. And so these are all really, really pro-inflammatory receptors. They are either able to respond directly to DAMPs, and the key there is that they're responding directly to them. So these are the primary initiators of inflammation in our bodies. Or they are DAMPs themselves. So I'm going to stop using my laser, I think. Some of the cytokines that are activating these receptors are actually DAMPs themselves. But today I'm going to talk to you about one of these proteins. It's called SARM. And the reason I've chosen to talk about SARM today is because Fighting Blindness actually funded all of this work, so I thought it was appropriate to talk about that. So SARM is one of the TEER adapter proteins. It's one of the 5 TEER adapter proteins, but it is by far the most evolutionarily conserved adapter protein. It's actually conserved across all biological kingdoms. Plants have it, sea squirts have it, worms will have it, and zebrafish, mice, and humans. Like, it's really incredibly conserved, which really goes to show us how important this protein is in some function. The interesting thing is, though, despite its really, really highly conserved status, it really wasn't known what SARM did until about 20 years ago. So SARM was found to regulate the innate immune system, but a very small amount of work has been done on this, relatively speaking, because about 12 years ago, it was found that SARM actually has a very, very powerful role in neurons of the brain. So this is really interesting for us because we're interested in the immune system and also because we're interested in the retina, and the retina is an extension of the brain. So now we're able to apply knowledge from chronic inflammatory disease and potentially neurodegenerative diseases of the brain into our therapeutic target identification for the retina. And what was found was that endogenous SARM, so that's SARM inside neurons, was able to promote neurodegeneration. And so if you were to activate SARM through trauma, so in this case, these are peripheral neurons, and they've been cut. And you can see on the bottom row there how if you don't have SARM, so if you have SARM removed from the system, those neurons are really, really protected by comparison to the top row where you have very, very clear neuronal degeneration. And in fact, this SARM1 triggered axon loss or neuronal degeneration is conserved also across biological kingdoms. This is its main role. So it seems to be a metabolic sensor. So I talked about how the innate immune system senses damage or danger. And what SARM seems to be doing is sensing when a cell has really low levels of energy. And so, and kind of cells, what SARM will do then is it'll just kill the cell, so you're not wasting energy trying to keep it alive. However, for photoreceptors or for retinal degeneration, we want to try and keep these cells alive, and help them if they are struggling. And so what SARM is doing is it's recognizing this kind of switch or increase in NMN NAD plus ratio, which is basically telling the cell that it's struggling, and it cleaves any NAD plus that's left, and then you have a complete energy crisis, which leads to degeneration. And the reason that you get this energy crisis and degeneration when you activate SARM, is because SARM cleaves NAD+. And NAD+ is absolutely critical as a cofactor for our energy metabolism. So glycolysis and the Krebs cycle are really important for generating ATP, which is the energy molecule of our cells. And so when you don't have NAD+ there, you actually can't drive glycolysis or the Krebs cycle, and so you get a complete loss of ATP and cell death. So that's what SARM does, leading to this energy crisis. So why is this important for retinal degeneration or photoreceptor cell degeneration, which is what we were particularly interested in when we started to look at this? So there's a couple of reasons. We know that the retina is really highly metabolically active, so it requires the ability to be able to generate lots of this ATP to work. NAD+ is a necessary survival factor for photoreceptors. That work has already been done. And in all models tested of retinal degeneration, blocking apoptosis, which is one type of cell death, has never been sufficient to prevent photoreceptor cell death. So we asked the question, is there a role for SARM1 in photoreceptor degeneration? So the first thing we looked at was whether SARM1 was actually expressed in the retina. And we found that, interestingly, SARM1 is most highly expressed in the retina, only second to the brain, to fetal brain. So it's really, really highly expressed in the retina, which again would indicate that it's probably going to have a role there. When we looked to see where in the retina it was expressed, we were able to show that that the SARM1 was mostly expressed in the neural retina. So we split this is mouse tissue, so we split mouse tissue into neural retina and the RPE choroid, and we really saw absolutely no SARM1 in the RPE choroid. And it was so clear, but I was so skeptical. We did mass spec analysis on it as well, and again, really, really clear indication that SARM1 is very highly expressed in the retina, but not in the RPE. So lots of neurons in the retina, obviously. So we were kind of going, well, is it in all of them or is it in photoreceptors? And so we used the rhodopsin knockout model, mouse model of photoreceptor degeneration to kind of answer this question. Because in this model, you have no functioning rods, and the cones die off very fast. And so over time, you get basically no photoreceptors in this model. And so what we were able to show was that in the rhodopsin knockout, the level of SARM, and this is it quantified over here, is much, much lower in the row knockout compared to wild type. So kind of telling us that the majority of SARM is expressed in the photoreceptors. So knowing that then, we wanted to know if we could actually activate SARM in the photoreceptors. And so we used a method called fluorescence lifetime imaging microscopy, or FLIM, on tissue explant from wild type and SARM knockout mice. And so what you're seeing here is a pseudo-colored image of the NAD plus NADH pools. So what FLIM allows us to do is use their natural kind of excitatory fluorescence to pick up the different amounts of NAD plus NADH pools within the photoreceptor. So because of the way we can use the two-photon microscopy, we can also say for sure that these are the photoreceptors that we're looking at. And so we used a toxin, a mitochondrial toxin that we know activates SARM, called CCCP, and added it to these explants. And we could see that when we had explant that was deficient in SARM1, that we had a greater amount of NAD plus NADH pools. That's up here. And so now we knew that SARM1 was expressed in the retina, in the photoreceptors, and that it had a function in these photoreceptors. It could cleave NAD. So we crossed the SARM knockout mice to the rhodopsin knockout mice and created these double knockouts. And this is on the bottom row here. We have the double knockouts. On the top, we have the rhodopsin knockouts by themselves. And so you can see over time, this layer here is the photoreceptors, and you can see that over time in the rhodopsin knockouts, you end up with 1 to 2 rows of photoreceptors. Whereas when we lost SARM, when we had SARM deficiency, we can see that we were able to protect a number of these rows of photoreceptors. And what was really interesting, I think, was that there seemed to be kind of a gene dosage effect. So in mice that had one SARM still expressed, or one copy of SARM still expressed, we were able to see that there was a kind of a midway point of protection. So even heterozygotes for SARM were protected, or for SARM deficiency were protected in this model. When we looked at flat mounts to have a look at the actual cone pedicles themselves, we were able to see that, again, in the SARM deficient mice, we were protecting many, many more cones from cell death compared to the single rhodopsin knockouts. And we were able to show that this was likely due to the fact that in the SARM-deficient mice, we had this extra amount of NAD plus NADH pools. So it's probably just allowing more energy to be efficient in these mice. So that's what we're seeing here in the double knockouts. But that's all very well and good, but kind of can we rescue function? And so we used ERG to measure the function of the photoreceptors in these mice. And what we saw was that in the row knockout mice at 9 weeks here, we're getting very, very little in terms of the B-wave cone response, whereas we're protecting that when we have the double knockouts when SARM is deficient from these mice. And by 12 weeks, it's almost flat line here, whereas again, we have some protection in the SARM knockouts, and we can see that in the flicker wave response as well. So removing SARM from the retina in this retinal degenerative model actually protects the cone function as well as viability. So it's kind of interesting to think what might actually be activating SARM in vivo. There's lots of different possibilities, and one of them is oxidative stress. And oxidative stress is an interesting stressor for the retina because we've seen across multiple different types of retinal disease that there is an increase in oxidative stress. And so we used the sodium iodate model to see if, by inducing really acute oxidative stress, if we could also protect photoreceptor cell death in the SARM knockout mice. And so this is a really acute model, but within 3 days of sodium iodate administration, we can see that in the SARM knockout mice, we had very, very clear protection of the photoreceptors. So again, it's cross-model. And then because cell death doesn't tend to occur in isolation, or types of cell death don't occur in isolation, we started to look at whether there was a crosstalk with other cell death pathways. And actually, we did see that there was a reduction in apoptotic pathways in the SARM knockout mice as well. So this is something that we can continue to look into. And I think the potential for therapeutic inhibition of SARM1 is really, really strong. So already there is a gene therapy for SARM1 for mice, where Jeffrey Milbrandt and his group over in the US have shown that this gene therapy works really well for peripheral neuropathy. And in Lilly, they have used small molecule inhibitors to show that a small molecule inhibitor of SARM can protect against peripheral neuropathy as well. So I think there's hope there that SARM1 might act as an adjunct therapy or another option for slowing progression of retinal degeneration. So I just want to mention the people in my lab who did most of this work. Luke Gibbons and Emma Ozaki did nearly all of the work shown today, and thanks to the funders, especially Fighting Blindness and HRB, who funded this work. Thank you.
Fantastic, Sarah. So we have time for a question or two. One over there. We got a mic over to the right, please.
Thank you. Great talk. Do you think variations in SARM1 alone could be a cause of retinal degeneration?
So that hasn't been shown in that I haven't come across SARM1 on RetNet or anything like that. Possibly. I mean, I think there's room for that, but I think it would probably also have an effect in the brain as well, in the CNS. Yeah.
Thanks.
Okay, if there are no more questions, I'd just like to maybe one final question here.
Thanks for that great talk. You said SARM1 starts, it kills the cell when it's low on energy, but is the cell still functional at that time? Did you look into functionality of the cells as soon as the SARM starts killing the cells? Or is it just there, still alive, but not functioning before the SARM attacks it?
So I think that once SARM is activated, the killing is almost instantaneous. But I think what it's doing is it's sensing cells that just have that kind of, they're struggling a bit to make enough energy. And so I think what we can do is to come in and block SARM from working so that the cells are still absolutely functional. They're just, maybe they need the Sophia's drug as well to kind of hoist up the mitochondrial respiration a bit and then block SARM from killing them. So.
Okay, thanks.
Great, thanks, Sarah. We'll have a short announcement in a second, but I'd just like to thank all the speakers who spoke in this session for some really wonderful talks. Thank you very, very much.
So just to add, I'd like to thank everybody for both sessions as well. It was fantastic, fantastic morning. But I think we're more worried about your tummies than anything else. And so although we're running a little bit late, we're going to take the full hour and have lunch and have a good bite to eat and hopefully discussions over lunch and chats. And we'll see you in an hour's time. Okay, thank you.
Great. I'm just a minute or two early, but it's just to encourage people to take their seats. So we had a call of an hour ago. Jane said to be back in the room in an hour. So I read that as 2:45. And while I know we've had comments about Irish time, we will try and keep strictly, because we are running 45 minutes behind schedule. So we just take our seats for the next session. And if anybody's near the back, do they mind just giving a little shout out to anybody hovering around in the coffee area to come in for the next session? Please. Okay, we're going to give it 2 minutes. 14:47, we're going to start with Professor Elaine Dolphus in 2 minutes. Oh, sorry. Professor Bartleroy, excuse me. So the format we're going to use here, just while you're taking your seats, so we're going to have a talk of about 15 minutes, and we'll have time for questions after each talk. And then if we do manage to carve some time back, we can try and have a group discussion at the end around the challenges around the genetic architecture in IRDs. I think with 20-minute talks, we're going to give a chance. We're going to stick to time, and we're going to wrap up the session and at 3:45, as we're going to hand over to Professor Silvestri and the AMD session. Okay, I got everybody in the room. So we are going to start the first of the afternoon sessions. Thank you very much. I hope you got to enjoy your lunch and catch up with people and see some of the posters and visit some of the exhibits upstairs. We had an excellent morning session, and we'll keep up that quality for the afternoon. So this session is on genetic architecture and its relation to inherited retinal degenerations. I'm delighted to welcome an expert in the field and a very good friend and colleague, Professor Bart Leroy from Ghent University, who's going to speak first on the issue of genetic architecture. And then that will be followed by Professor Elaine Dolphus and then Professor Franz Kremers. So we're going to have going to have the presentations, and then we'll take 1 or 2 questions after each one. Bart, the floor is yours.
Thank you, David. So I've structured it such that I'm the introductory person, by training medic, ophthalmologist, and clinical geneticist. So I work mostly in Ghent, Belgium. And I'm going to use slides mainly for myself. I'll be very descriptive for those of you who can't obviously see many of them. So I work with a lot of companies. I don't get the money myself. We pay our ophthalmic clinical trials unit with the money. Basically what I'm going to try and run through is quickly introduce concepts that you've heard about, and then go through a couple of diseases and leave you with a couple of research thoughts. And so we all know, as Dan was saying earlier, that our eye translates light into electrical signals. We do that at the level of the retina. We use 2 types of photoreceptor cells to do that, rods for night vision, cones for daytime vision. They connect up to other cells, and then these other cells finally bring it through the optic nerve to the back of the brain, where we realize what we see. So the interpretation of what we see is done at the back of the head in our brain, the visual part of the brain, right and left-hand side. Cones and rods are the 2 photoreceptor cells. We have outer parts of these cells that actually sit engulfed within the retinal pigment epithelium, and that's where the thing happens. What? Biochemical translation of light into electrical signals. I often, when I zoom into that outer part of a rod and a cone, think about it as a stack of discs. And this stack of discs, you can imagine, is a stack of pizza calzones. So pizza calzones are the pizzas with the good bits inside, and the dow at the, on the outside, and floating in the dow of the pizza calzones are multiple proteins that eventually help as a biochemical domino game to translate light into an electrical signal. We understand many of those proteins, they're encoded for by genes, and those genes can be mutated and cause retinal degeneration. We also use vitamin A as an intimate partner to the first domino block in the domino game of translating light into electrical signals. And that vitamin A needs recycling because we use it over and over again, millions of times per second as we look at one another here in this room. And we need to recycle that, and we do that at the level of the retinal pigment epithelium. It's kind of the mother cell of the retina. It does many more things than just recycle vitamin A. So just put my hat on as a clinical geneticist and mention that we have 46 chromosomes, or large blocks of genetic material visible through the microscope in the nucleus of every cell. There are 23 pairs, basically, because we get through the sperm cell from dad 23 and through the egg cell from mum also 23. Males and females don't differ much. It's the 23rd couple of chromosomes that differs XX in females and XY in males. In total, we think that we have about 20,000 genes times 2, and of those, 325 genes approximately are involved in retinal and optic nerve disease. So what is important is that you know your gene, and referring a patient to an ocular genetics specialist is, even in those days, and even in countries like Ireland, Belgium, the UK, France, the richer countries, I can say, of what is the EU, even there I see quite a few patients who don't have a molecular genetic diagnosis. And there's a reason to have that. We are going more and more towards precision medicine. For example, in infectious disease, people are now categorizing much more on the infectious agents, and we are more and more categorizing based on the gene that we say first, and then we add something like IRD, inherited retinal disease, or maculopathy, or rod-cone dystrophy, etc. So let's look at what are the large categories of the generalized retinal diseases. And RP, retinitis pigmentosa, is actually a bad old name that I still continue using as well. But basically, it is a rod-cone disease. I say first what is most affected. What you get is first night blindness. Then after the night blindness period, you also are losing your ability to navigate without bumping into stuff, which means that the cones during the daytime also lose their function and eventually die off with the rods, and you end up with a tunnel vision. There are many people who are much more specialized in what I just said than myself in this room. So the background is genetic. It can be dominant, recessive, and X-linked. I don't want to dwell on this. We know multiple genes. In total, 24 genes known for dominant disease. They're very different. The mechanisms underlying those conditions are thus very different, but there's a final common pathway. And so, for example, in the slide that I'm showing now, you can evaluate a hyper-intense white ring in the center that is surrounding that part of the retina that is still working. Why is that still working? Because it's incredibly cone-rich. And remember, RP is a rod-cone disease. X-linked retinitis pigmentosa exists. We have a condition that is very similar if it's X-linked, recessive condition, 53 different genes known. So we're going slowly towards 100 genes that we know for retinitis pigmentosa. And if we look in an eye of a patient, they all look very similar indeed. We have to think, however, that sometimes clinically we can make a difference between what is referred to as a patient with retinitis pigmentosa, and they will suffer, for example, from a condition that we call choroideremia. Why is that important? Because different therapeutic pathways are going to be available in the future. And so obviously with molecular diagnosis, as you will hear from Professor Dolphus and Professor Kremers, is helping us towards that end. There's another condition that we call gyrate atrophy, and that is a condition with diet that you can potentially do something about. There's another condition, for example, adult Refsum disease, where when you give a diet, those people that can be life-saving for those people. So sometimes as an eye doctor, I ask people, can I see your feet? And people obviously don't prepare themselves often when they come and see me to show their feet, so they're embarrassed oftentimes, but I do have the ability to wash my hands, so that's not a problem. So I'm going to quickly dwell on Leber congenital amaurosis, named after Theodor Leber, a German gentleman who described quite a few conditions, and he described a condition where young children get born and barely see. The condition is typified by a mother and a father who feed the child, and they don't get that eye contact that you normally get. We now have 24 genes and counting that can be at the basis of this genetic condition where no electricity is made in the retina or barely any. There is a continuum, obviously, because we can categorize, but there's a continuum in the sense that obviously you also have people who are fairly normal at birth and then in the first few years lose that ability. And so there's a continuum of conditions. It's not one and the other. No, there's a continuum. So obviously, when I spoke about recycling vitamin A, I spoke about the fact that you need to throw that vitamin A that is used in the translation of light into electrical signals. You have to throw that into the retinal pigment epithelium cell. That is going to recycle it and throw it back to the photoreceptor for a new cycle to begin. And there are some proteins that are essential to do this. There's one on this slide that I colored blue, which is the RP65 enzyme encoded for by a gene that we can now bring, thanks to the work of Professor Bennett and co-workers, we can bring that to the patient as Luxturna. So I'm not going to dwell on a couple of different types of Leber congenital amaurosis. I said 24 genes. Again, the retina doesn't translate light into electricity as it should be from birth, and very different genes at the basis. We also can have cones at the forefront of disease. We have cone dysfunctions. These people will confront us with, I hate intense light, I can't recognize you at the other side of the room, and I don't see color, because those are cone functions. You can have cone rod dystrophies, not only cones, but cones and then rods that get involved. Many different mechanisms at the basis of this condition. And then we have stationary conditions. We have a whole group of conditions where you can be troubled at the level of vision, for example, in the group of conditions that we call achromatopsia. Now, literally translated, this means no color vision. Now, obviously, there's more to that. The cones are there, likely, but they simply don't work. So people are photophobic. They don't like intense light because they use their rods to observe the environment. They have nystagmus, or searching movements of the eyes, to try and find that better vision that never comes. And they have poor visual acuity with no color vision, but when we look in the back of an eye, it looks entirely normal, oftentimes, unless you do that later in life. Sometimes on scans we can see of the retina, we can see that the center is devoid of the photoreceptor cells of the cone type. But generally speaking, they look fairly normal. And we now know that there are 6 genes that can be at the basis of this genetic trait. They all need 2 mistakes, a mutation in both copies, for you to be affected and for your cones not to work, with 5 of the 6 being involved in that cascade that I was talking about, that domino game, specific in this way for the cones, translating light into electrical signals. And I'm not going to dwell on that. I am going to quickly touch upon a second stationary condition, because with achromatopsia, you can tell the patient and the parents, your vision's not perfect, but there's very likely no change to be expected, which is very important for a patient, obviously. There's also congenital stationary night blindness, a group of conditions, very different actually if you look at the mechanisms, but suffice it to say that those people have a better cone function than their rod function. The rod system simply doesn't work. Sometimes they look like very myopic when we look at the back of the eye, but we can't really see anything more that is abnormal. And the mechanism is very complex. I can give you a 2-hour talk on how that really works. I'm not going to do that to you, but suffice it to say that most of the congenital stationary night blindness patients have an issue of getting the electrical signal from the rods and the cones to the next level up, which is the bipolar cells in the retina that then give the electrical signal to the cells, the ganglion cells that make the optic nerve. So I don't have to dwell on that. And the last little thing I wanted to address are dystrophies with predominant macular involvement. And I can be short about a condition that is very frequent because we have the Stargardologist in the room, who is Professor Kremers. He will be talking about this, but I'm using this as a platform to show you something. So Stargardt disease is a condition where we see white flecks in the area of the macula or central retina, and the patient often presents with loss of vision early in life after a period of normal vision. And we have to think about differential diagnoses or a group of conditions that can look like Stargardt when we look in the back of an eye, but are caused by other genes or mutations, mistakes in other genes. And we, in Ghent, we saw a total of 60 patients where we saw something that looks like Stargardt, but we tested the ABCA4 gene that you'll hear about in the talk by Professor Kremers, and nothing came back. We tested all the other genes that we know can mimic Stargardt disease, and nothing to be seen. And so the slides that I'm showing, for those of you who can see this, the patient's phenotypes look a little bit like Stargardt disease, but again, we didn't find causative mutations. And the brilliant team of my colleague and friend, Professor Elfriede de Bara, went through the molecular data, and we identified a new mutation that is a Belgian founder mutation in the RPE65 gene, which is the gene, if you have 2 mutations, you have early onset disease and you can have treatment with Luxturna. We are in Ireland, and there is an Irish mutation that in that same gene, in one copy of that same gene, that causes yet another condition where you have atrophy of the retina much more extreme. Whilst in the Belgian patients, we noticed that it looks like Stargardt and it really affects the central part of the retina. It does not affect the peripheral part. So this looks a little bit like the same situation that we have, or a similar situation that we have in the conditions that we call bestrophinopathies. So bestrophinopathies are a group of conditions. The Best disease is called after Friedrich Best. He, in 1905, described it. He was an Englishman, I believe, but Friedrich Best was a German gentleman. John Adams had described that in 1889 earlier, but that was not recognized. And we found mutations, different mutations in that same gene that can cause different conditions. So what I just wanted to say with these couple of slides, these last slides, is that it is very complex sometimes, but we are discovering more and more. So the last 2 slides, I'm going to just say you need your genotype. You need to know which gene is at the basis of your condition. It gives you closure if you know what you have. If you know that it is not syndromic, but isolated disease, or vice versa, you know what to expect. You know what is the recurrence risk for my children, and we can have you benefit from options for pre-implantation and prenatal diagnosis. And we need your gene for most of the gene therapy trials that are coming. So basically, what you need to do is demand that you have your gene known. As you will hear from my fellow speakers, not every gene is found in every patient, but we're working very hard to that end. So basically, I want to thank the group of people that I am blessed to work with in Ghent and previously also in Philadelphia. And thank you very much.
Bart, thank you for a fantastic talk. So have you coordinated with Elaine that Elaine speaks, and we'll do a question for both of you afterwards? Yes. Okay, you prefer to that. Okay. It's a real privilege to introduce Elaine Dolphus, Head of Department in Cargo in Strasbourg, a geneticist, very good friend, but also, crucially, our leader of the ERNI group, and who myself and Bart and many others report to. Thanks, Elaine.
Thanks a lot, David, and thanks to all the organizers. It's really a fantastic meeting. So I will go for the genetic testing for retinal dystrophies, and it's a perfect match with the Bart, because my first slide is like his last slide, and indeed, genetic testing does matter for the patient. It's the first concern for genetic testers and for genetic for people who are in this field is what matters for the patient, and to know precisely not only that you have a retinal dystrophy, but what gene is involved is really very important. And this, as Bart said, for the genetic counseling, the mode of inheritance, pre-implantory diagnosis, prenatal diagnosis, and also, of course, and we have heard about that all day long, about the therapies, because the therapies, most of them are gene-dependent, and even if they are not gene-dependent, I think it's good to know the type of gene involved in the patient's phenotype. The other thing is that there is a huge number of genes involved in retinal dystrophies, more than 300. And also, with the new technologies, we realize that our dogmas are a little bit falling, and what we call genotype-phenotype correlations is something that is much more complex than we imagined before. So, with the progress on the scientific biotechnological progresses, we are really getting deeper in the knowledge. We are bringing better diagnosis, but we also have a lot of questions rising. And today, as I remind you, we had the DNA discovered in the 1957, and today in 2024, we are able to take a single cell and do a very precise molecular biology on just one cell. So we are really very powerful, including the fact that we can sequence the whole human genome, and also for diagnostic perspectives. So you were reminded about the number of genes we have in our huge genome, 3 billion base pairs. You were reminded that it's like a huge encyclopedia with recipes, and the chromosomes are like books where all these 20,000 genes are coding for specific recipes. And if you have a mistake in one of these genes, well, you have really problems, and if it's a specific gene for retina, then you have retinal degeneration. So what you have to realize is that most of the mutations, what we call pathogenic variants, are really in a very small part of the whole DNA. It's in the coding part, the 1% of the whole DNA where there are the genes, the 20,000 genes, and this is what we call the exome, and 85% of the pathogenic variants of the mutations are located in this coding area. If you sequence the exome, you can find a lot of mutations, roughly 85% of cases, but you have also variants that are in the other parts of the genome or that are not detected by the current technologies. And whole genome sequencing enables to detect other types of mutation types, and we will see that a little bit later on. The technology has evolved very much from what we call the gold standard, that is really the old way, the Sanger sequencing, where you were taking each base pair after each base pair, extremely long, extremely not costly, but extremely long. And then we were able to test what we call panels of genes. That means taking 10 or 20 genes or even 100 genes that are responsible for a specific group of disorders by sequencing technologies that were novel next-generation sequencing technologies that have evolved towards testing the whole exome, the 1% I was speaking of just before. This is called the whole exome sequencing. So you can test the 20,000 genes, but only in the sequencing aspect. And then you have the whole genome sequencing that would test the whole genome, and that will also provide a huge amount of data. And Jane Farrar was saying, yes, indeed, a huge amount of data, and finding what is wrong in this huge amount can be a challenge. So bioinformatics is really something really important for whole genome sequencing. And there are different steps. I will not get into the details here today. But overall, if we consider all these techniques, today, if you look at the literature for retinal dystrophies, panel exomes and genomes, we have a positive diagnosis according to the different cohorts between 65 and 85% of the patients, meaning that 35% to 15% are still without any genetic diagnosis. So these are numbers that are taken from the literature, and the cohorts in the literature may be quite different according to the type of ERD you are considering. So this explains why there is this variability with the successes in diagnostic testing according to different groups. So I would like to focus now on how we work in France for providing genetic testing in our country. And also would like to stress that the European Reference Networks is also very concentrated on the problem of genetic testing. We have a specific group that is led by Elfriede Debert and Susan Cole. And we have actually tried to map where are the needs, and we have seen that in Europe there is a big deal of heterogeneity. But now I will focus on the French system. Just to set the scene, we have 24 networks in France for rare diseases, one of which is dedicated to sensorial diseases, so including eye diseases. And so we are more than 70 different types of centers. I'm not getting into the details here, covering the country. And in parallel to the rare disease plan, there is a genomic French medicine initiative that is carried out for cancer, for common diseases, but also for rare diseases. And to begin with, this initiative was based on 3 research pilot projects, one of which dedicated to rare diseases, that was on intellectual deficiency. And I was personally very much involved in this one, and I will give you an indication at the end on how a genome can really be useful for non-coding variants. So the organization in our country is based on 2 platforms that are doing whole genome sequencing, covering each half of the country. And there are today 79 rare diseases who can benefit from whole genome sequencing. And in the rare eye diseases, we have 3 types. I will get into the details in a minute. So just so you understand the system, and very briefly, the French initiative is based on the healthcare system, but it also collects data for research. And in the future, these genomes will be used for research purposes. And Jane mentioned gene modifiers, and indeed, you will have to need a lot of data to identify this type of modifiers. So there is a collection of data that will be used for research afterwards. But what is important is the healthcare pathway. So when the patients enter this genomic initiative, all the university hospitals are trained to have a specific pathway with a medical consultation, the consent form of the patient, e-prescription. Then the sequencing is done on one of the 2 platforms after the blood is drawn for a trio in a family, meaning the parents and the children. This is the typical case, but there can be variability. Then we have a clinical biological meeting that can be held either before the prescription, but also after the prescription. And we deliver then a result to the doctor, who will deliver the result to the patient. So this is very well organized. And we also have in each university hospital a new type of profession that are genetic assistants who are helping the clinician and the whole system to draw the blood and to obtain the consent, and that all the system works. And it's really very useful. So for the clinical pre-indications, I told you there are 76, and 3 are dedicated to rare eye diseases. The first on inherited retinal dystrophies, the second on ocular manifestation. A very recent one was on optic neuropathies, and another one is on early onset deafness. So in fact, there are 4, but 3 on rare eye diseases. So we are a group of ladies, actually. You can see here girl power, the French girl power, with geneticists, ophthalmologists, yes, molecular biologists, and we are responsible for the good, so that it goes in the good way. So just to tell you, if the diagnosis is evident, for instance, choroideremia, we will not ask for a whole genome. So this is only done if the patient comes in and we have no obvious diagnosis, usual retinal dystrophy. It can be retinitis pigmentosa, for instance. And today we have switched from panels to genome first, meaning that the genome is analyzed sequentially. We don't analyze the whole genome in one go. It's analyzed first on a panel of genes that are responsible for retinal dystrophies in silico, so it's what we call an in silico panel. Then it's analyzed for the whole exome, and then more for if we don't find anything. So we have performed today, and it has been launched in 2022, more than 1,500 tests, and we have 60% of our cases solved today. And we deliver to the patient class 4 and class 5, and the class 3 are not reported to the clinician. So what is very important for us is the multidisciplinary meetings we can have before the prescription, and also once we have the result, because sometimes it doesn't really fit what we were expecting. So it's very important to have these meetings. So now I'll go very briefly through a number of cases to illustrate why whole genome can really be useful. So very briefly, a case of a female who is nearly 70 years old and who is deaf and who has retinitis pigmentosa. So we all think about Usher syndrome, but the panel was negative, especially the Usher panel, and this lady had no diagnosis. Now, using one of these whole genome platforms, we realized that, in fact, this lady has not Usher syndrome, what she thought she had her whole life. But she's carrying mutations in 2 autosomal dominant retinitis pigmentosa genes, and in 1 autosomal dominant deafness gene, meaning that the genetic counseling is totally different from what we expected at the beginning. And this illustrates what we call multi-allelism, meaning that in the same patient you have 1, 2, or even sometimes 3 different genetic diseases. And this occurs in almost 5% of the cases in genomic medicine. The case number 2 is a female patient who has Stargardt disease. When she comes into our clinic, she really had all the signs for this. For years, she had this diagnosis. And curiously, on the Stargardt panel, there was no really solid results. So looking at the genome results, she was found to carry 2 variants in one of the Bardet-Biedl syndrome genes, the BBS10 gene. And when we examined the patient, we didn't find any other symptoms of the Bardet-Biedl syndrome, obesity, kidney insufficiency, etc. So this is something that has been mentioned already this morning. You can, a patient with retinal dystrophy can be carrying mutations in a gene that is responsible for a syndrome without developing all the other manifestations of the syndrome. And for Bardet-Biedl, Elise Zéon and our group have also published a number of these BBS genes that have isolated retinal manifestation. The case number 3 is a young girl. When we saw her, she was young, actually, but she came with her father, and her grandmother has RP. And she also developed symptoms. So in 2006, that's why I say time has passed, we started to explore genetically this patient, and we did all the different tools we had during the years. And finally, with genome sequencing, 18 years later, so this illustrates the odyssey, what we call the patient odyssey, waiting and waiting and waiting for a result for genetic testing, and an intronic variant in the PRPF31 gene was found, explaining also the variability of penetrance in the family. The 4th case is a lady who has very bad vision since childhood, extremely bad. She has had multiple injuries because of her vision. And she has a classical RP fundus, but also a very pale disc, but we do not pay any attention to this. But after a negative panel, we performed a genomic sequencing, and she was found to be mutated in a gene that is called RTNAIP1, which is a gene that was well known for recessive optic atrophy. But, as I said, with all the tools we have, the power of the tools we have, we are discovering new entities. And indeed, this gene can also be responsible of retinitis pigmentosa associated to optic atrophy. So we are learning all the time, and the phenotypes may be not as easy as we imagine to analyze. This case number 5 is a boy, 13 years old, with various serious visual problems. He had the genome first, and he didn't manifest any other problems. But in fact, we found what we call the classical NPHP1 deletion. It's a gene that is responsible for nephronophthisis, which is a serious genetic condition where the people have severe onset kidney failure. And in fact, thanks to this test, we have a precise diagnosis. We know he can benefit from a very strict nephrological workup, which was abnormal, actually. Case number 6, it's my last case, I think. It's a person who had difficulties since 12 years old. And there were peripheral bone spicules discovered at that age. He was 10 at that age. And now we see this man at 33 years old, still with all the symptoms of retinitis pigmentosa. And looking at the imaging and all the functional testing, there is no doubt that this patient has retinitis pigmentosa. So he has been through a number of different panels during the years. And we also performed in this patient a genomic testing. But this testing is negative. So does that mean that it's not genetic? No, that means that finally our technology is probably not powerful enough. Unfortunately for this patient, the odyssey for the diagnosis is not finished, but there are different tools now that are developed to go even further in the analysis of the genome, not only bioinformatic tools, but also ways of sequencing, what we call the long-read sequencing or optical mapping, meaning that you take photographs of the chromosomes. So there are a lot of different tools that are out there to get even deeper in the genome. And I mentioned intellectual deficiency at the beginning of my talk. Just a few weeks ago, a very important gene for intellectual deficiency was discovered. It's a non-coding gene. So it's thanks to the genome sequencing analysis of a lot of patients that this non-coding gene has been attributed to intellectual deficiency, which is one of the major rare disease cause of consultation in genetic departments. So that means that if you have an inherited retinal dystrophy, that you do not have a genetic diagnosis, that doesn't mean there is not one for you that will come later on, and it's not abnormal to be in this situation. This is what I say to my patients all the time. So 75% roughly of the patients are solved, but there is still a lot to do, future research, and I would like to insist on my last word on the importance of working together, the multidisciplinarity between the researchers, the medical, the clinicians, the patients, the molecular biologists, bioinformaticians. This is a whole multidisciplinary adventure. So thank you very much.
Thank you very much, Elaine. That was a fabulous run-through and really illustrative cases. They are the real-world issues we deal with when you run an inherited retinal degeneration service, or indeed any genetic service. Can we distill right through the cases that Elaine showed us there? I think we'll have a few minutes at the end. We'll bring all the questions together. I think there's some themes coming. So it's a great pleasure to introduce Professor Franz Kremers, Nijmegen, as Bart and Elaine have already alluded to, really is a world expert in our field, particularly around Stargardt disease, but also around genetic testing. And there's a fabulous project, so any of the Irish patients in the audience are probably aware, but Franz has been looking deep into your introns, in particular, particular conditions over the last couple of years. So Franz, interested to hear your thoughts on genetic architecture. Thank you.
Thank you, David. Yes, I also was already here 34 years ago. So 1990 was already one of my first meetings, and I was here actually at that meeting. So it's wonderful to be back, and it's a pleasure to give this talk. So this will all be centered around one gene. So I'm taking a couple steps back from the whole genome and going back to one gene. And I think the gene is interesting because we've learned so much over the last couple of years by studying this gene. And so I will hope to give you the highlights of that in the next 15 minutes. So this I will not go into, just to say that ABCC4 is involved in classical Stargardt disease, but can also be seen in cases that have cone-rod dystrophy or late onset maculopathy, and we even have found a case with congenital blindness that has ABCC4 variants plus something else. So when we set out to do the studies about 9 years ago, the state was that we could find causal variants in typical Stargardt cases in 60% of the cases. And in 40% of the cases, we could not find the variants. And I will give you some examples what we did find after all these years. And of course, these elements that I put in here, prognosis is important, closure for the patient is important, genetic therapy may come along the way, and family planning in some families is also important. So the question that I put myself into at that moment is, can we use ABCA4 Stargardt disease as a model disease that shows what can we find in this gene that may also tell us something about all the other genes? And I think especially the last part of my talk will be quite novel for many of you. So bear with me. So I put in this slide that there are three main factors that we are missing at that time. Variants in the non-coding regions, undetected structural variants, so deletions and duplications, and the last part of my talk will be about non-Mendelian inheritance, that I think is also there for Stargardt disease, in the proportion of Stargardt disease. So, what we found for, let's say, all the IRDs at that time, we also found for Stargardt disease. It's similar. 6% solved, 40% not solved. So Stargardt disease is really classically one gene, one phenotype, or maybe a little bit broader. It is the most frequently mutated gene that we know in this whole spectrum. 1 in 10,000 individuals carries variants in this gene that give an eye disease. And the cone vision defect precedes the rod vision defect in most cases. So just give you the location of the protein. It's sitting at the rims of these pizza calzones that Bart described to you. And the reason why it is at the rim is that the intradiscal domain is so large that it cannot be sitting in this flattened part of the discs. So it can only be in this bulge that is at the end of these pizza calzones. And that's probably why it is only at that position. So the protein is a transmembrane protein spanning, let's say, from the intradiscal side to the cytoplasmic side. It transports effectively all transretinal, so let's say the inactivated ligand of rhodopsin, from the inner to the outer leaflet of the disc membrane. About half of the mutations lead to really absence of proteins, which is at the right of this slide. And in the middle, you find all the amino acid changes. We call them missense variants. And they make up the other half of all the variants. For the protein truncating variants at the right, we can interpret them very simply and very easily. For missense variants, that is still a challenge also for the future, because many of them are very rare, and to know for sure that they're causal needs a little bit more studies, which actually we don't do, but should be done in the future. So the spectrum of clinical conditions, and I just put in 3 here, early onset Stargardt disease if you have 2 severe variants in ABCA4, you have classical Stargardt disease if you have 1 severe and 1 mild variant, and you have late onset Stargardt disease if you have 1 severe and 1 mild incomplete penetrant variant. And this incomplete penetrant variant, I will come back at the end of my talk, maybe even the most interesting part of my talk. So in the last, let's say between 9 and 3 years ago, we collected about 2,000 cases worldwide that had unsolved Stargardt disease. They had 1 or no mutation. Most of them were pre-screened, not all of them. Literally from all over the world. So to screen 2,000 cases in a research setting meant we had to develop and design a technology to do this very cheaply. And we did. So we sequenced the entire gene, but just only the ABCA4 gene, in a very cheap manner. I will not go into the details, but we use a technology that is sort of targeted. It's called SM-MIPS technology. And just to say that if you look at the sequencing part itself, we can sequence about 360 cases for the same, let's say, giving the same genetic information as what you would have for whole exome sequencing that is done routinely in diagnostics, or a lot in diagnostics. Meaning that we can sequence 360 cases in one study for the same price, more or less, than what you can do for one person in whole exome sequencing. Of course, this is a very simplified picture, but it just tells you that by putting in a lot of samples in one screening effort, we can do it very cheaply. And so that's what we did. And so essentially, we sequence not just only the exons of the gene, the protein coding elements. No, we sequence the complete gene, including the introns. And that brought us a lot of novel information. So if you look at the picture of ABCA4 at all the mutations, we can find all the different types of mutations that you would expect that we know of. But here I want to focus on the debentronic mutations that we found in about 10% of individuals. to know what these variants are doing requires functional studies, lab studies. Essentially, what they do is they create or they strengthen a sequence that will make the cell believe that there is an exon at a position that normally is an intronic sequence. So a sequence that normally does not code for protein is now recognized by the cellular splicing machinery to think that is also an exon. And of course, if that exon is built into the messenger RNA and then into the protein in the end, it means that there's probably an error in there, because these intronic sequences, they are full of nonsense information. And in most cases, there will be a stop mutation at that site. So I just give you the, let's say, the overview of about half of the ones that we know. We know now about 55 of these deep intronic variants, different ones, and about half of them are displayed on this slide, just giving you the message that they are dispersed over the whole gene with a few clusters in some introns. And so we learned a lot by just finding these variants, by testing them in vitro in assays to see the splicing defects, and we could show that they are all causal in one or the other way. So 10% of the unsolved part is now solved. So let's move on to the next part, which is the structural variants. Now, we've done these studies in two larger studies, and the one that just came out very recently was done by Zelia Corradi, and she's done some very interesting studies just to find these structural variants. So what are these? These are deletions, larger deletions in most cases, or duplications, meaning they take out a part of the gene, a couple of exons, or they may duplicate some of the exons. In both instances, they then lead to sort of null mutations, meaning there will be no protein made from these mutations. On the top, I show you two classical deletions, one with the breakpoints in introns and one with the breakpoints in exons. They can be found quite easily because we cover the whole gene. The lower part is a duplication where a part of the gene, in this case it's exons 44 to 46, which is duplicated in tandem. And we can find the breakpoint very easily using this technology. And I just want to give you one interesting, quite recent finding, where we found a quite complex situation that I never saw in my career, that is a duplication deletion. What it means is that there is a duplication of exons 31 to 40 that is not then duplicated next to this fragment. No, it's duplicated later into the gene that we call a downstream in the gene, where it's actually a deletion of another 4 exons. So there is a deletion of 4 exons, and at exactly that breakpoint of the deletion, the duplicated fragment is inserted. So for the biologists among you, you probably would suspect there must be a mechanistic link between the two events, and probably there is. But we just do not understand it at the moment. But it's interesting to see that such a complex duplication deletion can be found with this technology, and we're doing some clever lab work, of course. So if you look at all the deletions and duplications, I asked Zelia Corradi, can you give me a very nice, colorful picture to show all the deletions and all the duplications? And she did. She thought about this, and creative as she is, she then put in all the deletions and all the duplications in one picture. So what I show on the slide is the position and the number of alleles that we found for all the deletions and duplications. Above the line, which is about 1/3 of the gene, all the deletions, how big they are, how many alleles are there, with one that is present in 16 alleles, just deleting exon 6, and also some duplications that are below this line, and you can see how far they stretch over the gene. And if you do this for the whole gene, you get this colorful picture where you can find all 70 structural variants, deletions and duplications that are spread over the whole gene. I think it's an interesting picture altogether. So that adds another 2% of the unsolved, or let's say missing heritability of the gene. And let's look at the last one, which probably is the more interesting one for all of you. So this picture you've seen, I show again the genotype-phenotype correlation. At the very right of the picture, you can see the cases that have a severe and a mild incomplete penetrant variant, and they have an onset of disease beyond 40 years of age. So what's so interesting about these? The term incomplete penetrance means that that particular mutation does not always lead to disease. not even when it's in combination with a severe mutation. And so if we look at this from the phenotypic spectrum, we can see these 4 stages for Stargardt disease, from mild to the left to the very severe stage at the right. And these 4 stages are seen in the early onset and the classical Stargardt disease cases. And so they have a severe or severe mild genotype. But if we look at this severe, very mild variant at the other allele, and I give 2 examples here, they will come at this early stage and the second stage, but they never reach the very severe stages, meaning that they will never have an involvement of the choroid and the atrophy that is very broad in the retina. The phenotype will restrict itself to just within the let's say, the vascular arcade, with some phenotypes which are at the end of their life still not that severe. So we call them late onset. So we found already some years ago that we had about 5 families where we had siblings, where one sibling would be affected with this severe mutation and this very mild allele, but we also would have a sibling but would not be affected, would have the same 2 variants. And of course, this tells you that there's more than just APC4 to get this person on the right side to have Stargardt disease, and the other one does not have Stargardt disease. To make a long story short, what we found out was that the variant that is in this family has a very high allele frequency in the population, but we also know that it only explains disease in 5% of individuals that have this combination of variants. So just think about it, only 5% that have this combination will develop Stargardt disease. So now you already understand that there must be much more than just APC4 that determines that there is Stargardt disease. And to tell you the truth, we just don't know what other factors there are. We just don't know at the moment. Moreover, which I find even more interesting, is the fact that if we look at this group of late onset Stargardt disease cases, that we see more females than males to be affected. Now that's also something that is mind-boggling, because in our field, we think of these things as very simple divisions between cases and phenotypes. And if you have these mutations in this gene, you have the disease. And it's equally spread between males and females if it's autosomal recessive or autosomal dominant. But in this case, that's not the case. For the group of Stargardt cases that have a late onset, we have more females than males to be affected. And this was found for one particular variant early on, for another one a little bit later. And then we tried to see whether we could find this even more. And we did a meta-analysis and coupling lots of studies together. And by doing so, we could show clearly that indeed there is about 60% of females to have the disease compared to 40% of males. And this is compared to people that do not have any of this very mild variants. So we believe, work done by Stephanie Cornelis, that there is a clear distinction for that group of Stargardt cases. So to put it all in one picture, we think that we found many different variants, that we have to look at this as a spectrum of cases where if you have less than 40% of ABCC4 activity, you will have ABCC4 disease, probably. And if you have more than 40% in these cells, then you are healthy. But the mutations themselves only tell you part of the story. The other part are other factors, genetic or non-genetic, that we do not yet understand. So I'll skip this and say, I think by doing this, we could add another 12%. And so far, I think we can solve about 85% of all the Stargardt cases. A little bit more to be discovered, but I think this is what it is. And studies by other people have shown indeed that these findings can tell us a lot about other conditions as well. And I'm pretty sure that these concepts are sort of similar also in other IRDs. So just to thank all my colleagues in the lab over the last couple of years, especially for this talk, Zelia Corradi and Stephanie Cornelis, who did a lot of the work. And yeah, I thank you also for your attention. Thank you.
Maybe we might just get a couple of microphones, if we could get a couple of microphones up the front. Thank you very much, Bart, Elaine and Fred. if we can get some microphones up, we might just do a 5 minutes question. Are you happy to come to the podium here? Franz, I'm just going to ask, actually, any question from the floor? It would be very rude of me if I'm going to jump in there. Great. Dan. Ben, sorry. Ben. While we're getting the microphone over there, would I just ask, so really, one of the questions maybe to consider, and we'll hear Ben's question, and then, but the impact of mild phenotypes. We're looking at the genetic architecture, and we're talking in the era of gene therapy and gene augmentation about predicting any deleterious effects from gene augmentation therapy on a backdrop of milder disease. Just to hold that, that may have been the question anyway. Ben?
Yeah, I'm sort of going down that path. It seems like you have somewhat of a predictive genotype-phenotype model, and we're looking now at Stargardt disease therapies that are mutation agnostic, the vitamin A and vitamin A modifiers, and hopefully in the not too distant future, genetic therapies, ABCA4 based therapies. Do you think knowing the mutation, the genotype of the patient is going to be really critical in enrolling those trials?
Franz, do you want to take that?
Well, if it would be a genetic therapy, for sure, right? Because then you want to treat the ABCA4 disease. And although I put some doubt on, let's say, what is the role of modifiers, of course, the basis of Stargardt disease is still the ABCA4 variant. So you would like to know that these variants are in that individual before you do a genetic therapy targeting the ABCA4 gene or the environment of the ABCA4 gene. Yes.
But I guess to follow up, but knowing if it's a mild genotype versus a severe is very important as well, is what I think I hear you saying.
Yeah, well, so if there is a mild and a severe variant combined, you know that the degeneration will be very slow. So in terms of follow-up and to look at markers later, of course it will be a challenge to see the effect in a short term. That's clear, right? So that's the disadvantage of having a very mild disease, right?
Great. I think any other questions out there? I might just throw it out there, maybe in the context, Barry, you brought up the RP65, that dominant disease and those different variants. Would you make a comment on strategies in around that? And I know we don't quite have gene therapies for dominant disease, but how your thinking would be in that context?
Yeah, so David, thank you. And I think what you mean is, would you consider, or what would you do to potentially consider Luxturna therapy for dominant disease due to the Irish mutation, dominant disease as we saw with the very mild maculopathy, mild in certain cases, not mild in others, due to the Belgian mutation. Those are two conditions due to one mistake in one copy, one allele of the gene. And then obviously the other thing you could immediately ask is those recessive RP65 IRD patients who present late, they do not present as Leber congenital amaurosis. That is, they don't even present in childhood, but they only present later on. They may complain of night blindness early on, but then in their 20s sometimes get diagnosed. Would you treat them or not? So for the dominant disease, we don't know enough yet. We need models. We need to try and understand the dynamics of what the mutation does to the enzyme that is encoded for by the gene. And up to that stage, until we know that, no treatment. There have been people who have been treating disease models with of the dominant mutations. The Irish mutation, I think, has been treated once with Luxturna. I'm not sure whether this gave anything, but I would certainly not go there until I understand much better the mechanism of disease. As for the milder conditions, due to two mutations, I think that we could treat later, but I, for one, am always thinking about do no harm as a doctor, So you always have to balance out the benefit of a treatment where you see, due to Luxturna, that the increased sensitivity to light is really helping people navigate in lower light conditions. But you obviously also have those cases, a quarter of cases, showing some level of complications, which is chorioretinal atrophy. And we need to understand that much better before we do it. Elaine?
Thank you, Bart.
Yeah. No.
I was going to ask it as a question.
No, but just a comment, because this raises the question about the newborn screen, you know, predicting what is going to be the phenotype, what is actionable, when, etc. So we are really in the field of this questioning.
That's a really good point. We don't really know that the gene is necessarily going to equal that phenotype down the line, and that's the balancing act that we would ask our patients to work with us a little bit on. Elaine, just to come back to you on, please put up hands if you want to join in here. Oh, do we have a question? Yep. So just down here. So as we're getting that across, Elaine, I want a quick priority. Panel-based testing for all, or do we continue that pursuit of deep diagnostics for the few that we have under our care? You can choose one.
I think it depends very much on the country where the patient lives, what is accessible, and how the healthcare is organized. I mean, yeah, really, it depends a lot. I mean, for us, for instance, now it's genome first, but we do the in silico panel. So we start with the panel, but it's in silico. So, of course, we would not look at all the genome at once. We start sequentially.
Question here? Yes, thank you. So all these genetic tests are done on the level of DNA to find changes or alterations in the sequence. Now, what I am thinking of is, the question is, how much of the missing percentage could be explained by epigenetic landscape in the retina, which will be difficult to find out by looking at blood samples, for example, because this epigenetic landscape can be different or is different between cell types. So I would appreciate some light shed on.
I'm sure Franz wants to answer that.
You're happy that I give the answer, right? No, we just don't know how to tackle that at this moment. And it's not entirely sure whether a blood analysis would not give the best result. There are indications that there are some elements that are also conserved in blood cells. So I think it has to be tested to know whether epigenetics can be sort of tested in that manner. But yeah, it's a valid question. We just have no answer now.
There's one more. We just have one question here, and then we'll come back. Okay, the last two. We take this question here. Last question here. I'm conscious of the AMD session starting, and Julie Silvestri is staring straight at me. So I want to make sure.
Sorry, just a quick question of your trio analysis that you're doing. It was very interesting. And just how you're doing your prioritization of all the data that would be generating. Are you going purely by inheritance patterns? Are you using exomizer scores? Or how are you actually analysing that data?
All the tools are available, of course, and it's the variant prioritization that is done on inheritance, but also on the type of gene. I mean, there are a lot of different tools that are used to analyze the data, and sometimes you do not have trios. So this is also a challenge, but we have done a study on the intellectual deficiency pilot project where we show that solos, just the patient DNA, is more powerful than just the panel and the exome. So of course, not as powerful as the trio, but it can be done too. So we use a whole set of tools for that.
Thank you. Last question.
Any idea of the causes of the difference, gender difference in the late onset Stargardt?
No, I mean, one could consider that there must be hormonal factors acting here. And myself, I thought, okay, if a female goes through different phases in life, there's many changes in the hormonal, let's say, levels. And that may be triggering some of this, but that's all we can tell.
Thank you very much. Thank you very much for a fabulous session. We're going to hand over to Julie Silvestri to chair the AMD session. Great. And colleague Julie Silvestri. Thank you for those talks.
Thank you very much, David, and that was a wonderful session. So we're going to move on to age-related macular degeneration, and we have three great speakers. So I'd like to welcome Kieran Keenan, Tiernan Keenan, who is homegrown. He works at the NIH at present. He has been a Fulbright Scholar, which is very prestigious, and has worked in the Moran Eye Institute. So Tiernan, can I invite you to the stage to tell us about AMD and AI. Thank you.
Thank you. Yeah, thanks so much, Julie, and thank you for this invitation. It's a great privilege to be here and to speak. As Julie says, I'm from Ireland originally, but it's been about 7 or 8 years now. I've been working in the States at the National Eye Institute. And I should mention, I have no conflicts of interest to disclose as a federal employee. I was asked to speak a little bit about AMD in relation to artificial intelligence, and which I'll certainly do. There's just a couple of other things I wanted to see if I could mention in addition. Some very recent data, just a paper of ours that came out a couple of weeks ago where we've updated the AREDS AMD simple scale. And then also have what I think is some quite interesting, very recent data on oral supplements and the role they might have in geographic atrophy that's not yet published. But I was going to start on the AI side of things, and just to set the scene a little bit, maybe even before pandemic, but certainly post-pandemic, there are a lot of challenges to our traditional models of care in medicine and ophthalmology. So we certainly, I think, face a growing burden of eye disease. With population demographics, millions of attendances in ophthalmology departments around Europe. And in AMD specifically, we know in Europe there's as many as 1/4 of people aged 60 and over have at least some degree of AMD. So that equates to about 70 million people in Europe and growing. And so the challenge is, how do we provide, consultant-level retinal care to all of those millions, and even reach into the population through screening and pick up many of those who are currently undiagnosed. And I think in that context, AI, I don't think it's the full solution, but I think it's an important part of the solution to think about how we change our paradigms of care. Can we make our care models more convenient to patients in terms of location and timing, high street, teleophthalmology approaches? And are there tasks that AI is really good at that are kind of laborious for humans, that we don't necessarily do that consistently or accurately? Can we kind of offload or get AI to assist us with some of those to save our time for the tasks that only humans are good at, including that vital kind of human connection with the patient? And just very briefly, really, a couple of words about AI. What do we mean by it? And for the purposes of here, I think we'll just say we're talking about trying to train computers to do tasks that, typically only humans would be able to do. And there's two main things that have led to this recent boom or renaissance, really, in AI, and that's, massive increase in computing power, as we see with these kind of pictures. And secondly, the growth of big data sets, because it's typically big data sets we need to be able to train these AI models. And it's deep learning specifically, a subset of AI that's enabled this renewed interest. And again, I won't dwell on that too much, but we talk about artificial neurons. We borrow some of the vocabulary from the brain and nervous system. But these are just kind of mathematical operations going on. And when we connect these neurons together in massive networks, so-called deep learning, it turns out that computer power, arranged in that way, is really good at learning by example. So that's the difference with deep learning. And it could be, diagnosis from an image. It could be many different tasks. So in the States, I'm part of a group called the Collaborative Communities that was set up in conjunction with the FDA in this kind of new era. And our role is to try and think about how we can design and implement AI systems, and also how do we tackle the kind of regulatory hurdles, because this is very new territory for the FDA. And so we put out a white paper recently trying to think about all the different tasks and settings, actually, where AI can help us for AMD specifically. And that can be from screening to diagnosis to prediction. But in the settings, it's very variable. The kind of AI we might want in population screening in the high street or GPs, for example, would be very different to what we might be wanting in a retina specialist clinic. And essentially, I was just going to spend a few minutes covering a couple of examples to make this a bit more real and see which ones are at FDA approval stage and which ones are hopefully coming in the next few years. So in terms of screening and diagnosis, this is some work from my own group, the NIH, where essentially we're trying to see if we can train deep learning models to do the kind of tasks that typically you'd need a retina specialist to do, in clinic. And so we have the patient represented for us by a pair of fundus photographs, a pair of retinal images, and we're lucky at the NEI through the very large AREDS-1 and AREDS-2 studies to have many hundreds of thousands of these kind of images, and each one very kind of painstakingly annotated by reading center graders for AMD features. And just to cut a long story short, in the interest of time, it turns out, given enough data set, given these annotations, and given neural networks, we can train deep learning models to do these kind of tasks. And in this case, we used about 50,000 images for training. And a bit of a recurring theme is, if you train deep learning models to do, here we're doing, detailed AMD severity classification. The AI model can not only meet, but typically it can surpass by a decent way the performance, even of retinal specialist experts. So that's the kind of screening diagnosis side of things. That's using relatively simple, two-dimensional images. What about OCT, which we use increasingly? And that's obviously a more complex three-dimensional image. This is some really nice work from Piers Keane and his group in London at Moorfields, working with DeepMind. And essentially, they have a kind of two-step approach to handling OCTs in an automated way. And so in the first step, the neural network receives the OCT. It does an automated tissue segmentation process. And then that segmented tissue is fed into this second deep learning model, and that does the kind of diagnosis task. It considers a whole range of retinal diagnoses. In this particular example, it's predicted with 99% probability that this is a case of new wet macular degeneration. And it actually comes with a referral recommendation as well. It says 99% urgent, it believes. This is not FDA approved, but this is an example of the kind of paradigm where AI might be not taking over from us, but might be assisting us potentially in triage, but ideally with human oversight. So that's considering things at a single point in time. What about prediction? Can we use AI to kind of help us look into the future in the form of personalized medicine, which is something we really aim for? And this is an extension of my previous work, so we call this deep CNET prediction. And again, we've got the patient represented in front of us by a pair of color fundus photographs. But this time, we don't just want to know, how much AMD is there. We're going to ask the deep learning model to give us back specific percentages. So what's the percent of this individual developing advanced AMD? And we asked over a wide variety of time points, 1 year, 2 years, 5 years, 10 years. And again, just to cut a long story short, we had two different AI approaches. One, a more traditional one, a kind of getting deep learning models to do feature classification. A second, more kind of black box one, where we call it deep feature extraction. The models can extract whatever features they think are most predictive. And if we compare the performance of those two AI approaches, and their results are shown on the bottom here, with the two existing clinical standards of care in the hands of retinal specialists, and those two approaches are shown at the top. Again, the same recurring theme. We see that the predictions that come from the totally automated approach are not only as accurate, they're even substantially more accurate. So again, for personalized medicine, AI seems to be able to help us in AMD. Switching gears a little bit towards monitoring now, and this is more like the kind of tasks we might be useful for us in our retinal specialist clinic. And let's say we've got someone, we already know they have advanced AMD. This, I think, is a task that lends itself really well to AI. If we've got someone with neovascular AMD, it becomes all about the OCT, detecting fluid to alter our kind of treatment and injection intervals. As humans, we can't quantify retinal fluid. For AI, this is relatively straightforward. So this is one example. This is the Notal AI algorithm. It takes the OCT, it detects fluid, it can actually quantify it in nanoliter levels. It can show it to us, color code it to make sure we agree. And in fact, just very recently, in the past couple of weeks, this has been taken one step forward. This has actually now been FDA approved. in the form of the Notal Home OCT device, which is kind of amazing. You know, this is a personalized OCT device that a patient with wet AMD takes home, and they perform daily self-imaging. The OCT goes to the cloud. It's analyzed by the Notal AI algorithm, which detects and quantifies the fluid, and it plots it on the chart, you know, the patient's record that you see towards the bottom there. And again, this allows potentially a really high level of personalization of care. You know, someone's at home, they're not due to come into the clinic for a couple of months, but daily self-imaging before any symptoms are there is starting to show fluid recurrence, and then a threshold quantification is set, triggering an alert, and the patient's recalled to clinic. And the 2 pivotal trials, which should hopefully soon be published, showing the results of this kind of approach. So that is FDA approved. I think equally for advanced dry macular degeneration, with the advent of some treatments now, for GA, there's an urgent need to know kind of in real time in clinic, what is the GA area, and ideally even what is the predicted GA growth. And we've done work on that. This particular paper is from Genentech. In industry are already using these approaches in their clinical trials. So a kind of a rapid tour through a whole different range of tasks and settings where AI may be able to help us in AMD, some already in the clinic or in clinical trials, and others hopefully on the way in the coming years. And a couple of themes maybe to come out of that. As I was emphasizing, in some cases, it's about accuracy. We can get even higher accuracy than the retina specialist. I think a much more interesting second point is the ability of AI to massively, expand potential access to care, including in, difficult to reach populations. And thirdly, there's sometimes tasks that humans just can't do that AI is able to do. So 3 kind of themes there, I think. If we've got a few minutes, I just wanted to say another couple of quick words, some related to AI, some not so much. I've been doing a lot of research in the past few years on geographic atrophy in the context of AMD, and kind of consider it in 2 different stages. Firstly, getting to the point of geographic atrophy, and secondly, we have the second stage of GA expansion. And this is what it looks like over the course of 2 or 3 years, and we can see these in inherited retinal diseases as well, of course. This kind of relentless progression of GA across the macula. In this piece I wrote last year, I tried to pull together and synthesize a lot of work from our group and from other groups, trying to think about GA a bit more specifically and consider it in these 2 stages, getting to the point of GA and then secondly, GA expansion. And this is too small to be seen in detail. It's just to show the approach. I wanted to look at all the risk and protective factors for those 2 stages separately. And when you put them together like that, we might expect them to be exactly the same. And certainly there are some risk factors that are shared between the 2 stages, and smoking is a good example in our research. But actually, it's quite impressive the extent to which many of the risk and protective factors don't actually overlap between the 2 stages. And the most striking is probably on the genetic side. So I'm just redoing with others a very large GWAS of GA expansion rates. It turns out the genetics of GA expansion rates are almost totally non-overlapping with the genetics of GA risk in the first place. So it's telling us, I think, something really important about the driving mechanisms differing at least partially at each stage, and therefore, of course, we need, therapeutic approaches to be tailored to each stage. Moving on from that, I was going to say a word about the updated ARED simple scale. We've done a lot of work in the past few years looking at reticular pseudodrusen, this more newly appreciated macular feature, or retinal feature, and trying to understand this question whether or not it is a feature that increases the risk of progression to late AMD, which has been a bit controversial. Because, of course, a lot of our classifications and scales are based just on the traditional features, drusen, pigmentary abnormalities, and so on. And I don't know how well this projects, but this is a nice example of a patient from our clinic who demonstrates this. 2 distinct populations of drusen. In the temporal macula, we see the traditional soft drusen, but elsewhere in the macula, we see this striking presence of RPD or STD. Even, in that 1B scan, we see both types of drusen. And so we really didn't know for individuals like this, are they at much higher risk of GA or late AMD or not? So we set out to try and analyze that, and we wanted to look in the AREDS-1 study. That's the ideal place to look. But AREDS-1 being an older study, it was based only on color fundus photographs, and humans can't grade RPD reliably on color fundus photographs. And so this is, again, a really nice example where we can use AI to do something that humans can't do. we trained deep learning models to do kind of automated inference of RPD, even from color fundus photographs when human eyes kind of can't appreciate them. And I won't dwell on this, but the way we were able to do it was because in ARIDS 2, a different data set, we had about 8,000 FAF CFP image pairs. And paired with AI, these multimodal image pairs or triplets are really helpful. You know, in this case, we had all of the FAF images graded for RPD status, but then those labels can be applied not only to the FAF images, but also to their corresponding CFP image pairs. And then, you know, we can feed thousands of CFP images into the neural network, and it turns out deep learning models can detect RPD in this way. I'm going to skip over that. So we took that deep learning model trained on ARIDS 2, and now we can apply it to ARIDS 1. And, you know, doing the analyses, taking into account all of the different lesions, it turns out RPD is a risk factor for late AMD. It essentially doubles the risk of progression. Whatever risk you have from the other features, if additionally RPD are present, your risk of late AMD doubles. It's a little more complex than that, but I'm not going to go into it too much. The degree of excess risk differs a little bit upon where you are on the scale. But the other interesting thing to note is RPD disproportionately increases the risk of GA much more than it does the risk of neovascular AMD. And so that's a little bit different from the other features, the typical drusen, for example. So a nice example of how AI can help us add a whole new dimension to a historical data set for a study like ARIDS 1 that can really never be repeated at that scale by this kind of inference of features that even human graders can't detect. And the final implementation of that we published just a couple of weeks ago, and this is the updated ARIDS simple scale. So everyone seeing patients with AMD will be really used to this. For the past 20 years, we've had this simple scale. You grade drusen, pigment in each eye, you add them together to get a score between 0 and 4, and you read off the 5-year risk of late AMD. But being 20 years old is a couple of limitations now. Firstly, it's got a pretty old definition of late AMD. It includes only central GA, not non-central GA, so we wanted to see about making that update. And secondly, as I was saying, it doesn't incorporate RPD, so it loses accuracy that way in both directions. So to cut a long story short, here's the new scale. On the left is the one with the first update only, updating the definitions, and the one to the right is the final updated scale with both updates. And essentially the most parsimonious way to do this was to make it into two, one for those without RPD and one for those with RPD. And we see what the doubling of risk means. 4% becomes 8%, etc. One thing fell out quite fortuitously. The numbers on the left I'm showing now are from the very original scale, and they're these really easy to remember numbers that kind of double as you go up. And just luckily, it turns out the new scale for people without RPD has pretty much identical numbers. So that makes it much simpler. We can carry on using the old numbers. We just have to remember if RPD are present, the risk is pretty much doubled. The final thing, if we have a minute, I was going to say a word about oral supplements. Kind of ARIDS 2 type formulation supplements in geographic atrophy. I was going to talk about diet tomorrow in another session then, so I'm going to skip over this, but we've done a lot of work showing that a healthy diet, for example, more of like a Mediterranean pattern, is very strongly associated with slower progression of AMD. Even at the stage of geographic atrophy, it's strongly associated with slower GA expansion, which I think surprised us and many people. But what about supplements? ARIDS 2 supplements are indicated from the point of intermediate AMD onwards. We know they decrease a lot the risk of neovascular AMD. Sadly, they don't really decrease at all the risk of geographic atrophy. And equally, once GA is present, we'd kind of assume they don't really do much to slow GA growth either. But we hadn't really looked at it in the past, so I wanted to look at that in ARIDS 1 and also in ARIDS 2. And I wanted to look at it using two different metrics. Most clinical trials use a kind of area metric, and that captures GA progression well for cases like this that are center involving. But what about cases like this that are not yet center involving? Area isn't the whole story, I don't think. We're also interested in how quickly or how slowly GA moves to the center point, because that's crucial for visual prognosis. And we have this phenomenon of foveal sparing, which is stronger in some, weaker in others, and I think we see also in IRDs sometimes. And so we had these proximity variables. I developed a kind of proximity-based growth rate. A growth rate, how quickly or slowly does the GA progress to the center point over time? And that really varies between individuals. And I related those two metrics back to the underlying study randomizations. And so in AREDS 1, that was oral antioxidant daily supplements or oral zinc supplements. And to cut a long story short, interestingly, when you look according to area, you don't see any treatment effect. But for eyes with non-center involving GA, when we look using this new metric, we do see a big treatment effect from the antioxidant component. And so, individuals randomized to antioxidants, their non-central GA grew about 35% more slowly to the central macula. So pretty large treatment effect, particularly for such a safe supplement. When we do the same in AREDS 2, now if we just concentrate on the primary randomization, we've got people randomized to lutein and zeaxanthin, and people randomized to these omega-3 fatty acids. And it's kind of the same story. For area, we see very little of interest going on. But for non-central GA, here we see lutein zeaxanthin is having a really big effect. So people randomized to lutein zeaxanthin, their GA grew about 35% more slowly to the center point. And that was on top of the previous treatment effect I mentioned. So these are very new findings, probably will be published in the next few weeks or so. But cumulatively, it seems like 50 or 60% slower growth of GA towards the macular center from the AREDS 2 formulation, particularly the antioxidant and the lutein zeaxanthin components. And it may seem really surprising that we see that for the center, but not in the area metric. But I think for lutein zeaxanthin, it makes a lot of sense. They're only found in the very central macula, so I think they're contributing to this known phenomenon of foveal sparing. And it turns out we can actually augment it with oral supplementation. But the area metric is dominated by the peripheral and mid-peripheral macula, and they're not there, so they're not going to slow growth there. I think that's the most likely explanation. So potentially an extended role for supplements, hoping to kind of to replicate this prospectively in a new trial in the coming years. But this is very recent data. So quite a long whistle tour through a lot of AI and some other recent data. I hope there's something there of interest for most people, and really grateful for your attention. Thank you.
Thank you very much, Tiernan. That was great. Any questions? Don't go away. So while we're getting the microphones, maybe, the CAM classification that we're all hopefully going to be using for the GA trials, can it help us with that? Because it is very complicated for the day-to-day ophthalmologist in the clinic.
Is that on the artificial intelligence side? Can that help us with the CAM classification? Yeah, I mean, and we and other groups have developed lots of AI models that can, working from different imaging modalities, whether it's FAF or OCT, they can diagnose the different constituent features of, either diagnose C-RORA overall, or each of its 4 constituent features, and then you can overlay them and do all kinds of analyses. So again, these are quite often developed in the academic setting, and then there is this big kind of, hurdle or gap towards, implementation in the clinic, requiring regulatory approval and so on. And so that's the collaborative communities. That's part of our job to try and see if we can, get things being not just a publication or released code. Can we actually get adoption of these in the clinic? Yeah.
Thank you.
Thanks.
You have a question?
Hello, Professor. Hi, Professor Keenan. Thank you for the talk. It's brilliant. It's nice to see emphasis on big data and AI. I was just wondering, with regards to the first study you've had with the 50,000 eyes and images, we probably all know the importance of having lots and lots of samples to develop a strong model and a strong algorithm. I was just wondering, because there's been so much highlighting on rare diseases and rare retinal diseases, what your perspective is on the diseases that perhaps we don't have many fundus images, and maybe you've whether explored generative AI or generative adversarial networks for creating synthetic images, for example.
No, great questions. And I personally have done much less AI work in the field specifically of inherited retinal diseases. But I mean, I know I2 Gene, Nicholas Ponticus and others, and Moorfields and probably many others, trying to do that task specifically for inherited retinal diseases. I mean, I think broadly, it appears to be successful so far in that respect. I'm sure there's a lot of complexity to it, but I guess we're a bit spoilt on the AMD side in having, through AREDS 1 and AREDS 2, these very large data sets. There are drawbacks to them. I mean, I would say firstly, external validation on a totally different data set is one crucial step. And secondly, there's a question of, a characteristic like race and ethnicity and so on. For example, ARIDS 1 and 2 were 95-ish% white Caucasian. And so then, it's very important to be thinking about external validation to other, perhaps underrepresented groups. So, yeah, but we certainly do experiments with kind of, low shot or few shot or even single shot learning. Can we train models efficiently with only very small numbers of images?
Thank you. I think that will, unless any other questions, I don't see. Thank you very much. Now, it gives me great pleasure to introduce Dr. David Birch, who's the Scientific Director of the Rose Silverstein Retina Degenerations Laboratory, and he also represents Retina Foundation of the Southwest in Texas. And his focus is on genetic eye disease, macular degeneration, electrophysiology, and visual field testing. So, David, I'd like to invite you to the stage to tell us about outcome measures in retinal disease clinical trials. Thank you.
Thank you so much, and thank you for the invitation to present here. I'm not going to talk about anything that's related specifically to macular degeneration. I'm going to talk about night blindness sort of in general, and what we've learned about the measurement of night blindness and why it's important, and how it might become a great outcome measure in clinical trials for macular degeneration or inherited eye diseases. So, the data I'm going to present comes from the RUSH-2A study. This is a large Foundation Fighting Blindness sponsored study with a consortium of about 20 international centers. And we enrolled 107 patients, tested them annually. And we used it as kind of a test bed for different types of outcome measures. We were looking at what are the most sensitive measures, what changes from year to year, and how much, and how can we use this kind of information. So these are my disclosures. And so all this data is, is going to come from the RUSH-2A trial. And so the first thing I'll talk about is the full field stimulus threshold. And you've heard talk about this. It's come up a few times in different contexts. And it's important to recognize that the FST was developed 15 years ago to give us some kind of measure in labors, especially kids. So in patients who had very low vision, who couldn't really fixate, who couldn't push buttons, you couldn't do visual fields, you couldn't do standard testing. The full field stimulus threshold was kind of a quick way to get an assessment of their sensitivity to light. And it never went beyond that for a while. And it was kind of, we sort of knew about it, we used it in the kids, and we didn't think about it as an outcome measure for adult patients or for other kinds of trials. Turns out it's a very interesting measurement, very sensitive measurement. What it measures really is what's the minimum amount of light you can see. That's simple. You look into a dome, lights are presented, and it kind of goes through a complicated algorithm to determine the minimum number of quanta that you can pick up to form an impression. And what's nice about it is that it has a huge range. The machines that test this can go from just a few quanta right down at the threshold of human vision from over about an 8 log unit change, so a massive change. And so you can follow somebody from normal all the way to no light perception. And you have a continuous scale over that entire range. And it's also interesting because it has been shown to predict the performance of patients under low luminance conditions. So you get the worse your FST, the more likely you are to fail at low luminances on a mobility course, for example. So it's a measurement of real life function. So I'm going to talk about that, and then I'm going to talk about dark adapted visual fields, which is kind of an extension of that, looking at the sensitivity across the whole retina. And these were all part of the RUSH-2A trial. So the FST, it's done with white, blue, and red stimuli. And basically the way the test works is that it gives just a broad range of values, trying to figure out roughly where the patient is, what kind of sensitivity the patient has. Once it finds that, it then fine-tunes it. It presents dozens of stimuli around the threshold. And then it fits a nice function to the data and gives you a value in log candelas per meter squared of what it takes for a patient to see. And this kind of shows some representative examples of FSTs from patients in the RUSH-2A study. The way the stimuli are set up is the red and the blue stimuli are equal for the cones. So if you test each of these functions shows the percent correct on the vertical against the intensity. So the further it is to the right, the more light it takes for the patient to see. And if a patient has no rods, if their rods are gone, it's set up so the sensitivity to the blue and the red are the same, are equal for the cones. So at the top, you have a patient who's only got cones. It's cone-mediated. The functions overlap. The more rods you have, the more the blue is more sensitive than the red. So in the extreme case, in the normal, you've got about a 25 decibel or 2.5 log unit difference between the sensitivity to the blue and the red. As the rods degenerate, the blue becomes less sensitive, and the red, which is picked up mostly by the cones, stays about the same. So the difference becomes less and less. So if we look at the population that we studied, we studied most of the patients in RUSH-2A. Not everybody had the equipment. But this kind of shows that you can sort of tell whether it's rod-mediated or cone-mediated by the actual sensitivity. People that are below about -4 have rods. If you don't have rods, you're not going to see that. So, and then on the horizontal, you have the difference between the blue and the red. So if the difference is more than 2 log units, 25 log decibels or so, then you're going to be rod-mediated. So this lower left-hand quadrant is all the rod-mediated patients. And then there's a few that are in between that are kind of mixed. And then there's a quadrant up to the right where sensitivity to red and blue is about the same. Their thresholds are above -4, so it takes a lot of light for them to see it. These are patients that are basically functioning with cones. So we looked at baseline, at what the FST values reflected, what they related to. And interestingly, we had patients, they all had USH2A mutations, but within that population of patients, some had only recessive RP, others had the hearing loss as well. So there's 2 kind of subgroups, hearing loss and no hearing loss. And the patients with hearing loss were statistically worse at baseline than the patients without hearing loss. And there was also a very strong relationship between the duration of disease as reported by the patient and the FST value. So the longer they'd had the disease, the worse the FST, which is not surprising, but it would be a problem if it wasn't that way. So these are called spaghetti plots. This shows for each patient what happened over 4 years. And it's presenting the values in log candelas per meter squared on the left there, and the duration of disease on the right. And for each patient, you can see the changes over the 4 years. These are four-year measurements. And in general, the vast majority of patients show an obvious increase in their thresholds over the 4 years. And this is some of the strongest data, actually, that came out of the RUSH-2A study in terms of significance. So as patients have longer disease, they have the thresholds go up, and each year the thresholds are going up. And if we look at it, actually, for the white stimulus and the blue stimulus, the slope is about is a change of about 0.1 log candela meter squared per year, about 1 dB per year. Well, that's about a 20% change per year for the blue and the white. So that the rods are going at that rate. The red picks up primarily the cones, and the cones are changing at about half that rate, at about 0.05 per year. So it all sort of internally makes sense. And it's also interesting, we look over the 4 years, we have patients that were rod-mediated at baseline and then switched to being either mixed or cone-mediated by year 4. But the opposite never happened. We never had anybody that was cone-mediated at baseline who became rod-mediated later. So once you've lost your rods, it's determined by cones. We also were able to look at the repeat variability. We did each of these tests in triplicate each visit. So we know very well what the repeat variability, correlation of coefficient of repeatability is for each of these years. And this shows it for the 4 different years. And it averages about 0.39, I think it is. Yeah, 0.39 over the entire year. So we know that any change beyond 0.39 is unlikely to be due to variability. It's going to be real. So that's the real change. And we looked at how many patients showed a change beyond that coefficient of variability over 2 years and over 4 years. And over 2 years, it was 30%. After 4 years, it was 47%. So we're getting a significant change in about half the patients over a 4-year period. And, it doesn't sound like much, but it's actually a higher percentage of patients changing on this test than it was on virtually any other. Certainly, visual acuity, they hardly change at all. Cone visual fields, they change at about half the rate to this. So just to summarize the FST, there's a significant decrease over 4 years. There was no real floor effect. The other nice thing is about it, you can test everybody, no matter how good they are, how bad they are. And it does relate to disease duration and to also changes in the ellipsoid zone. It's also highly correlated with the ellipsoid zone. It's a high signal to noise. It means you get a fair amount of change per year versus the repeat variability. And it has good reproducibility. So there are some disadvantages. What are the limitations? It can't detect localized improvements, okay? It's detecting the most sensitive part of the retina. So if you have gene therapy and you're given get some improvement in one part, unless it becomes the most sensitive, it may not be reflected in the FST. And it also, we don't really have a strong feeling about what the change in FST means among patients that have good vision. It's pretty clear that it's related to ability to function under low levels, but in young patients, we're just not that clear. So for those reasons, we sort of expanded after the first year. We added a second test, which is dark adapted visual fields. And so now what we're doing is we're looking at the sensitivity all across the retina under dark adapted conditions. And we did it because we really are not measuring rod function typically in these diseases. We measure cones and acuity and visual fields and OCT, all these things. None of them are specifically rod mediated, but we know that the earliest changes are to the rods. Most of the mutations, a good many of them, are in the rod system. So it kind of makes sense that early on in disease, or as we're looking at treatments, we'd want to make sure that our treatments were benefiting the rods. So these measures are kind of designed to pick up the slack and to use visual fields to look at rod function. And this is just, again, the rationale. If you look under dark adapting conditions, the rods are much more sensitive to blue than they are to red. Under light adapted conditions, it's roughly the same. So down below, if you look at visual thresholds across the retina, for example, to blue light, totally dark adapted in a normal, then the blue is much more sensitive than the red. Whereas if you turn the lights on, do it light adapted, so the cones are mediating them, they're roughly the same. So you can sort of tell from the difference between red and blue, whether it's rods or cones. And we did that in the Rush 2A trial. We it's a Medmont device. It's made in Australia. Not too many of them around, but it's a useful device. It can be done with other machines too. It's nothing specific to the Medmont. But we asked each investigator to try to determine, based on the red and blue, whether or not the patient had rods at baseline. And if they had rods, then we would continue to follow them over time. If they didn't, then we just kind of didn't continue it with that patient. And then we took the values. So here we have cyan. These are the values to cyan, and then the values to red. We take the difference, and for every place that it's rod-mediated, we feed that into software that plots the hill of vision. So it gives you a 3D plot across the whole retina of where the rods are located. And kind of in a normal, it has sort of a flat shape falling off on the edges. It doesn't look like the cone visual field at all. It's very different. And we can do this kind of hill of vision calculation. It splines all the points, and then it gives you the areas in all different parts of the retina. So we can look at the total area of vision, of rod vision. We can look at the area within the central macula, 30 degrees. And we can look at the mean sensitivity. So we get a lot of measurements out of this, very useful. And then we just use statistical methods to estimate the annual rates of change and looked at correlations with other visual functions. So out of the 49 that we tried it with, 78% had some evidence of rod function. So that's kind of, we were a little surprised by that because a lot of patients are night blind fairly early, but it turns out that if you look across the field, there are patches where there are rods even in more advanced patients. So more than half, about 3/4 of the patients had some evidence of rod function, and those are the ones that we followed. Not surprisingly, the visual field sensitivity was highly correlated with the FST, which kind of makes sense. They're both testing the same thing in a sense, in the rods. And then the longer the disease duration, the fewer points we had that were rod-mediated, and the total volume of the field was reduced as disease progressed. So this just kind of gives an example from a patient where it's really obvious what's happening over 4 years. They're not all like this, and this is kind of misleading in a sense, but this shows you the 3D volume of the hill of vision at baseline, or year 1, then year 2, year 3, 4, and obviously a big change in this patient, and that kind of went along with the reports of their rapid progression. But they're not all like this. This is another example where there's a small change, measurable, but it's only a relatively less change over the 4 years. But we looked at it over all the patients, and the spaghetti plots here, these are going the other way. So the duration of disease, as it increases, the mean sensitivity decreases. So these are plots for each patient, how much they're changing over the 4 years. And this is the same thing for the total. The other one was the mean sensitivity. This is the total. And basically what we saw over 4 years is that, again, the majority of patients really didn't show a change, but there were many more patients showing a decrease in sensitivity than there were an increase. So overall, there was a significant decrease in rod function. And this just shows it in different ways, showing the mean and 95% confidence intervals for V total in this case. So what we've learned from the visual fields is that the recessive patients, again, had a greater change over time compared to the USH2A participants. And there is an association with the measures over time, with the enrollment age. Younger patients have greater change over time. That's another thing that we kind of learned throughout the RUSH2A trial with all the measurements, get much more change early on in the disease. The older the patient gets, the more it kind of plateaus out, or the less change per year and on a percentage basis. So the average yearly change was 1.57 dB. So that kind of, the rod field loss is greater than the FST, sort of along the same lines, and it's certainly greater than in cone-mediated visual fields. So rod fields change faster than cone fields. And the conclusion is that both these measures give you a good indication of rod-mediated function. They change significantly over time. And we'd think they might be useful, especially for the early detection of a treatment benefit. I think as gene therapy, as we become more confident in the procedures and we move the treatment into younger patients and things like X-linked RP and other conditions, I think we're going to use these kind of measures to get a feeling for whether the treatment's really helping over a year or two. You know, if we can get a quick readout on benefit. And I think as far as using this as an outcome measure for the regulatory authorities, they want to know more about what FST and what fields mean to the patient. And so our plan with the RUSH-2A trial is to bring the patients back at 7 years and at 9 years and see if we can sort of predict what their vision is going to be based on these results from earlier. And you know, I think if we can make a good case that a change in FST or something is highly predictive of future visual function, I think we'll make a stronger case for the use of these kind of measures as outcome measures. So I will leave it at that. Thank you.
Thank you very much, David, for a very clear overview of that. Do we have any questions? Just while we're getting to the mic, repeatability is this a difficulty with low vision patients, or do they do okay?
They do okay. They're not, they're a little bit higher under the age of 10 than they are over the age of 10, but not too bad. I mean, Sam Jacobson has done quite a bit of art sedation, have looked at a lot of kids and find that the repeatability is, if it's about 4 decibels in adults. It's maybe 5 in the kids, so not greatly different.
Thank you. Any questions out there? So are the FDA considering it, or, well?
Yeah, they keep getting presented with it. So especially FST, and you know, there's been a change at FDA, and so we're not exactly sure what the new authority or the new people are going to be saying. EMA has been quite receptive to it. I don't think any treatment has been approved based on it, but they have said repeatedly that they, you know, accept that it's a good measure of visual function.
Thank you. That sounds good. Thank you very much. Thank you. I'd like now to welcome Dr. Geoffrey O'Callaghan, who's also homegrown, works in Trinity College Dublin at the Matt Campbell Lab, and he's published on glaucoma therapies and retinal degenerations, and he's going to talk to us today about the inner retina in age-related macular degeneration. Geoffrey.
Hello, everyone. My name is Jeff O'Callaghan. I'm a postdoc in Professor Campbell's lab here in Dublin. And you normally find me talking about gene therapy strategies for glaucoma, but I'd like to thank the organizers today to let me speak on behalf of the Campbell Lab on the topic of the inner retina in age-related macular degeneration, or AMD. So just briefly to recap what we may have heard earlier today, AMD is the leading cause of central retinal blindness. 1 in 4 Europeans over the age of 60 have AMD, with a total of about 196 million people affected by the disease worldwide. Now, the early stage of AMD is characterized by the accumulation of these yellow drusen deposits in the central macular region. And patients affected with early stage AMD don't necessarily exhibit any significant visual impairment. However, they do need to be monitored closely for progression into the late stage of AMD, which we call geographic atrophy, and is characterized by these atrophic lesions on the central macular area. And patients with geographic atrophy do exhibit significant clinical visual impairment that's irreversible, specifically in the central region of their vision. And what happens is that the RPE, the retinal pigment epithelial layer, degenerates over time, and this leads to a loss of rod and cone cells. So AMD is typically characterized as a disease of the outer retina. But what about the inner retina itself? Well, the retina is highly vascularized, and because it's such a high energy demanding organ, it requires a lot of glucose. So the dense network of blood vessels in the retina facilitate this glucose transport to the tissue. And these vessels themselves, they look a bit like this. They're very structurally similar to the blood-brain barrier. And we know from current literature that the blood-brain barrier, any dysregulation to the barrier can lead to pathology and disease. So we can therefore infer how similar they are. We can infer how important that the blood-retina barrier is in retinal homeostasis. So our lab focused a lot on tight junctions. And the tight junctions are the glue that hold the endothelial cells of the vessel walls together. And in the endothelial cells of the blood and the brain and retinal barriers, these tight junctions are very tight. They give the cells very low passive permeability and a high transendothelial electrical resistance. They act as a gate to either block or selectively allow molecules to pass through the barrier. The tight junctions themselves are composed of numerous transmembrane and scaffold proteins. They interact with each other and they link tightly to the cytoskeleton. And in particular in our lab, we focused on a tight junction protein called claudin-5. And claudin-5 is the most highly enriched protein in the endothelial cells of the blood-retina barrier. And we know that claudin-5 is also tightly regulated by the circadian clock. So in the brain tissue, claudin-5 expression levels are very high in the morning, and they get much lower in the evening. So it's high in the AM, low in the PM. And we've seen a similar pattern in the cells of the blood-retina barrier. So we can look at the expression of claudin-5 in mRNA and in protein, and we can see that expression is significantly elevated in the AM compared to the PM. So we know tight junctions cycle in a circadian manner. But what about vessel permeability itself? Does that also cycle? And how do we visualize permeability of the blood vessels? Well, we use a technique called fluorescein angiography, or FFA. And that's an image of the retina, like this one, that highlights the blood vessels. And this is done by injecting intravenously a fluorescein dye. We can subsequently take images by these special cameras. And angiograms are performed safely as outpatient procedures for over 50 to 60 years now. And they're done to confirm diagnosis, to monitor disease progression, assess treatment efficacy, and in our case, to study the retinal vasculature. Unfortunately, there are quite a few shortcomings with FFAs, mostly that they're strictly qualitative. They require an experienced professional to interpret these images. Single snapshots usually provide a limited idea of ocular health, which is why we need to often take images over the course of a 10-minute imaging session. And small changes may not be immediately visible to the naked eye. So doctors today are still interpreting these images by eye, so it's very easy to miss small changes. And it's also, as we can attest to in the lab, it's very time-consuming to process and analyze multiple images by hand. So to address all of these shortcomings, we developed a software called FOVAS, which stands for Fluorescent Ocular Vasculature Analysis Suite. And FOVAS essentially gives us a fast and automated analysis and quantification of these FFA images. So we can input into FOVAS any number of patient images. We can use it with any images from any acquisition hardware. We can compare the data to our own internal data sets, and we can correlate the data to any demographic or qualitative variables that we may have. FOVAS will then output an automated statistical analysis. It'll give us a detailed assessment of the retinal vasculature and provide an unbiased quantification using our own image processing methods. So FOVAS is compatible, as I mentioned, with any commercially available system. It's compatible with any species, whether it be species with a macula or species that are amacular. And it's also compatible with any different image file type that you might have. So I'll not go too in-depth into the inner workings of FOVIS, but in brief, the workflow is to identify the type of images being presented to FOVIS. So we're extracting things like time points and metadata. We can also then do any image pre-processing that's required, such as cropping of the image, illumination correction, and presenting quality control images to the user. We can then perform what we call an automatic registration. And registration is very important to align all of the images over the course of a 10-minute FFA session. As you can imagine, at over 10 minutes, a patient's head might move slightly, which results in an image being slightly off-center. So all of these images need to be aligned properly to be able to do downstream analysis. So that's registration. Once we have all of these images aligned properly, we can then extract and identify different regions of interest that allow us to look more closely at different regions of the retina. So in the case of a mouse that doesn't have a macula, we can use macro vessel masking, which is basically to block out all of the main vessels and only look at the microvasculature. So you can see an image of that here on the right. And in the case of a primate, we can use the ETDRS grid, which is a grid that looks like this, that's centered around the macula, and it gives us these 6 different regions of interest to look at. So we can look at the fovea, the parafovea, the perifovea, the fovea and the parafovea together, the extrafovea, or all of these regions averaged together. So this is how we determine our regions of interest. And once we have those, we can now go ahead with extracting our fluorescein intensity data and doing our downstream analysis. So just to talk a little bit about some of the data that we have on the circadian permeability of blood vessels in the lab. The first study we have is to look at young wild-type mice. And you can see that we've got images of the FFA. So in the top row, we've got images taken in the morning, and in the bottom row, images taken in the evening. And in the columns, we have a representative image from 2 minutes, 6 minutes, and 10 minutes. And you can almost see a difference yourself by eye in the PM versus the AM. But when we plug these images into FOVIS, FOVIS tells us that there's a significant increase in the cumulative signal at PM, and there's also a significant increase in signal at PM when looking at a specific phase range of time points. In this case, it's the 2 to 6 minute frames. We can also plot this out in terms of intensity over time, and we can see that in the morning, in blue here, the AM signal increases as you inject the dye and then peters out quite quickly. But in terms of PM, this signal persists a bit longer and takes longer to peter out. So you can really start to see this difference in the AM and PM the further out you take the fluorescein images. So there does indeed seem to be a circadian-related effect going on. In contrast, when you look at aged mice, again, we've got the AM images on the top, the PM on the bottom. They do look quite similar, and FOVIS quantifies this and shows us that there is no significant difference between the AM and PM. You can see the patterns between AM and PM do tend to look quite similar. On average, there's a small increase in the PM signal, but in general, this is non-significant in aged mice. So we wanted to see next whether this circadian effect translates into primates. So we have a study involving non-human primates where we're again comparing the FFA images from the AM and the PM. And this time you can see a pretty strong signal as fluorescein is injected, and then that deteriorates over time. However, the PM signal does persist and last a bit longer. When we quantify this in FOVIS, FOVIS tells us that at each of our 6 regions of interest in the ETDRS grid, the PM signal is much greater than that in the AM. So any of these median lines above the 0 line here mean that the PM signal is brighter. We can also visualize this using pseudo-color maps, which show us where exactly in the retina the signal is brighter or lower. And we can also use a method called signal decay, where we can see how the signal changes from its maximum, and in this case, compared to its 6-minute time point. And in this case, the PM signal doesn't decay as much as the AM signal, which means that fluorescein is persisting in the retina at this time point, so it's not being cleared as quickly. So again, we're seeing a significant effect of the circadian difference in the AM versus PM. So next, we wanted to see whether this would translate into into humans and, of course, AMD patients as well. So we first started with a young cohort of individuals from the ages of about 18 to 30 years of age. And again, we're looking at AM versus PM. And in the images, you can't really see by eye any significant change here. But when we put these into FOVIS, we can see that there's about a 25% increase in all of our regions of interest in the PM. So it's significantly greater in the PM, so that's our circadian differential. But similar to the mice, when we look at older individuals, these are 60 years and older, we can see that this circadian differential is quite reduced. We're now looking at about a 10 to 15% increase in the PM, and it's not significant except for the fovea that still remains significant. But when we look at AMD patients, this time what we're seeing is that there's no significant difference in any of our regions of interest. And in this case, at the 2 to 6 minute time point, we're seeing that there's only about a 5% increase in signal. When we look further out to about 10 minutes, the circadian differential is completely lost. So all of our bars are sitting right in the middle at 0. So to summarize this study, the healthy young controls, we were able to see a circadian differential between AM and PM. That effect was reduced in the age-matched controls and completely ablated in AMD patients. And you can see from these images that there does seem to be increased leakage as you progress with age in AMD compared to our young individuals. So with all of these different groups that we have, we can now import these internally into FOVIS so that any new patients or new disease or treatment groups that come along in the clinic, we can compare them internally to what we already have, and we can assess whether these new patients or new groups look more similarly to a healthy group or to a diseased group. And FOVIS will quantify all of these comparisons for us. So to summarize everything, the tight junctions we know cycle in a circadian manner. We know that vessel permeability also cycles in a circadian manner, and we've shown this in young mice, we've shown this in non-human primates, and we've shown it in young humans. And our software FOVIS aids in assessing this permeability. And vessel permeability, its cycling is impacted by age, and it appears to be completely ablated in AMD. So these give us more insights into the circadian nature of vessel permeability in AMD. And it also highlights that tight junction proteins such as Claudin-5 could be potential therapeutic targets in the future. So with that, I'd like to thank everyone in my lab, in Matt Campbell's lab, and especially thank Dr. Natalie Hudson for pioneering and leading this project from its inception. I'd also like to thank our collaborators in Progressive Vision and Versio, with a special thanks to Mark Cahill for helping to guide FOVIS's development towards clinical utility. And of course, if you have any FFA datasets of your own that you would like to be quantified or you would like to find out more about how FOVIS works, please get in touch with us. So thank you very much.
Thank you very much. Very nice work. Any comments or questions? I don't know anything about circadian rhythm. So what controls it, and why is it sort of reduced in normal, older mice and people?
Yeah, so it seems that so light is what the major factor is in this. So we can see that in mice and in primates, light is triggering the brain to produce Claudin-5 or these tight junction proteins in the morning, and it's reduced in the evening. Yeah, and it seems really important, mainly for, it can be for clearance of junk throughout the day. That's why the vessels are becoming leakier. And it's also maybe important for providing nutrients to the retina, which is why the vessels open and then close again. And so it seems to be a very important thing for homeostasis in the retina.
Thank you. So the potential treatments would try to restore that then?
Exactly, yeah. So with disease, we can see that these barriers are always open and they're not closing properly in the morning. So they're brighter, they're more leaky in the morning, and they're more leaky in the evening, so they're always open. So treatments would hopefully come in to help close these barriers when they need to be closed.
That's great. Thank you. Any questions? No. Yeah, here.
Just going to ask whether you think there's any relationship to cyclic shedding and photoreceptor renewal. You know, in the morning hours, you need a lot more protein
Right.
to get to the outer segments.
Yeah, very true. Yeah, I'm not sure. It's not something I've looked at directly, so I can't comment fully, but it does seem to match that sort of pattern. Yeah, exactly. Yeah, thank you.
Okay, thank you very much. And no one else? No. So we're going to have a small alteration to the programme. If I could ask, we're going to have our comfort break now, and if you collect your teas and coffees and bring them back in, and then we'll start back with the short oral presentations. Thank you. Could we have the presenters for the short presentation up to the front, please? Maybe they're already here. Are you on? Okay, and Avril ready, coming up, yes. Right, thank you very much. I think we'll make a start. Okay, thank you. All right, so can we take our seats, please, and we're going to make a start. So this is the session for the selected short oral presentations, and we have 3 presentations. Teresa Rogler, Avril Reddy, and Tom Daney, who are all sitting up here on the stage. So about 5 minutes each, and I don't have control of the bell, so I'll come and stand beside you if it's time. Okay. So I'd like to invite Teresa up first to tell us about human retinal organoids as a test bed for assessment of AAV transduction efficiency and functionality. Thank you.
Thank you for the nice introduction. Hello, everyone. My name is Teresa Rogler, and I'm a PhD student at the Savannah Lab in Munich. We have heard a lot of lovely presentations today about different treatment ideas and disease modeling in different situations. And for our lab, what interests us at the moment the most is like Leber's hereditary optic neuropathy. And on the other side, the RPGR mutations, the different variants of them leading to different phenotypes, such as retinitis pigmentosa or cone-rod dystrophy. And for that, we need a suitable model organism. Therefore, we have selected organoids as our model organism. They really nicely portray the human retina, including the ratio between cones and rods, and also you can keep a lot of them to have a nice sample size. We can do all kinds of different measurements with our organoids. We do neurophysiology assessments where we can track the signals that are transduced throughout the whole retina. Using calcium imaging, and we can use cells derived from patients that can be reprogrammed into induced pluripotent stem cells and then grown into our organoids. Now that we have the organoids, what can we exactly do with them? A nice important question is how can we determine the efficiency of different AAVs for the gene therapies? And therefore, we have developed an automated mechanism which allows us to see the 3D images of our organoids, the retina, the layers. It will segment all the nuclei and the transduction of the organoids in fluorescent, and then it will calculate the overlap between the fluorescence and the nuclei without the need of a nuclear translocation of the fluorescent protein. And all of this can then be shown in 3D. This can be done in different organoids. They can be living, so we can track it over the time. We have different time points. We, for example, here did 7, 13, 30, and 40 days after transduction, and we can see how fast the onset of the transduction is and also how efficient they are. And thereby we could see that, for example, this one serotype to an N is faster in the onset of the retinal transduction than the others, and also reaches deeper into the tissue. Furthermore, I brought my colleague with me, Vlasta Hadalin, and she is working with these AAVs, where we can transduce the organoids with shRNA, which can then target different isoforms of the RPGR gene, which has been suspected to can cause different phenotypes, and therefore we are hoping to see like if actually different concentrations of the isoforms of the RPGR can actually lead to different phenotypes in our organoids. So if you have questions or felt inspired, you can chat us up. We're very happy to talk about our project, and thank you for your attention.
Thank you very much. It is very inspiring. Any questions, or we could take one question if... No, I think we're all happy. They'll speak to you later. Right, I'd like to invite Avril up now to tell us about disrupted CSF1R signaling, a potential mechanism for articular pseudodrusen formation and increased inner retinal vascular permeability. Thank you.
So hi everyone. First of all, I'd like to thank the organisers for inviting me to give a quick overview of my PhD research at Matt Campbell's lab in Trinity College, Dublin. So as we said, the title of my talk today is Disrupted CSF1R Signalling, a potential mechanism for reticular pseudodrusen formation and increased retinal vascular permeability. So CSF1R, or colony-stimulating factor 1 receptor, is a receptor tyrosine kinase found on the surface of mononuclear phagocytes, such as microglia and macrophages. Signaling through this receptor is crucial for the proliferation and survival of this cell type. So our lab also studies neurological diseases, such as adult-onset leukoencephalopathy with axonal spheroids and pigmented glia, or thankfully ALSP for short. This disease is characterized by variants in the CSF1R gene, and it manifests similarly enough to severe early-onset dementia. I can, of course, yeah. Sure, sorry. So, a previous member of our lab, Conor Delaney, did his PhD research on the neurological manifestations of this disease. So here he found that in post-mortem brain samples, there was an accumulation of amyloid beta around the blood vessels in their brain. There were signs of neuroinflammation, shown by CD163 staining here, and there were signs of astrogliosis, as shown by the GFAP staining here. When he isolated macrophages from these individuals, he found that in individuals carrying two different types of variants in the CSF1R gene, they had decreased phagocytic ability as compared to macrophages isolated from healthy human donors. So as these neurological findings were stark, and that the retina is an extension of the brain, as Sarah mentioned earlier, we invited some of these individuals in to undergo retinal imaging with our clinical collaborators at Progressive Vision. So this individual here, a 51-year-old male who was diagnosed with ALSP, when he went for the retinal imaging, we found that there was reticular pseudodrusen deposits in both eyes. So firstly on the bottom, I don't know if the laser pointer works, we can see, in this fundus fluorescein angiography, there's a pooling of fluorescein in what corresponds to stage 2 deposits. In his right eye on top, there seems to be an absence of fluorescein in around these more advanced stage 3 deposits. This individual's younger brother, who carries the same genetic variant but is asymptomatic for disease, has relatively normal retinal imaging results, but there seems to be some signs, as indicated by the arrow over here, of some early potential stage 1 deposits forming. So to understand the mechanism behind the deposition of these deposits, we generated the CSF1R heterozygous animal model using the Crelox-P system and the mononuclear phagocyte-specific promoter CX3CR1. However, when we went to characterize some of the results from these mice, we found that the Cre-positive heterozygous mice had no difference compared to the Cre-negative littermate controls. So here, the fundus fluorescein angiography results, they look relatively the same. There's no change in OCT, and there's no change in retinal histology. So when looking at families that carry these CSF1R variants, we realized that the age of disease onset is quite variable. So obviously, genetic variation does explain a lot of disease, but it doesn't always fully explain disease progression. So we decided to introduce an environmental variable. So reticular pseudodrusen deposits are more commonly found in age-related macular degeneration, as Dr. Keenan introduced earlier, thank God. So where there are a strong biomarker, for progression to late-stage geographic atrophy. So our lab also studies dry AMD. So in a lot of our animal models here, we use this high cholesterol diet, because diet is a risk factor for AMD as well. So we decided to apply a similar approach here, and we fed these CSF1R heterozygous mice this high cholesterol diet. And we found that after 9 weeks, they had increased retinal vascular permeability. And this is all analysed with FOVIS, the software that Jeff O'Callaghan spoke about earlier. Despite this increase in vascular permeability, however, there was no differences in the retinal histology as shown by OCT here. After 18 weeks, however, this increased vascular permeability was maintained and we began to see deposition of material above the RPE, similar to what we saw in our ALSP individuals. When we examined this material in more detail, we saw that it contained an accumulation of apolipoprotein E, potentially indicating that there's some defect with cholesterol or lipid metabolism or clearance in these retinas. So this leads us to the overall conclusion that individuals that carry one copy of variant CSF1R display retinal abnormalities, including the formation of reticular pseudodrusen deposits. However, modelling this in mice with CSF1R heterozygosity, we found that this wasn't enough to induce this phenotype. When we added in this environmental variable of high dietary cholesterol, however, we saw we got increased intraretinal vascular leakage and a formation of these reticular pseudodrusen deposits. So this leads us to the overall idea that disrupted CSF1R signaling may impact cholesterol and lipid metabolism in the retina and influence the progression of retinal degenerative diseases. So CSF1R signaling is being looked at as a potential therapeutic avenue in a number of diseases, mainly through CSF1R inhibition. But this highlights that maybe by restoring regular CSF1R signaling by use of agonists such as CSF1 or IL-34, this may be a potential therapeutic avenue for retinal degenerative diseases associated with these deposits. So I'd like to thank you all for your attention. I'd also like to thank anyone who gets involved in scientific research by being patients in our studies. And I'd also like to thank all the members of my lab, and particularly the names in bold here. Thank you.
Thank you. Any questions? So are cholesterol-lowering agents the thing to be giving people? Yeah.
In a lot of studies of AMD, I think statins have not really materialized as a potential therapeutic, which is quite interesting because there's a lot of similarities, particularly between dry AMD and cardiovascular disease. So obviously it just indicates that it's not that simple. Maybe it's harder for them to access the vasculature of the eye to lower, like, I suppose, cholesterol deposits and that. It obviously requires more research, so thanks very much.
Thank you very much. I'd now like to invite Tom to the podium to tell us about the economic impact of IRDs in the Netherlands and a cost of illness study. Thank you.
Thank you. And good afternoon, everyone. Good afternoon, Retina International World Congress. I'm really happy to present to you, but only in 5 minutes, so it will be high level, the cost of illness study of inherited retinal disease in Netherlands in 2023. I'm sorry, I'm also not sure why, but I forgot to add my name to the slide, but indeed, I am Tom de Ney, as is said in the agenda. First of all, why would you conduct a cost of illness study in the first place? I don't know if you have any ideas, but for us, what is really important is, first of all, to increase our understanding of that particular disease, so in this case, inherited retinal diseases. And secondly, and also maybe even more relevant, to share this understanding with the wider public, with all stakeholders that play a role in the field of inherited retinal diseases, and get a seat around the table and discuss how to address this impact, how to try to reduce the impact. So that's what and why we have conducted this cost of illness study in the Netherlands. Now, moving on to the study itself. What we have done is, first of all, taking the studies, the cost of illness study that were previously conducted in the US, Canada, UK, and Ireland, and localized that to the Dutch setting. We focused on 11 different types of inherited retinal diseases that you can see here on the left side of the slide. Then we assessed the impact of these diseases on 3 socio-economic cost domains. Direct healthcare cost, for instance, the cost of visiting a retina specialist. Direct non-healthcare cost, an example of which is a visual aid, which is often paid out of pocket, and indirect non-healthcare cost. The main example of that is work productivity. Then moving on to the right side of the slide, which is more about approach, I just would like to call out one thing which has been instrumental, and that was our collaboration with both Retina International, but also the Dutch patient advocacy group, the Oogvereniging. I know it sounds very Dutch, but I'm Dutch, so that's why I think I can pronounce it, but appreciate it's a bit harder for others. But with the Oogvereniging, we worked very, very closely, especially on conducting the survey. And thanks to this close collaboration, we managed to get a response from 80 people living with inherited retinal diseases, and that in just a few days. Which was also great to see is that the survey participants, they had very similar baseline characteristics to the participants in the other cause of illness studies. Particularly for this Dutch study, 63% of the respondents were in the working age, 52% were male, 65% not working, 69% had retinitis pigmentosa, and 82% received genetic testing. Moving on to the outcome of the study. As you can see on this slide, the total annual cost summed up to approximately 156 million euros. Of these 156 millions, the main contributing factor was indirect non-healthcare cost. 78% is non-direct cost. I will go into this a bit deeper, but that sums up to 121.5 million euros per year. The overall cost of about 156 million in 23, that is similar to about 30,000 euros per person per year. Then this last slide provides a bit more detail on these cost domains. Starting off again on the left side with the largest proportion, the indirect non-healthcare cost. As you can see, the biggest three items were reduced working participation, presenteeism. Presenteeism is when you are going to work, but you're actually not productive at work. So that's what we call presenteeism. And then informal care is the third element here. Then in the middle, the direct healthcare cost items. As you can see on this slide, the biggest item here was the specialist care with 19 million euros per year. And then last, the direct non-healthcare cost. So I'm talking about aids and modifications. That summed up to 8.8 million euros. This sounds maybe not too considerable compared to the other cost elements. However, this is often paid out of pocket. So people living with inherited retinal diseases feel this directly in their pocket. So overall, as I already started my presentation, I hope these insights help to get that seat around the table for all of you, for everyone who's playing a role in this field and who would like to address the impact and reduce the impact of this disease for the better in the future. Thank you.
Thank you very much. Any questions or? No, yes?
Thank you, Tom. I have a question, and it's actually a question that someone asked me during the conference, but I think it's better that you address it. So it is a survey that you did? And how did you do that? Like, is it online? Is it face-to-face, by mail? Can you just explain a bit how you have done that survey?
Sure, and thanks a lot for the question. So the survey was done both virtually, so people living with inherited retinal diseases could fill it out digitally, but they also had the option to do it as an interview form. So especially for people with severely visual impairment, that option was often made use of. So both options were available. And important to mention here, as I also mentioned previously, our patient advocacy group has played an instrumental role in rolling out this survey.
Thank you.
Thank you.
Yeah. Well, thank you very much, and I'd like to congratulate all three presenters for having their posters selected. So thank you very much. So we're just going to finish this session, but I'm going to invite Dr. Nabeen Podal to tell us about evidence-based advocacy in retinal degenerative diseases. Thank you.
Good afternoon. Thank you very much, Julie, for that kind introduction. So I'm going to shift gears and talk about patient-centric, or what can patients do in terms of research and advocacy. So we've heard a lot about clinical research, basic science research from academia. Now I'm going to talk about what can patient organizations do in terms of research space in inherited retinal disorders as well as age-related macular degeneration. So this is a quick outline of my presentation. So I'll give you a quick introduction to patient advocacy organizations. I'll talk about evidence-based advocacy, following up with the activities, research, and policy actions that Retina International has done. I'll talk a little bit about or invite clinicians, scientists, and other stakeholders to collaborate with patient organizations to generate robust evidence and meaningful outputs, and conclude with some take-home messages. So what are patient advocacy organizations? So traditionally, patient advocacy organizations were those organizations that would work on the provision or of help to their members or advocate for their rights and interests. But with time, they have evolved quite a lot. And they have actively intervened in war on disease with the aim to find cure of diseases. So certain patients and representatives acquired scientific and medical knowledge, and they are known as lay experts. And some of the patients and patient representatives actually collected, formalized, and circulated patients' experience as a legitimate body of experiential knowledge, and they are known as experts of experience. Now, the four particular items that patient advocacy organizations do are support patients and their families affected by a challenging disease, in this case, retinal degenerative diseases, to make right choices, help clinicians to give the best service possible to the patients and prevent bad and outdated practice, influence regulators and payers to make sure that they're basing their decisions on patient preferences and needs, and tell politicians to do policy for patients, not just about patients. So again, patient advocacy organizations, the work done by patient advocacy organizations fall into these three buckets. So one is patient support and funding for research in these diseases. The other one is health policy, to influence health policy and patient care, and research, in some cases independent research or in partnership with clinicians, scientists, and industry. So in terms of Retina International, we do bits of patient support and funding, but we are majorly focused on health policy and research, shaping health policy, as well as doing independent research with our partners, clinicians, academicians, basic scientists, clinical scientists, as well as industry. So you may be thinking, why do we need patient-based advocacy? There are brilliant scientists, academicians, basic scientists, clinical scientists, industry people, but the thing that matters is the perspective matters. So in this comic strip, let's see, on the left, we have a young child lying on a crib, and looking up to the mobile toy. There are a couple of toys hanging above his head, and the parents are sitting beside the crib and thinking that, wow, the baby is really enjoying the toys. But when you look from the the child's perspective on the right, he's just looking at the blobs of the toys. He's not actually looking at the whole perspective, whole image of the toy. So that's what. Imagine the child is the patient, and the parents are the clinicians and policymakers and decision makers. So that there is a room for improvement to adjust that toy and make it more engaging. In other sense, to optimize the treatment based on the person who receives it. So the key message here is that the perspective could differ depending on who is looking at it. I hope that makes sense. Now, let's talk about evidence-based advocacy. Quick definition of evidence-based advocacy that patient advocacy organizations practice these days is advocating in targeted, evidence-based, well-educated, and professional manner. And not just advocating in this manner, but also measuring the impact. What is the impact of the advocacy work, the research work, the policy work? Where has it gone? Is it moving the needle in terms of making decisions by the decision makers, or just publishing papers and getting some track records? So those are the things that evidence-based advocacy focus on, and those are the things that particularly Retina International is focusing on. So you might be thinking, what is Retina International done in terms of that activities that I've just described now? So we have, although patient advocacy organizers are not academicians with limited capacity, there are some stakeholders where publications matter. And we have jumped onto that boat. And we have published quite a few papers. So one of the key papers that we've done is to look at the impact of inherited retinal disorders. Economic impact of inherited retinal disorders in 4 countries, UK, Ireland, USA, and Canada. And we found that the economic burden of this disease is severe. There is a substantial burden, which is mostly driven by the non-direct healthcare, non-direct costs, which Tom mentioned earlier. So thanks, Tom, for that lovely introduction there. So this work has been really highly appreciated by the community. And if you look at the impact of this work, it has been cited more than 40 times in the last few years, which is one of the highest impact. It is in the top 5% of the publications in that year. So the other work that we did was the cost of illness in late-stage age-related macular degeneration. So again, we try to kind of work on these areas as well as disseminate the information on different platforms, such as the conferences relevant to eye and healthcare, eye and vision care. And we presented this work to one of the world's best conferences, such as the annual meeting of the Association for Vision and Ophthalmology Research and EU Retina and so on. So the work that we did on the late stage AMD was looking at the burden of the disease in the society in terms of direct health costs, indirect health costs, the well-being and mental health impact, as well as the productivity impact, that absenteeism, presenteeism, job loss, job reduction, and so on. So this work was presented. So again, we have been disseminating our work widely to clinicians, scientists, as well as our patient community. Again, in terms of the inherited retinal disorders space, we have done surveys in relation to what are the experiences of patients obtaining genetic testing for IRDs. And surprisingly, I think Helene mentioned earlier that the odyssey of diagnosis is quite long. They had to wait for more than 5 years. I think it was 40% of the people had to wait for more than 3 years to get the diagnosis, genetic diagnosis. So we've identified so many gaps in the literature from our surveys, and that could be addressed by clinicians, policymakers, and other stakeholders. So we also did a genetic testing landscape in Europe in 2020, which has taken quite a bit of time to get published, but hopefully in the next few months it will get published in one of the decent journals. So now, just talk about what do we do with this data? Do we just publish it and just let others utilize the data, or do we take actions? The answer is we take actions. We get this information, we get this data, we analyze the data, we take this information and utilize it in our policy activities. So some of the policy level activities that we have been engaging in is we have been utilizing the data that we obtained from our studies to inform some engagement with different organizations such as United Nations, World Health Organization, European Commission, and so on. And we've been giving advice to our company industry partners, to academicians in different areas, in different European commissions in food and health safety, to different scientific communities, as I mentioned before. And also, we've been sitting in different clinical trials, designing committees, legislation, and so on. So we put the information that we obtain from our data, and then inform the knowledge obtained from the data to policy actions. So now, with that sort of information, I'd like to just let you know what sort of research that we can do, we are competent in. So the sort of research that we're competent or we're able to do are not basic and clinical science research, because we wouldn't have the clinical data and so on, but the research that we're particularly interested in are treatment adherence of therapies, inequalities in access to diagnosis or therapies, development of PROMs, patient-reported outcome measures, or patient-reported experience measures, quality of life, daily living experience, disease burden, cost of illness, that's similar Tom presented earlier, core outcome sets to identify meaningful outcomes, because patients are the consumers of the drugs. So why not consult them before you are initiating a therapy? So patients can be very helpful stakeholders in developing meaningful outcomes, as well as patient preferences on health and risk of benefits. So again, with that, I'd like to invite all of the relevant stakeholders, academicians, clinicians, scientists, industry partners to come with us. And if you have any ideas, or if you have found any gaps in the literature, because we're all working for the same goal, which is to foster innovation, to provide equitable access to new and novel therapies to patients. So we're very much welcome with your ideas. With that, a couple of take-home messages, and I'd like to conclude this session with a couple of take-home messages. So basically, patient advocacy organizations support, educate, do research themselves, and take actions of this from this data. Patient groups, mostly, particularly Retina International, follow evidence-based advocacy, which is a targeted, well-informed method, as well as measuring the impact. Retina International is particularly involved in evidence generation and taking action, which is doing high-impact cost of illness studies in IRDs and AMD, patient experience journeys, research and policy priorities, and consensus building on impact and experiences of people. We take action, so we use the data to inform policy actions. And again, I'd like to invite you all to collaborate with us and support us in our goal in achieving equitable access to new therapies and foster innovation. So thank you very much.
Thank you very much. Wonderful work. Any comments or questions? So this is probably unfair. If you had one thing you could, which would make the most difference, other than treatment, widespread treatment? Any of which would you pick that would make the difference? You know, to the.
It's
hard.
I don't have any preference, to be honest, because all of them are equally important.
Yeah.
But I think engaging with patients to have that meaningful outcome, what matters to patient, that's very important. And I think I would advise all of the stakeholders to engage with patients early in the life cycle of research.
Thank you very much. Thank you. So I'd like to thank all the speakers. This concludes this session. And whilst the panel are coming up to the stage for discussion, we'll move on. Thank you.
Hello, everybody. Wow, what a day. This has been an incredible meeting, targeting the cures. And when I think back to what it was like 34 years ago, And what we would have thought today, I think we would have found it daunting, all of the things that we needed to learn. And maybe it's good we didn't know what we didn't know at that point in time, because it would have been too scary to proceed. So today we've heard about genetic disease and architecture and clinical diagnoses and reports and results from different clinical trials using gene augmentation, editing, and agnostic approaches, as well as compassionate care. We have learned about different outcome measures. We learned that Mark Panesi likes baked ziti and that Bart likes pizza stromboli, or calzone, I guess it is. We've heard about different mechanisms of disease and how to generate different animal models. We've covered so many different topics. But there are things we haven't covered. There are new approaches on the horizon. There is going to be more gene editing, I'm sure. There's going to be RNA editing. There are going to be dual vector approaches for treating different diseases and dual reagent approaches for treating different diseases. Transplantation with or without gene therapy, readministration if levels go down. And there are going to be new vectors which home into different cell types depending upon where they are administered. We haven't spoken about some of the approaches that are being used now to develop treatments for different forms of macular degeneration and geographic atrophy, such as anti-neovascular approaches and anti-complement approaches. We're just on the cusp of seeing a lot more promising therapies, the things that you can give your patients and that will save people from going blind. But there are a lot of problems and a lot of issues that must be addressed before this can all happen, one of which is funding. These studies are extraordinarily expensive. Manufacturing, how to make all of these different reagents, how to educate the population about this, how to make sure that the surgery is done the same way every time, or at least we know what the variables are that are important in controlling certain complications that may happen. And this is something we can do. And I think this is just a taste of perhaps what this panel will be discussing in the next few minutes. I'll give it to Bart.
Thank you, Jean. You probably all know us or know some of us. I'd love to quickly just introduce ourselves, so I'll start. I'm a genetic ophthalmologist, and I work in academia on IRDs and gene therapies. Jean, do you want to very quickly, so that the panel is known to the public?
I do translational work, trying to move ideas from the clinic from the laboratory to the clinic.
Dan?
Dan Chung. Been in the retinal gene therapy space since 1994 when I was a fellow at the NEI, and just worked in that space trying to get drug development to the folks that are living with IRDs.
So my name is Avril Daly. I'm the CEO of Retina International. I'm a patient advocate. I'm a person who's living with an inherited retinal degeneration, and my work is really to focus on the gaps in policy and legislation that are creating roadblocks to innovation and to bring the patient voice to the discussion.
I'm Mark Panesi. I'm an inherited retinal degeneration specialist. Much like Bart, I've been trying to live up to Bart's image for many years. I still haven't gotten the style down. But primarily these days, I'm conducting clinical trials, mostly for gene therapies, antisense oligonucleotides, stem cells, all of the kind of novel first-in-human type studies for IRDs.
My name is Darren Morrissey. I'm the CEO of an organization called NIBRT, which is the National Institute for Bioprocessing Research and Training, which is a bit of a mouthful. Essentially, we are a biomanufacturing research centre based in Dublin, focused on supporting the manufacturing process for new medicines, monoclonal antibodies, recombinant proteins, and the brave new world of advanced therapies. So I bring, I guess, the manufacturing voice to the discussion.
Thank you.
Yeah, and I'm Todd Durham. I'm with the Foundation Fighting Blindness in the United States. I'm responsible for clinical research activities, which includes the Foundation Fighting Blindness Clinical Consortium and the My Retina Tracker Registry. I view my job as working with industry representatives like Dan and other companies here, but also researchers like investigators here, Bart and Mark, to find ways to de-risk therapeutic development and do what we can as a foundation.
Thank you. So now we know, do you guys want me to, is this working? Can you hear me now? Okay, good. So we don't want to not be transparent. We've seen success with gene therapy. Okay. Is this better?
Yes.
Good, thank you. We've seen success with gene therapy. Luxturna is an example, a beautiful example. Up to now, actually the only example of a gene therapy that was solidly developed scientifically with huge promise in studies and then finally made it to market. The company that was a spin-off company of Children's Hospital of Philadelphia and the University of Pennsylvania, where Jean Bennett, the mother of Luxturna, developed this product with her team. Spark Therapeutics did a good job, but then Spark Therapeutics was acquired, and if I say something that's wrong, Dan was on the team in the development and then switched over to Spark Therapeutics. All of that was very successful. Roche bought the company, and then you can now think about Roche and then Novartis, who bought the rights to actually distribute the product in all the countries bar the US, where Spark does it itself, the Spark does it in the US. They have not sold a lot of vials. And so you could say that this is scientifically certainly a success, but for the current owners of the rights and of the product, it's not been a commercial success. And so we shouldn't be not transparent about the issues here, because what you see is that some other products have been so-called failing or haven't lived up to what was initially thought to be, oh, we're going to cure blindness and so on and so on. There's clear signs of improvement, but it's been quite a rocky start to what is gene therapy and providing gene therapies for more individuals, for more genes. And so that is the situation currently. We badly need another success. There is investment coming into the field, but not enough. And so there are some issues, and we want to address them in this discussion panel. Obviously, in an hour, we can't cover everything, and we want to end on a high note at the end. But I want to bring up a couple of issues to the panel. And because we, for example, have Darren here, who is a specialist in manufacturing of viruses, also Jean knows a thing or two, and Dan knows a thing or two about how you manufacture viruses in high numbers so that the product is good first to do the studies and doesn't cost too much. And then how do you bring that to market? Because it is the virus in a vial that is the medicine. How do you do that, and how do you make sure that the quality is always the same thing? We've seen some issues with qualities in studies, and maybe, Darren, if I may, do you want to chime in as to where you think that the problems are currently and how to solve them?
Okay, so Bart, I first and foremost, I might stand myself back as being the expert in manufacturing viruses. I guess what we do is, as an institute, we have a range of principal investigators, researchers who focus in on different modalities of treatment, of biologic type treatment. And what they do is they try to focus on just what you said, I suppose, the consistency of the biologic manufacturing process, the efficiency, and ultimately, I guess, the stability and safety of the product. So there are our golden rules before we ever start. We work in collaboration with companies, big and small, and particularly with academic labs who are developing new products. So bringing it from discovery mode, from benchtop mode, through the early stages of manufacturing. So we have a facility that is not a GMP facility, so it's not a scale up and scale it to a level that you can put it into a patient. It's before that point in what we call the pre-GMP setting. And pre-GMP is where you can iron out the kinks, for want of a better term, in the manufacturing process so as you de-risk. I've heard that term used a few times over the course of the day. You get through what can be the ultimate fail point for new medicines being developed. So with a lot of, what I would say, foresight from the Irish government in investing in this space, what we're trying to do here is working with the big manufacturing companies that are based in Ireland. Most of the world's largest biopharma manufacturers are here. We're trying to get into the early stage of that development process through the manufacturing steps. And as I say, iron out those kinks, but as a consequence, get more medicines through that early stage of the step between discovery and bringing and scaling up for manufacturing to patients. And again, when you talk to VC companies, investing companies, they will often talk about the valley of death in the development of a new medicine, be it a viral gene therapy or a cell therapy or whatever. In fact, you probably have about 4 or 5 valleys of death in the development of a product. And we are very much focused on the first and potentially the most intractable one, which is that point of bringing it from the bench into early stage manufacturing.
Thank you, Darren. And maybe Jean and Dan, would you want to comment on what it then means to get it into Spark Therapeutics, for example, and making sure that the product is at a GMP level, and how difficult or easy is it to keep that at high level? Also, the production of good quality GMP vector, that is the virus, is that something that every country in multiple sites should be doing, or should we concentrate? What is your opinion? Maybe Jean first, and then...
Sure. Well, by definition, GMP, good manufacturing practices, is high-quality vector. And what we did with Luxturna when we were in phase 1 studies was use an academic-based GMP facility to generate that vector. It was a facility run by Fraser Wright, who had experience in a previous gene therapy company, and he knew how to do all this. And it was pretty straightforward in an academic setting to set up this facility. It occupies a small amount of square feet, and it was rigorous in terms of generating a product which was sterile and so forth. And one of the attractions of working with the eye is that you can make one batch of this reagent, and it's enough to treat every person in the world with this material. But one of the advantages of doing this in-house in an academic setting for phase 1 is that you don't need all the bells and whistles that you would need for a phase 3 registrational trial, where you need to prove that you can make multiple batches of this reagent, which all fit within the same criteria in terms of levels of endotoxin or levels of cesium chloride, et cetera, whatever is going to be measured. In other words, the quality control assays are much, much more rigorous. Even if you are getting a reagent of the same quality, you have to do many more of these studies to be labeled as a late phase product. And so when Spark Therapeutics was formed, it set up a different set of facilities where it made multiple batches of this vector using that more stringent criteria, making multiple batches. When we started our phase 1 studies for LCA5 that I described earlier this morning, I established a vector core facility at University of Pennsylvania, which basically outfitted, removed all the lab benches from a lab and inserted a prefab unit, which is sterile and you can keep clean, and you walk in a certain way and you walk out a different way, so you keep things very sterile inside. We generated our own vector and used the similar quality control measures as what Fraser Wright had used at CHOP, including showing that we had minimal amount of empty capsids because those don't do any good. And that approach makes you much more nimble, first of all, but it cuts the cost by orders of magnitude. So it is one way of moving forward quickly.
Just a couple things to add on that is that once you get into the industry space, and obviously things have progressed since the days of Spark, and things such as, we hear the term scalability, and some of these earlier things and techniques that were used, they're not really scalable. And you'll argue some of these don't need to be scaled because a lot of the IRDs that are currently being addressed, as Jean said, can be done in these smaller manufacturing areas with older techniques. But if you want to scale up to, instead of treating hundreds of patients or even 1,000 patients, but go to thousands of patients or tens of thousands of patients, those techniques don't work, and the scalability is where things get a little complicated. And what had happened was there was such a difficulty of getting a manufacturing slot. They're called CDMOs, or contract development manufacturing organizations. And sometimes you would have to wait a year, 18 months to get a slot just to get your virus made. And what happened is then companies started to think, well, we'll just make our own CDMO, and we can produce our own. And then the pendulum swung that now there's so many of those CDMOs that they're actually looking for people to use them. And with that, we in the industry space have found that some are closing. They may actually have a contract with you, but then all of a sudden they decide to close, and then you have no vector. Or they make a vector, and as Jean said, you have to go through this high amount of quality control that the vector just doesn't cut the mustard. It's just not good enough. So just because they make it doesn't mean that it's good enough for a patient. And the reality is that you can do clinical trials, and as Jean mentioned, there are different standards for each one. There's another standard once you go into the commercial realm. And so each level, there's this scrutiny and quality control. So this is kind of where we are right now in the sense that there are a lot of slots, but the expertise, and I'll just speak on my opinion on this, is not across the board. You know, it doesn't have the expertise that we had at CHOP and at Spark across the board, where every batch that came out was pretty good. And so things like purity, all those endotoxins, all the QC things really have to be up to snuff, and that's where we're falling short right now. So, and each one of those runs, you still have to pay for.
You're obviously the patients in this room and their treating physicians are interested in high-quality product. I think one of the things that I can say as a member of the Philly team of genes is that the quality of the production was so sky-high compared to competition that was one of the reasons why the product was so clearly working. So people are interested in getting high-quality product. If you say across the board we cannot find the same quality, how do we solve that?
Well, this is a good question, and this is where we thought about the idea that industry folks start talking to each other and start to get shared experiences of what was a good CRO, which is a clinical research organization who'd run clinical trials, and CDMOs, who we had good experiences with. And try and keep this word of mouth instead of all of us in industry kind of working in silos, because there are a lot of things that we can't share, but there are a lot of things we can share. And I think that's one of the things is we just need to start talking to one another.
Thank you, Dan. And this may be a segue into another topic, high unmet medical needs. Where are we? We've spoken about vector technology, vector production, the issues with it, how to solve it. The other thing is that many of the gene therapy companies are small startups, and they need money. They don't have the money, so they need investors to come and give them money, and then those investors get return on investment. So Dan, you've been speaking about how difficult the current climate is in getting sponsors to pay for these trials and getting the money in. Where are the issues and how to solve them?
Well, I think part of the issue is that we need investors who are willing to look at the long term. And I'll say that there are all types of investors, those who want to invest and get a quick return in 4 or 5 years. And then there are investors who understand what gene therapy is like and what IRD is like. And they're willing to be, a 10-year kind of long-term goal. And I think that's part of it. I've heard the term ROI, which is return on investment, more than I'd like to hear recently. But it's true, that's their jobs, they're investors, and so they want to work for their clients. But I think the idea is if we can just start getting more of these approvals, then people understand that this is more possible, and that even helping out 500, 600 folks, that's significant. And I think we just need to get that message out a little more. That really changing somebody's life because of their visual capabilities has changed, I think can be a powerful statement. And I don't know if we've been as good on the industry side talking to investors about that. So I think that's one thing. We have to change our message and how we're going after things, because there's a lot of good things that are happening, and that message, I'm not sure, is always getting through.
Avril, I'm going to come to you now. The ideas that people at European Medicines Agency for Europe and the Food and Drug Administration for the US have about what a success is with gene therapy varies. Food and Drug Administration and European Medicines Agency obviously are agencies that work for the patients. What they do is their role is basically to try and control overly zealous people who want to do all kinds of crazy treatments for patients, and they make sure that the patients benefit from it. But they also, and that is something that I saw transpire over the last years, actually do not know a lot about what you can expect from a patient's eyes who suffers from IRD. So they seem to think along lines, like 3 lines of best-corrected visual acuity, which is easily achieved with products for example, diabetic disease of the retina, because that patient mostly has seen 10/10 vision and has gone down, and with an anti-vascular endothelial growth factor injection, such a patient could fairly easily gain 3 lines. Obviously, people with IRD sometimes have never seen 3 lines, and so outcomes and expectations are not well understood by the regulators. And so the first question to you, Avril, is what do you think from the community that you're a leader in is enough to achieve with a gene therapy by making somebody who doesn't see well a 10/10 vision person, which is most of the time not something that we can do. But what is enough, and what type of level of effective treatment should be defended at the level of the regulators?
Well, it's in some ways a simple question, but it's not really. And, you know, as a person who's sitting here, as a patient who advocates on behalf of our community, many of whom are in the room today, first thing I would say is one of our members came up to me yesterday evening. We had a workshop yesterday on value. What is value? What does it mean to us? And trying to have a conversation about the fact that we are valued in society and we have to demonstrate that value consistently. And Tom's presentation earlier around the cost of illness, we as patients have to provide that data to show that we are worth the investment. And that can be a shock and a surprise to many patients, you know. And I think it's important to have this conversation with our patients here today, who often come to us in Retina International and say, why does it take so long? Why is the research that we have been funding for 30 years taking so long for us to get to, you know, access points? And you know, we see the results of clinical trials, but there are so many different steps along the way. And even today, I'm learning about this issue concerning the investment that Dan has raised, you know, issues of accessing funding to take us to the next level. Who's really making the decisions about whether we get our treatments or even whether these treatments come to fruition in the first place? I think it's really great that we're tackling this subject, maybe for the first time, but certainly not the last here today. So to go back to your point about what is enough, it's come up several times even in this Congress and in our own Retina International meetings over the past couple of days. We're living with degenerative diseases, and we don't know what that reality will be, how long and how fast the degeneration will be, what age and stage a person has with their condition. The answer may be different, but the uniform answer that we get from our patient representatives, who are many in the room today, would say, to be able to preserve the useful vision that you have would be an enormous win. And at a meeting we were at on Wednesday, where Sylvia Seralini from Eyes on the Future, who's a mother of a child with RDH12, played a video. And what struck me was a little boy who was asked, you know, what would it mean if your eyesight could just stay the way it is? And he said, the world. It would mean the world to me. It would mean the world to me if my eyesight stayed at the level it is right now. You know, preservation of vision is important. It means that people are independent, that they can engage in society. We know from our studies that 25% of people living with vision loss are employed, but within that figure, they're employed for 8 hours a week. So what are you actually measuring here? There are people who can be active, or already are active, members of the community, but could be so much more. But also we have to consider their families, those that support them, and the invisible, intangible burden and cost. And that needs to be taken into account as well when we're assessing value and the importance of these treatments. And it really, you know, is important to say also that those of us in the room, that doesn't mean that we should lower the bar, right? It doesn't mean that we strive for just this or just that. But are we looking for cures? Of course we are. The name of this Congress is Targeting the Cure. But really, if you were to ask patients, patient by patient, condition by condition, we're looking for solutions. We're looking for a better outcome for us, for our future generations, because these are genetic diseases, you know, and that's really what's important. But I also wanted to just bring something up, and I hope you don't mind my doing a slight detour.
You can get away with it, Avril, you know.
Sorry, but it's important because many of us in the room here, we are advocates. We sit at tables, we have discussions with decision makers, and we use the data that Nabeen was talking about, you know, that we have the studies, we take the voice and the perspective of the people in this room and what their condition means to them. So it's not just Avril Daly sitting in front of a regulator, or David Sanchez, or Franz Badura. It's all of us. We represent all of us. And that's really important. But we also work within the rare disease community, and we see other disease areas, you know, and we learn a lot from how they coordinate around, you know, their specific issues. And the challenges are numerous. They're the same across the board. Yet there are instruments and there are opportunities out there for us as a community to avail of. You know, there are policies, there are actions, not only here in the EU, but also globally. And the frustration we have as patients that have the privilege to travel to these congresses in different continents and provide our perspective is everyone is having the same conversation in a slightly different way in a different room. And it's siloed. The conversation needs to be brought together. We attend events like the World Orphan Drug Congress. If you attend the World Orphan Drug Congress in Barcelona, you're talking about European action. If you attend the World Orphan Drug Congress in Boston, you're talking about what's going on in the United States. If you attend the one in Asia, it's about what's happening in Asia. Why are we not having a conversation about harmonization? Because we're all human beings. And the reality is that we all require the same thing. So why can't we all get in the room and avail of the opportunities that exist in all of these continents and really strive together? Because I really feel that the IRD community in particular is missing out an opportunity by being a little bit too siloed. And that's something that we need to consider also.
Thank you, Avril. Certainly a point carried by many in the room, apparently, and with that, I want to use that as a segue to bring Mark Panisi in as one of the leading IRD experts in the world and works in 2 places in the US. So Mark, do you want to chime in and then pass on to Todd from the FFB point of view what to do about it? But do you want to chime in and inform our public here where the issues are with, for example, 2 bodies, EMA and FDA, not always asking the same things? And can you inform the public here, and can you state what the issues are, and then maybe bring in Todd as to how we are going to handle this? You can talk more about what we did on Wednesday. I've not said anything about that, but bring it in.
Sure. So, you know, I think that I kind of hold 2 conflicting feelings at the same time. I have a feeling of frustration because I want cures today, but then I also have a feeling of hope because I see all of the treatments and trials that are coming and on the horizon. But there are, you know, certain barriers, I think, that, you know, we've been sort of running into head on, and they map out to different domains. And, you know, as you heard earlier, you know, funding, I think, is one big, you know, issue. And, you know, in the past, many trials and such were funded by governments. You know, if you look at the budgets of, you know, different governments, they've been very flat or even decreasing, and so that's, you know, worrisome. You know, we get money from, you know, venture capital who, you know, as Dan mentioned, are looking for a return on investment, and that can create, you know, issues, especially for rare diseases. And we also rely on philanthropy, but there, you know, there might be some other, you know, creative ways of finding funding, and I think maybe Todd can talk about that. You know, things like bio bonds, but you know, that is one big issue that we need to work on, and I think that's actually one place where the people in this room can make a difference, because you know, we need the patient advocates to go out and lobby, you know, for funding, for research. And these days with social media, you know, you can have a profound influence, you know, on your various, you know, governmental agencies. There's also another barrier that we hit kind of on the regulatory side. As Bart alluded to, you know, oftentimes the regulators really are not domain experts. And, you know, that's not necessarily their fault. There's a limited number of people in these government agencies. They can't be an expert on every disease. But we often find, for example, with the standards that we are held to with rare eye diseases or eye diseases in general at the FDA is much more stringent as compared to other diseases. If you look at neuromuscular diseases, for example, and drugs that have been approved, in those domains, under much, easier goals to meet. And we're kind of held to these very difficult, standards, which is frustrating because we've seen a number of trials where we often will see some evidence of success, and you'll talk to the patients and they'll say like, yeah, I feel like this treatment helped. And you can, the question I always ask that I think is most telling is, if we've treated one eye, say, well, would you have the other eye treated? And if the majority of the patients say yes, that's a pretty good sign that it is doing something. But we've had a lot of these trials where we've heard this, and then the endpoint, they fail to meet the endpoint, and at that point, the money, to go on sort of evaporates, and the program dies. And there are a lot of these, programs that have, shown something, but not quite made it over, that valley of death yet. But I'm actually hopeful right now because I think the tide is turning. I think we're right on the cusp of having a few more successes, and I think that is going to compound. But we do need to solve, some of these problems, kind of the regulatory barriers, the lack of harmonization, between, some of the regulatory agencies. For example, even with Luxturna, there was one country that demanded that they do a second trial in their country specifically because they didn't trust all the other countries. And stuff like that is just really inefficient. And, I think about that we were treating patients in 2017 in the US, and there are some countries out there where Luxturna still has not been approved. That really needs to change. And I, welcome Averil's idea of, like, how do we come together more as an international community? I think we're getting there. Like in the US, we've created a clinical consortium with Foundation Fighting Blindness. We're aligning now with ERNI. We have representatives in South America and Brazil. So I think we really are starting to kind of grow that global network, but we need to keep on it and make it bigger and, really get everybody at the table. So.
Thank you, Mark. And I think you said something that Avril was also saying. Do not forget the power of your voice as patients and as IRD experts. Do not feel that you're on your own. You're in a community. And as Mark said, with social media and the abruptness and directness of information that is being shared these days, you can make an impact. We have seen regulatory differences. For example, for FDA, there are now some anatomical measurements that if you give a patient for AMD, age-related macular degeneration, certain drugs, and the lesion of atrophy, the quality loss, the size of that quality loss is actually measured and doesn't necessarily need to correlate with increased function or function that is preserved. That is seen as a proper outcome measure for age-related disease. It's not yet been used and approved by the regulators for IRDs. That is not logical. The thing I said first is not recognized by EMA, European Medicines Agency. And so the cost of trials, certainly for small companies, Dan is Chief Medical Officer of Sparing Vision, a fairly small company. If he has to do a trial in the US and he has to do a trial with different structure and outcomes in the US, so the two different areas, that's not efficient. It costs loads of money. So Todd, I want you to chime in because you've been privy to many discussions. You're very well connected, and you are part of a very powerful organization, FFB. How do we address those things, the differences, because these are barriers to the development of as cheap as possible and as good as possible drugs? What's your opinion?
Yeah, so I'll start by letting everyone know some of the other activities that we've been involved with. David Birch presented results earlier from the RUSH-2A study, which is a study we started back in 2016. And we've taken the 4-year data from the RUSH-2A study, and not just as an academic exercise to show at a Congress, but to take it to the next step and say, if we were designing a phase 3 study for USH2A associated retinal degeneration, and our goal was to demonstrate that the treatment would slow or substantially slow the further progression, either with a functional measure or with an anatomical measure like the EZ area, show a difference in slopes. How many participants would that take, and how long would it take to how long would we need to follow participants? And how large would that study need to be? Just to ask the single question, statistical question. And the numbers, when we first looked at them, I think were sobering, and it was kind of like a slap in the face. We're not going to find hundreds of people with an USH2A retinal degeneration, and we're not going to find a company or its investors willing to follow people for 4 years in a single trial, let alone 2 trials, which is often what the regulators will tell companies, even today, in rare disease. So this is currently not an acceptable state of affairs. It means that therapeutic development is infeasible if we're going to have to stick to these current level criteria, and we think they're unrealistic. So we, 2 years ago, we invited a representative from the US FDA to look at this information with us, Dr. Chambers, who's now retired from the agency. He was nice enough to attend, and he gave us candid feedback, and I think we exchanged some fairly strong opinions with him. Those talks have further progressed now. As Mark said, earlier this week we had engagement both with the US FDA and EMA, and through another forum, the IMA from Iceland. So now they're listening, and so that's progress. We still have a lot of work to do, and I think I echo Mark's position. We're impatient, and for success from our perspective is companies, when they have a promising technology, a breakthrough, something they think is going to work, like RDCVF, they're not asking themselves, what is the endpoint? How am I going to get this product through the regulatory hurdles? They're not asking that question. It's just a question of, will this product work or not? It's not steps we're going to take. So that's what success would look like to us, because we know all medicines won't work out. So I think that's the challenge. We have, I would say, a bit more science to do, but a lot more advocacy to do with the regulators.
Yep.
Thank you. Is there anybody from the public who has a question or wants to chime in? Feel free to do so. If you want to comment on anything that's been said, if you have a question regarding anything that's been said, there can be a microphone. Do you need one of my mics here? So any question from the audience? Yes, Franz. Maybe say who you are. Many people know you, but some do not.
Franz Badura, Chair of Retina International. It would be a dream when all these things became true, harmonization and this interaction, which we really want to inspire over the near future. I do really hope that we will be successful. There's one party we're not talking about it. These are the payers. Mm-hmm. Mm-hmm. I don't want to be too negative, but it is another big hurdle. I think we really have to think about it early enough to bring them in this discussion as well as we have this discussion.
Mm-hmm. That's certainly true, Franz, and thank you for that comment. So for the people who don't know, and this is also part of the message of hope, I think, and some of our panel have already alluded to this. we as a group, this is IRD experts, patient organizations, pharma companies, and the regulatory agencies, EMA and FDA, have started engaging on at regular intervals. We've done that the second time on Wednesday of this week. ERNI, the European Reference Network for Rare Eye Disease, has instigated this, but we've worked very closely with our partners, Retina International, FFB, and obviously our pharma partners. And we want to improve things. We want to say stuff that's been said in a very eloquent but elegant manner. We've been a little bit harder in the discussions also with the regulators, where we've made clear that it cannot be that there's inconsistency and between the regulators, and we've discussed matters through as to how to make more of these innovative treatments come to the patient. And the reason, Franz, why I've not yet pushed for the payers to be part of this discussion is because I do believe that we need them, but we also need to make the products cheaper first. I honestly believe that the pricing of the products is too high because of many factors, and not necessarily people who invest and want return on investment. We shouldn't slag those people off who invest because we need them badly. And if you look at your pension fund invests in pharma industry. So there's a little bit of a circular thing here. But we need to do better, and we definitely need to make sure that the production is far cheaper, far better. We also, I'm pretty certain, can bring down the price of a product where the regulatory agencies to sit in the same room and agree on what is a successful outcome, and not do one thing in Europe and another thing in America, and include also Asian organizations, Australia, Canada, the UK, etc., South America, Brazil, a huge country, to get them on board in the discussions and make sure that everywhere where trials are being held, the same demands are being put forward. Any comment on that? Dan, I see that you are.
Sure.
You have an urge to chime in.
That's one of the biggest things I learned when I switched from academia to industry. I always thought that if we just get this drug approved, boy, we are all set. And then I went to industry and realized that really starts a whole nother set of activities. To get it to the patient, you have to get it approved by the payers. And that was the final obstacle. And right now, Luxturna is approved in almost 50 countries around the world, but only 2/3 of them have payer authorization. Yeah. And there are a lot of other creative ways that they're getting things paid. In the United States, what Spark did was we blanketed the country and basically met with almost every single insurance group, made presentations, and we had about 96% coverage when it came to private insurers. And then for the public and government insurers, it was about 88%. But those were all meeting and presenting what Luxturna was about, all the development process, the effects of the drug itself. And so right now in the United States, there hasn't been one patient that's been denied Luxturna who has been qualified for it. And I know here in Europe, it's been a little slower, but most of the governments here have funded Luxturna as well. So the idea about payers, one thing, we talk about endpoints, and sometimes we don't think about, the endpoint is just not about registration and about regulatory. The endpoint is also to the payers, because the payers have to buy into that endpoint as well and see that this drug is effective. And so the idea of endpoints really goes to that. And plus the patient have to have confidence that this is something that's going to be work as well as your physicians. So the endpoint really is for not just regulatory, but it's for the patient, the physician, and the payer. So to me, the payer is the final check that we have to get, and they can be a little difficult at times, but they're up to reason. And I think the price, in my opinion, don't quote me on this, I think will come down, especially for a lot of these products that, things like what we're working on, where it's more about slowing disease than necessarily restoring function. So I do think that that would be in the future. Hopefully I'll be right, we'll see.
Darren, I saw you...
I flickered there.
I saw you confirm that pricing could come down. What do you think?
Yeah, I mean, the first thing I would say is probably to agree with your point that you were making. I'm going to paraphrase. There is shared responsibility needed here, and I would go through all the various steps in the development and production process before I even start talking about payers, because frankly, payers have so much other things to pay for. I mean, they have to factor in duty of care to I suppose to everybody who's feeding into that system. So for me, it does start actually with the return on investment question, and it starts with investors. And again, don't quote me on this, but investors, frankly, being realistic about the benefit of the product that's being developed, realistic about the size of the market, and actually factoring in some societal benefit that they're actually investing in as well. So that's the first thing. Second thing is, I think, and I would say this, I believe that there is massive underinvestment in manufacturing science. It took 25 years for monoclonal antibodies, which are now de rigueur in terms of treatment of cancer and so on, for the price of those to come down, because of stability and standardization in the manufacturing process. Today, viral vectors and the broad advanced therapies range, whether it's cell therapies you're talking about, it's more of a cottage industry. It's a range of cottage industries, and there's a lot of variability. Variability costs, because variability results in multiple different processes, multiple different vendors selling products against each other. It also, by the way, has a safety dimension to it. So any questionable safety will cost a system as well. So for me, there is a scenario whereby everybody takes a piece of that responsibility and engages on it.
Avril?
Yeah, I think this is really, there's so many things that I would love to say. Just to start with what Darren has said and this issue of manufacturing, because this is a slightly new piece for us that we're kind of getting our heads around. Because it was, to your point, Dan, what has been frustrating for the patient community is we understand that, whatever decision is made at clinical trial stage, whatever value proposition is put there, that follows the treatment all the way through the regulatory, which is becoming much more interested in value now. It used to be just efficacy and safety, to that HTA process. And you I've sat at review boards where they go back to the clinical trial data in every country, and now we're going to have joint clinical assessment in Europe, which should allow for a more consistent approach, because currently we have 70 agencies looking at HTA in Europe. But going back to the point about investment, there has to be some form of public-private partnership here. There has to be some sort of hybrid model, and there are discussions happening at policy level around this, because there is a recognition that this is a challenge and that there has to be investment at all levels of this journey. And, I can take in a European example here. The European Investment Bank, the shareholders of the European Investment Bank are the member states of Europe, so that's 27 countries. And that investment bank is driven by policy priorities within the European Union. And a policy priority within the European Union is research and development, as well as rare diseases, so also called orphan therapies, high technologies, ATMPs. And they are willing to invest in this space. And they are willing to invest in manufacturing. That's some clinical trial development, and manufacturing is one of the areas. But it has to be done in a way that is encouraging collaboration. So again, you know, small biotech companies, we know that they're up against the clock. You have very specific timelines, and that the statutory funding agencies are too slow. You know, so that's just never going to really work in its own silo. You know, it's just not going to happen. But there is scope that's starting to emerge to understand this challenge and drive it forward, particularly for very small populations. And if you consider inherited retinal degenerations, we have 300 different genes. We all know this. Maybe there's a way for a number of these conditions, you know, the similar tissue, similar approaches, that could avail of this type of initiative. And I'm sure there are others. And again, it goes back to my point about understanding what's happening in different jurisdictions and being able to avail of the possibility. And the final thing that I would say about this is as Mark has said, you know, it's really great to have these opportunities to discuss because, you know, the European Reference Network, which is our coordinator, Helen Dolphus, is here today. That is based out of European policy. So, you know, we are ticking the box here. We are allowed to work beyond Europe to South America, Australia, and the United States. There's room here, and there's scope here. And we really have to keep an eye, because again, to my point, we cannot afford missed opportunities. And the other thing that I was wondering, just when you were talking, Dan, and just to probably a very naive question, but I wonder what the cost of inconsistency is for governments. You know, if you sit there and you say, well, we have to do this trial and we have to do it again, where, with all of those decisions and those, you know, regulations, let's just say, that diversity of approach is a huge cost. And who's going to pay for that cost? But the agencies. So, you know, the European Medicines Agency, the FDA, they're funded by the state. They're civil servants, right? And they will say that themselves. So they're funded by us. And they're making decisions that is increasing cost that we as society are going to have to pay for. I'd love to know what the cost of that would be. I'm just going to leave it at that.
It's a very, very good point. Last thing before I let you go and have a drink, and the music is already warming up, as you can hear. The last thing, very ultra-rare disease. Mark, you've been one of the leaders of a CEP290 CRISPR-Cas9 technology trial that obviously treats a population that's fairly small. Jean Bennett has designed and helped set up a gene therapy trial for LCA5. What do we do, and what do we do with ultra-rare disease? And also, Todd, please chime in. So maybe you first, Jean. How did you do that? LCA5 is really ultra-rare.
Yes, it's really challenging. We basically did this on a shoestring. We did not have a flow of money to pursue it. And so it was basically using leftover funds or donations that came in and people donating their time. There are definitely, so what, to add to the whole concern about cost, there is a huge barrier just to collect the preclinical data, which will allow you to go to clinical trial. So, for example, to develop a gene therapy for BBS1, Bardet-Biedl syndrome 1, you would need to do a formal preclinical toxicity study, as well as having a GMP grade vector carry out that study, then have biodistribution studies and histopathological studies, et cetera. Those are extraordinarily expensive. And I think there should be ways where we can do those studies on animals other than non-human primates, which cost, I don't know what the cost is right now. It's probably $15,000 a piece easily. And then carry them out for 90 days instead of 180 days. Things like that can save costs. The other big cost is in contract research organizations, which really I think, overcharge. You know, they will make a special assay to look for biodistribution. If you inject in the eye, will you actually see the vector in the testes or the liver or whatever? And these are assays which can be done in a laboratory. We do them ourselves, but when you pay somebody else to do them, it's easily log units higher in cost. And that price that pricing continues through the phase 1 clinical trial, where you have a organization monitoring data, and they're asking you, well, what forms should we make to record this data? You're giving them all the information, what forms to take, what data to collect, what the schedule is, how many days, and they're charging you for all of that.
A lot.
A lot. So there should be ways to basically coalesce those contract research organizations and make a universal one that helps everybody and cuts the cost for all these different trials.
So before we go to the payers, we need to reduce the cost. Mark, chime in, and then Todd, and then we'll wrap up.
So when I was in medical school, there was the rise of something called evidence-based medicine. And what I would really like to see is the rise of evidence-based regulation. Because, you know, I think many regulations get made and they just persist, you know, forever. And you know, Elon Musk, whether you love him or hate him, one of the rules he has is that every regulation has to have a name attributed to it as to who wrote it, so that you can go and ask them why it is and whether it should still exist. And I think we need to go back and kind of challenge some of these things and update them, because we could probably reduce a lot of costs, because there are probably things we don't necessarily need anymore.
Todd?
Yeah, so from the foundation's perspective, Bart and Mark know quite well, we have launched a very large natural history study chaired by Jose Sahel from the University of Pittsburgh, with really the sole purpose is to get more natural history data on the most rare inherited retinal disease, so that we can enable clinical research.
Avril, final thoughts before I say a final word?
I thought I was done. I was nice and relaxed there, Bart. What I would say is this is a very important question. These are all important questions. This is a very important discussion. And I would hope that it's the start of this discussion, and that we can take it further, because I'll just repeat, you know, opportunity lost for a community like ours, where we're up against the clock. Every day, we're losing more and more of our vision. That's a fact. Every day, things get more difficult, and that's the reality. But it's hope that keeps us going. And we believe, and I think what you're saying, Mark, is really important, because we know that there are some really exciting opportunities for treatment into the future. But we must, as a community, make sure that there's health preparedness for that there's an understanding of why we're valuable, why it is important for us to be considered in the healthcare system. And I think if we work together, we can find the solutions that we need. And remember that a human being, as I've said, no matter where you are in the world, they're the same. So all of our systems should be more aligned, and we have the power to work to that end. But it's going to take time, and it's going to take all of the voices in this room to make that happen.
Avril, I can't say it better, so I'll be very brief. I fully second the last words uttered by Avril here. Feel empowered as patients, that you have a voice. You have a voice through FFB, through Retina International. The fact that you're here and you're educating yourself on what is new must empower you to feel that you can make a change. You need to help us, but I can tell you, rest assured, that the group here at the front, with quite a few other people, are working also very hard for you. And we will not rest before we bring a leaner and cleaner system to what is development of innovative drugs for IRDs. You've heard a couple of ideas, snippets of ideas, as to how we can bring down the cost and make it much more accessible to produce these treatments. In the meantime, talk to people, say that it is important for you to keep the vision that you have, or at least bring down the speed of degeneration. And obviously, any improvement would be total luck. And you'd be very happy to have that. But have a voice. We need to, like Avril was so eloquently saying, we need to stop isolating ourselves in silos. Southeast Asia, Brazil, Europe, America, Canada, we're all the same. And we will work very hard to do our best to bring improvements to you. But in the meantime, don't forget that you also need to help us fight for vision. And with that, I am very positive. It's going to be hard work, but I've never been afraid of hard work, and I think nobody in the room is afraid of hard work. And with that, I want to thank you for your attention. Thank you for sticking here into the room with us for so long. Go have a drink and enjoy yourself.
I am going to have to run.
Listen,
so I'm not
thank
sticking
you
around.
so much for...
Not at all, no worries. Can I ask a very quick...
Thanks to... Hello. Hi, everyone. Just before you leave, my name is Manny Mudley, and I'm the Vice Chair of Retina International. And I've been asked to do a very quick vote of thanks at this point. Firstly, I'd just like to say that I am feeling so hopeful about the future after today's session, and I'm really grateful for that. And with that, I'd like to thank all the speakers, participants, chairs of the various sessions today for your captivating contributions. It's really amazing and wonderful as a patient to hear all of those very promising words. And I'm sure we are all looking forward to the continuation of the presentations tomorrow. So yeah, with that, let's give all the speakers a hearty round of applause, and thanks.
Thank you, man.